Welcome and good morning to Biostasis 2020. I would like to welcome all speakers and all attendees to this new annual Biostasis conference series that now we will host for the first time. My name is Emil Kensiora. I'm a trustee and the president at the European Biostasis Foundation, and I'll be your host today. First of all, we very much hope that next year at Biostasis 2020-21, which we will announce at the end of the conference, we will be able to meet in person and network in an easier setting together in Zurich in Switzerland. But of course, due to travel restrictions and the general situation right now, we decided that this year it makes more sense to host Biostasis 2020 online. Of course, there might be some technical difficulties. If there are any, please chat with us. We have a team ready that will fix all problems. And if we should have any larger problems, like the electricity going out or an internet outage or something like that, please just stay in the streams. Within 10 to 15 minutes, we'll change to our backup location and basically restart the stream. Now, first of all, I would like to very briefly introduce the European Biostasis Foundation. That's the right way to say it. It's a non-profit foundation based in Switzerland that promotes research, development, application, and also education in the field of biostasis. And this is why we're running this conference and started this conference series. The foundation was founded by 6 people. Half of which is from Germany, half of it is from Switzerland, all of which have been involved in biostasis in the field for many decades. Now, why have we decided to start a new conference series? The reasons is basically twofold. First of all, we believe that there is an annual conference required for this field. And when I say required, I mean there should be a place where the community can come together once a year, where scientists can discuss with each other, where the community can be strengthened, where networking can be done, and last but not least, also where the general population gets a better understanding of what biostasis is and why it might be interesting to them. And secondarily, in the past, this whole cryopreservation field and so on was very much focused on the US and very much driven forward in the US. And we believe that it makes sense that there's also a strong European presence, and biostasis in this conference series will be one of the pillars of that focus. But don't get me wrong, the Biostasis 2020 and 2021 and so on conference series is by no means the annual conferences of EBF, the European Biostasis Foundation. Quite the opposite. We would actually like to host this conference with all the other providers, with scientific institutes, with companies in that field together. So if anybody of you is interested in running an organization in this field, please reach out to us. And then from 2021 and 2022 on, we're more than happy to host this conference in a collaborative effort with other organizations together. So, before we get started, just a few housekeeping things. So we have two streams. One of them is biostasis.com/stream. The other one is biostasis.com/stream2. If you should have any technical difficulties with the main stream at /stream, please feel free to switch over to /stream2. And then, as I mentioned, if there's any larger technical issues, we will bring back the stream within around 15 minutes. A few more things. We're running this conference under the so-called Chatham House rules. This is a set of rules that should or is supposed to enable open communication without any potential later on repercussions. So the rules basically go as follows, and we know that they're not super easy in an online setting, but we will still ask you to respect them. First of all, you can report everything that is being said, but not who said it. Chatham House rules, please respect them. Last but not least, please ask as many questions as possible. In an online setting, it's always a bit difficult to have an interactive kind of experience. So please ask questions, and I'm going to forward them and ask them to the speakers, either after their talks or at the end of the conference, where we have a dedicated Q&A and panel session. Last but not least, as I mentioned, I would like to announce Biostasis 2021. It will be hosted in Zurich in Switzerland from October 22nd to the 24th. Next year in Zurich. Please save the date. Now, without further ado, I would like to introduce the first speaker. The first speaker is Dr. Raman Risco. Just as a quick introduction, Ramon studied physics at the University of Seville. He earned his PhD in quantum physics. Now he's a professor at the same university where, among other things, he studies cryobiology and cryobiotech. And his talk will be about the problems and solutions for organ cryopreservation. Ramon, please take it from here. Ramon, quick thing, you need to share your screen. There should be a large green button on the bottom of the stream where it says Share Screen. Do you see the button? Just click that button and then you should be able to... That starts to look correct.
In order to achieve the cryopreservation of an organ, you need three things. You need a uniform loading of the cryoprotectant inside it, you need a uniform cooling of your organ, and you need a uniform warming. Besides, two sets of boundary conditions that must be fulfilled. One is relative to the concentration of cryoprotectant. It should be high enough to avoid the formation of ice, and but of course low enough to avoid the toxicity. And the second is relative to the cooling and warming rates that usually need to be beyond certain threshold. To summarize, it is what engineers called a problem of heat and mass transfer. In order to overcome this problem, we have explored a set of fields that I will talk about. They are X-rays, magnetorological fluids, ultrasounds, time-varying electric fields, and mathematical modeling. The reason for starting this research in X-rays came from a couple of papers that I read. After reading that, I found that an efficient method for measuring the amount of cryoprotection during cooling was mandatory. One paper was from Greg Fahey on rabbit kidneys, and the other from David Peck on articular cartilage. At that time, there were not many works on exploring this measuring of the cryoprotectant. Maybe the closest was one by Barry Fuller by using magnetic resonance (MRI). However, soon I realized that the temporal and spatial precision was not enough for that, and besides that, probably the cost of the technology was high. So the first thing that we did was to explore bioimpedance. Here you have a mesenchymal stem cell growing in AP3D, so by putting a set of electrodes around outside and by looking what's happening in the boundaries, you try to figure out what's happening inside. However, the efficiency of of this technique is not very high, although probably it is very cheap. So there were not many places to look at. The remaining one was ultrasound, but probably was not a good idea. So finally, we end up with the last possibility that was X-rays. When you look at X-rays, there are three reasons why the beam is attenuated. One is the Rayleigh scattering, the other is the Compton scattering, and finally you have the photoelectric absorption. In the case of medical devices, they work at high energies here, so the Compton scattering is relevant, is dominant, and the attenuation in that case is proportional to the density, and that is why they can distinguish different parts in the body because different density. However, when you go to lower and lower energies, as you can see here, the photoelectric absorption becomes dominant. So because the photoelectric absorption goes with the fifth power of the atomic number, in the case of the DMSO, because the sulfur atoms has an atomic number of 35, that in the fifth power makes something like a billion, and mixed attenuation is a bigger times higher than that of the hydrogen. So with this idea in mind, we went to the X-ray device and we put glycerol, ethylene glycol, dimeso, propylene and water. And as you can see here, the results was what we foresaw. That was that the signal for the DMSO should be evident. We test how scalable this technique, and of course it was working in dependers, it was working in 20 ml cups, and not only for detecting the DMSO, but also for detecting the presence of ice. That is important. Ice is where the DMSO is not. So here you have an ice cube floating in 50% DMSO, and here you have something like 8 droplets of frozen water of 1 microliter inside a vitrified kidney. So the first application of this was, of course, in the field of slow freezing cryovials. That is very common. Here you have a piece of ovarian tissue in the typical slow freezing, and you can see the very nice patterns of the dendrites growing. And by doing that, you can control the slow freezing process. Not only that, but we understood why there is so big dispersion in the results among different centers, and the reason is that these patterns depends, their form, their structure depends critically on many parameters: the amount of seeding points that you are using, the amount of liquid that you put in your cryovial, the position of your tissue, of course on the cooling rate, or also on the seeding temperature. So these dependence made that only the same hands can reproduce exactly the same patterns. So only a very well trained people can do in an efficient way this ovarian tissue cryopreservation. Um, when when looking at that, this technique is was not relevant only for the cooling, but
Ramon,
also for
quick
the
thing.
loading
Could you
process
speak up a bit?
before the
Yes,
preservation,
can you hear
and
me?
also
Yeah, could you speak a bit
after the
louder?
cryopreservation. In this case, was ovarian
It is
tissue,
the
because
maximum.
Okay.
in that
Can you?
application
Yeah.
you
Okay.
can
If you can't,
detect
then it's fine.
also if
Put
there is
the
some
loudspeaker
remaining
of your computer higher.
Yeah.
DMSO
Okay.
inside
At the
Thank
your
you.
maximum.
sample
Thank you.
after washing, but before the transplant or not. You do not want to transplant your samples with DMSO inside. With this, the last application of this technology was for the detection of fractures. Here you have a vitrified kidney, and here you have a kidney that you put directly in liquid nitrogen without any care, so you can detect a bunch of fractures inside it. With these ideas in mind, we started to do what we wanted, that is to follow the approach of Farrand, but by using organs. Let me remember quite fast what he did. He put quite the caps of increasing concentrations of cryoprotectant at a lower and lower temperature, and with a piece of uterus, or rat uterus, he was jumping from one cap to the other, and finally he vitrified by following this liquidus curve. Here you have the what's happening in the control. This is for the slow freezing, the contractions, and these are the contractions in this case of this stepwise vitrification. So we build this setup in which the important part is this CT device. This is how it looks like in the lab. This is for cooling. This is dry nitrogen for avoiding condensation. This is the pump for PBS. This is the pump for the DMSO. This is the computer for controlling the pumps and the system. And in this setup, we soon realized that there was a missing piece, a missing part, that were the hands of a surgeon, in order to have an efficient way of taking off the organs from the animal and making the transplant. So we had to stop that research till we find someone able to do this very important part, that is the surgery. However, we were able, in fact, to follow the Farrand approach, but for ovarian tissue instead of the rat uterus. It was a nice collaboration with General Electric, with Peter Kilbright and John Morris, and with Cristiani Amorin for the ovarian tissue. We modified one of these Stirling motors. We put here a robotic arm, and we can jump from one cap to the other in a controlled way. So all this assisted with the CT produce this vitrification of the tissue. The next topic that we explored were the use of magnetorheological fluids. These are ferrofluids. Imagine that you put one fluid in one of these syringes and you start to push, so you can move very easily the fluid from one syringe to the other one. However, as soon as you put an external magnetic field, the liquid becomes stuck, becomes solid. The viscosity rises orders of magnitude. This liquid is sold by this company who make these special dampers. The idea was to put the liquid inside an organ and to control the viscosity by an external magnetic field, and it's similar in certain sense to what Peter Kilbright wants to do with a non-Newtonian fluid, but in this case he is trying to control the viscosity with an external surgent pressure. However, when we put this liquid inside an organ, we try a few times. Although the loading was not that difficult, the washing process was for us almost really impossible. It was really challenging to wash the organ. To wash the organ out of this fluid or magnetic particles. The thing is that I have to give a thesis to these poor students, so we produce this rewarming of samples with microwave radiation by means of magnetorheological fluids for its application in cryopreservation. And the thing is that there are two reasons why these ferrofluids becomes very hot when you put in an alternating electric and magnetic fields. That is the case of a microwave. There are two reasons associated with the electric fields, and there are two reasons associated with the magnetic field. So if you put this inside the microwave oven, this is the warming rate that you have when you are working without this ferrofluid, and this is the warming rate that you have when you are working with the ferrofluid. So in the case of cryovials or ependos, the washing was not that difficult, as you can see. So maybe it has an applications in the case of slow freezing in cryovials. Now the work of John Bishop by using this fluid but controlling the magnetic field with an external coil is doing a much better game, and I'm quite sure that they are in the right way, in the right path for rewarming those samples by using this control by these external magnetic fields. The next thing that we were exploring, well, at the same time, in fact, were the ultrasounds. The rationale for this came from a couple of papers that were surprising. They are a bit old, but the results were amazing. They claim that treating something like 1 liter of water and leaving the water with ultrasounds and leaving the water on a shelf after 1 month, the water was very difficult to freeze. That result was surprising. More surprising was the fact that after those papers, you cannot find anything in the literature. We make a very deep searching, and there was nothing. And also a characteristic of these works is that they only give some quantitative information. They do not give any qualitative information. Only they tell about the frequency of the ultrasounds, but they didn't give information about the power or about the kind of water they were using, or for example about the walls of the container that they were using. Nothing at all. So we decided to do it in a more controllable way and see what's happening. So we built this setup. This is a flux of water. We put here a layer of Peltier plates and a second layer of Peltier plates, but playing the role of thermocouples. Here you put the droplets of water. This is the ultrasound transducer with the hope that treat the droplet with these ultrasounds. This is how it looks like, and here you have another view. And this is the last view with the ultrasound device here. So when the temperature starts to low, each time water droplet freeze, release the latent heat, and you can detect this release of the latent heat. I mean the frozen of the droplets by looking at what's happening at the Peltier plates on top. So after doing something like tens of thousands of droplets, with this device you can have a bunch of statistics. We couldn't find any effect. So this explains part of the story why there were not papers after those in the 47, 48. And it is because maybe for us, in this very controllable way, there was no effect. So anyway, we were quite sure that this author saw something, and thinking what could be, we end up with a very plausible conclusion. What they were doing with the ultrasounds were degasifying the water. Just by treating with the ultrasound, you remove the gas, so you remove the nucleation sites. In the case of small nucleation sites for the ice, we found that in a different parallel work that was working with degasification by using a vacuum pump. In that case, by using this degasification in the vacuum by the vacuum pump, with the goal of avoiding fractures, we found that this degasification was very effective in avoiding the fractures, and the same degasification could be done by using those ultrasounds. This exploration of the ultrasounds can have a second application, that is for the warming of the organs. This paper is about to be published it is in press in ultrasonics, and here you have an ovary of this size, and we are still making these simulations. So we pass from minus 114 to minus 20 degrees in a very uniform and quick enough way. Here you have this uniform region of warming, so you can see what's happening in the x-axis, in the y-axis, and in the z-axis, and you can see that the warming is very uniform. The next thing I would like to talk about is time varying electric field for cooling. This technology, as it is now, it can be applied only for gas, but maybe someone find sometime a possibility to doing it for solid or for liquids. The idea is the following. Imagine you have a bunch of water molecules in gas state in a container. They are dipoles, and you suddenly open one of the walls, so at the end of the day they start escaping, and the fastest will go in the front and the slower will go in the rear. So the dispersion in these velocities gives you, by following this Maxwell-Boltzmann statistic, gives you this dispersion, this width, gives you an idea of the temperature of this thermal beam. So now, if you put a couple of metallic plates in between, nothing happens, and nothing happens also if you put an electric field, because the molecules are neutrals, although they are dipoles, but there are some boundary effects here, because here you have a zero field, so there is an inward force due to this gradient of the electric field that is proportional to the dipole moment of the water molecules. So if you apply a higher force to the molecules that are in the front and a lower force to the molecules that are in the back, just by reducing the electric field inside at the same time that the molecules are escaping, then at the end of the day all the molecules will have almost the same velocity, and in that case you have reduced the width of your Gaussian, and you have cooled your beam. So we made all these mechanical calculation for computing the Stark effect in the case of water molecules, something like a mole of molecules passing from room temperature to 55 kelvins, and this cooling passing from this peak velocity to this peak velocity happens in something like 1 millisecond. So the cooling is really fast. However, as I said, for the moment it's only applicable to gas state, maybe I think because maybe mainly because you need this correlation between the field of positions and the field of velocity in order to apply the right electric field strength to each molecule, and maybe thinking in forces like Coriolis forces or magnetic forces that depends on the velocity, you can have this correlation between this position and velocity field. Finally, I would like to stress the role of the mathematical models, not only for the slow freezing but also for vitrification. In the case of slow freezing, what we did was exploring not only the typical linear profile but also quadratic or exponential profiles. As you can see, there are differences in the degree of supercooling depending mainly on the cooling rate, higher for intermediate cooling rate, and also exploring non-ideal solution models, because sodium chloride is a strong electrolyte, so deviation for the ideal case of Peter Meixer are expected. So this deviation can be up to 18% depending on the cooling rate and also on the temperature. And for in the case of vitrification of human oocytes and embryos, what we did was looking at these 10 minutes that the sample should remain in the equilibration solution. So we realized that in the first 30 seconds up to 90% of the cryoprotectant has already entered, so the rest of the time probably was producing some damage mainly. So it was enough with 1 minute, and this model have the feedback from the experiment. So we have reviewed all these aspects, and I have to give my thanks to my students and of course to my collaborators, now friends that I have met in this journey. Thank you very much and congratulations again for the organization of this very interesting meeting. Thank you very much.
Ramon, thanks a lot for the talk. Due to the time, we will only have time for one question. The question is, what experiments are planned for the future? Like in a 1 year or 3 year horizon, what are you doing next?
In this journey. Thank you very much and congratulations. Well, we hope to go to organs. As I said, that is a missing part in our setup. We need the help of a surgeon. So as soon as we find someone able to take the organs of the animal and to transplant them, we will start with probably kidneys. That is the goal, kidneys and hearts.
Perfect. Thank you so much. For more questions, as I mentioned in the introduction, we will have a dedicated Q&A and panel session at the very end of the conference. And Ramon, hopefully we can welcome there again to answer more questions. For now, thanks a lot.
Thank you.
As a quick note to all the attendees, if you ask your questions in the Q&A tab, then you can actually vote up and down the questions, which questions you would want to have answered with the highest priority, and then that question we will pick. For now, I would like to introduce our second speaker, Dr. Roman Bauer. Roman, hi. Nice to meet you again. Roman did his doctoral degree at the Institute of Neuroinformatics at the University of Zurich and is currently a lecturer at the University of Computer Science or in Department of Computer Science at the University of Surrey. His talk will be about computational methods in cryopreservation. Roman, please share your screen and get ready.
Thank you, Emil. All right. Hello, everybody. Welcome to my presentation. First of all, I would like to thank the organizers for having me here and for, of course, organizing this great event. For me personally, it's a little bit unfortunate to not have the meeting in Zurich, my birth city, actually. But of course, online meetings have many advantages too, and I'm looking forward to the meeting next year in Zurich. So in this presentation, I will talk about some of the research we are doing in my lab involving neural cryopreservation, including also usage of tool sets that are computational, so computational modeling. And I would like to start this talk by highlighting a very recently published paper. This was published actually this month, early in October. And in this paper, the authors have investigated brain tissue from a victim that is almost 2,000 years that was a victim during a volcanic eruption almost 2,000 years ago. And the authors essentially study and analyze brain tissue that was vitrified during this eruption because of the ash that was very hot, and then afterwards, when it cooled down quickly, some parts of the brain essentially got vitrified. And they analyzed this brain tissue, and they could show essentially that the structure of the neurons, the axonal trees, were well preserved. Here you see some electron microscopy images where even detailed individual segments of axons were shown to be well preserved. And so arguably this is the best preserved human neural tissue in archaeology. Nevertheless, because there was a lot of damage, I mean, these were usually small parts of the brain and cortex. Nevertheless, most people would agree with me. I think if I state that probably most of the memories and the information and the personality, essentially, the information on the memories is not anymore present in this brain. And so one question that arises is, what are the aspects, what are the factors that are essential in order to preserve the information that is in the brain? And one crucial factor are for sure synapses. So synapses are the connections between individual neurons. They are involved in learning and maintenance of memories. But then there are also other factors, like, for example, the distribution of receptors, which allow for neurotransmitters to impact on the electrical activity of neurons. We also know that glial cells play a huge role in neural function. So there are actually more glial cells in the brain than there are neurons. And the interactions between glial cells and neurons have been shown to be very important. We also know that excitability of neurons plays a big role. So a former student of mine, Sarah Soldado Magraner, recently published some work where the authors show that neurons change their excitability depending on their inputs, and this can last for a long time. And so this is also an essential feature of neural memories. And another example is so-called ephaptic coupling. So this is actually a way for neurons to communicate without synapses. Essentially, the neurons create electromagnetic fields that influence the activity of other neurons. And also this has been shown to play a role in the workings of the brain. And so the point that I'm trying to make here is essentially that the neural tissues are extremely complex. They have a lot of components that are important in the functioning of the brain. And it is currently yet an open question whether one can cryopreserve mammalian neural tissues while maintaining the information that is required for it to work. And so this is actually something, a question that we address in our research, and we particularly focus on the retina. So the retina is a neural tissue at the rear of the eye, and it is a good place to essentially focus on this research question because it's peripheral, so it's easy to access. So one can quite easily obtain this tissue, for example, from the mouse and do experiments with it. It's also interesting because it is structurally quite similar to the cortex in the brain. So the cortex also has multiple layers of different cell types, and the structure of the retina is quite similar to it. And also the one big advantage of the retina is that one can essentially measure the electrical activity of the retina quite easily using, for instance, so-called multi-electrode arrays. So multi-electrode arrays allow to measure spikes in the retina, and so they can essentially allow to see if the retina is functional, whether it works well or not. And we have recently actually published some work where we study the retina and how it changes during development in the human species. And there are many uses of this. So if one could cryopreserve the retina, then there would be many uses of this. For instance, pharmaceutical research would benefit from this, because if one can cryopreserve retinal tissue, then one can essentially bank a large number of tissues and do drug testing with those. Currently, one is quite restrained by the need to do these experiments very quickly. But if one could cryopreserve them, then one could essentially scale up pharmaceutical research with neural tissues. And also for generally for biobanking, this would be good. So one could transplant certain parts of tissues. And ultimately, of course, also brain cryopreservation would benefit from insights gained by cryopreserving the retina. So my postdoc, Dr. Sanja Bojic, has been looking from an experimental point of view into the cryopreservation of the retina. And she has done a lot of hard and great work. She has looked at the retina, at the quality of the anatomy of the retina after using different kinds of cryopreservation protocols. So we tested a large number of different ways of how one cryopreserves tissues. And here you see an example of a mouse retina where different colors indicate different cell types in the retina. And also you here see essentially the layered structure of the retina is very well preserved. And so this is one example of how one can assess essentially whether the retinal structure is well preserved after thawing. These are some other examples that Sanja made using other techniques. This is a so-called H&E stain, where again one can see that the structure of the layers in the control case. So the control case is the non-cryopreserved case, and here in the experimental case where we cool down to very low temperatures, you can see that after thawing we can still obtain quite good structure, anatomical structure. And also then we looked in great detail using electron microscopy images at those tissues. And again, also here, I think this is quite promising. Anatomically speaking, this looks very good. But of course, this is not yet a proof that it works very well. We have to do some further experiments. In particular, we would like to, as I mentioned before, to look at electrical activity and check, do those retinas that were frozen and thawed, do they still give rise to similar activity patterns as we had before? Because that will be essentially a proof that the whole architecture and the whole integrity and quality of the tissue is high. Which actually brings me to an important point, which is that cryopreservation is very complex. So if one wants to cryopreserve tissues, one needs to first cool it down, of course, and there are different ways of how one cools it down. One can use, for example, different cooling rates. One can cool it very quickly or slowly. One can use different kinds of cryoprotective agents and different concentrations of those cryoprotective agents. And also for the thawing, one has a similar challenge as for the cooling. How do you determine the optimal way of thawing? And this is really a complex problem because, for instance, if one uses high concentrations of cryoprotective agents, then one, of course, reduces the injury from freezing, but one increases the toxicity. So those agents are often very, very toxic. And so one has to find a trade-off. And currently this is done mainly based on a trial and error basis. And what we thought is, why not bring in a kind of integrated computational approach that allows to computationally model what is going on exactly in the tissue during cooling and during thawing in order to better optimize those cryopreservation protocols. And a few years ago, I actually started a collaboration with CERN and other institutions. So it has grown significantly since the start. And we have now created the Biodynamo project. And if you're interested, you can go to the website. We have created a software that is open source and that allows to simulate different kinds of biological tissues, so complex biological tissues, including like cancers or neural tissues. And we decided in this project to essentially use Biodynamo for cryopreservation. So my PhD student Jack is working on extending Biodynamo in order to simulate how temperature dynamics affect tissues and multicellular systems. And this package, which is called Cryodynamo, and which we are currently finalizing, allows to essentially simulate what is going on during the whole process? How are cells dehydrating during the process of cryopreservation? How are they their mechanical properties changing? And here you see an example of this. So here you see how the concentration of water changes over time during a decrease of the temperature. So you can see that the color becomes lower, and we have a 3D dimensionality here, so we can really model at different spatial coordinates how things change dynamically. So this has many applications. For instance, one application that we looked into is for the optimization of cryopreservation of umbilical cord blood. So it is known that the volume of the samples that one cryopreserves has an impact on when ice crystals form. So ice nucleation the ice nucleation temperature is essentially when ice crystals start forming. And we could simulate with cryodynamo quite well, in agreement with experimental measurements, when this happens, depending on the volume of the sample that we're looking at. And based on this kind of modeling approach, one can therefore optimize and improve the quality of such samples. And this is just one example of an application of cryodynamo. Then also we looked into the cooling rate optimization. So it is known, for instance, from experimental work that different cooling rates affect the quality of the cells after cryopreservation. So in this case, Jurkat cells had the best quality at about 2.5, at the cooling rate of about 2.5 degrees Celsius per minute. And we could essentially, without having to do a lot of experimental work, we could create this similar curve, which allows to optimize the cooling rates just computationally. And so you can see this value of using computational techniques that help people in practice to optimize cryopreservation. And of course, ultimately, we would like to apply this also for neural tissue cryopreservation and combine this with all the results we are now collecting with Sanya's great work. So I want to summarize that I think the retina is a great tissue to look at if one wants to better understand how cortical tissue and brain tissue in cortex is essentially where higher order thinking happens, where planning happens. And by looking at the retina and how it behaves during cryopreservation, we can get a much better idea of how cryopreservation affects the brain and cortex in particular. And we have made some important steps in this direction, but we need to do more. We need to know better how is exactly the structure affected. How are those different factors that I mentioned at the beginning, how do they change with cryopreservation? Because only that will allow us to really say whether it works or not, whether the whole the information that is contained in the tissue really is preserved with a good quality. And so we need to do more research there for sure. We have done some important steps towards a computational framework that can help people that do experiments to guide their protocols, to be able to better know what kind of parameters they should choose. And I think this also shows more and more how modern computational approaches can help biomedical research and cryopreservation research in the future. So I would like to thank my colleagues, of course. There are too many to all include in a slide, but there have been, this research is the product of a wide range of people that were involved in one way or another. And I want to thank you for your attention. Thank you.
Roman, great talk. Thanks a lot. There are a few questions from the audience, and since we have like 2, 3, 4 minutes, I would run through a few. Right. So first question is, could it ever be possible in the future to recover not the consciousness, but at least some memory fragments from partially vitrified brains of long dead people, like persons from before for AD, like the example is AD 79.
Right. So, based on this paper, I'm not an expert in archaeological data, of course, but based on this data that I presented, that I showed at the very beginning from this study that was published this October, I think the volumes that were preserved were really too small. I think in these cases, I think there's not enough information to have a meaningful representation of complex memories maintained, because memories are also stored in the brain in a very distributed fashion. So we will need to have information from different parts of the brain. And I think in this case, this is probably, there's too much injury, I think.
Right. I actually have a good amount of more questions, so I'm running through a few. It was someone's understanding that retinal tissue can already be regularly cryopreserved and is viable. What's your opinion on that?
Well, it depends. So if it's immature, that's true. So there are papers, about 2 or 3 papers that show that immature retinal tissue can be cryopreserved, but as soon as you mature them, then you have serious problems. I mean, I work with people that grow retinal tissue from human embryonic stem cells, and currently it's not really possible to preserve in the long term those tissues.
Okay, cool. Then the next one is, how close, in your opinion, are we for using these advances to help preserve donated human organs?
Yes, so that's a good question. I mean, this is, of course, ongoing research. I think it will take time in order to scale up those techniques and approaches. I could not give a clear time frame. I think, especially now, also because of the economic situations, I think this will slow down. I think research generally for many types of research. So I think if things go very well, we might be able to achieve good success for certain types of volumes, because that's one of the big bottlenecks. There is the volume, of course. And so I think this will take some time, and I hope, of course, I very much hope that there will be funding available to do this direction of research more.
Perfect. And then, due to time, the last question, unfortunately, which is also very highly voted. The approach using cryodynamo is fascinating. How did you obtain the experimental data linking simulation parameters, temperature and osmolarity, to cell viability?
So we essentially have 2 different types of data. One is the published data. So one of them I cited in my presentation just now, where we looked at Jurkat cells. So there is data out there in the public space. And then also we are using currently data that we're collecting in our lab. Sanya has done some great work, and we have already collected some nice data that will help inform this data. The modeling, but of course we need more data. You can never have enough data as a computational biologist. So if you have data that you think would be interesting to use for such a computational approach to inform, please get in touch. We would be very, very interested to look into this.
Perfect. Thanks a lot, Roman. See you later at the Q&A. Just a quick note for all the attendees: if you ask questions, please always put to which speaker this question relates to, because we will most likely not have time to answer every question directly after the talk, and so we're collecting them, and then we'll ask the most rated or highest vote rated questions at the very end at the Q&A and panel session. And it would help us, of course, if you know to which speaker this relates to. So next is the upcoming speaker is Dr. João Pedro de Magalhães. Joao, hi. Unfortunately, this was the wrong intro video, but I think
that's okay. Ghent is a beautiful city.
You're at the right place.
I mean, frankly, I would prefer to be in Ghent than in Liverpool. It's lovely. I've been there many times when I lived in Belgium. So
right, awesome. Quick introduction: Joao is a scientist and futurist. After doing his PhD and after his training at the Harvard Medical School, he's now a professor at the University of Liverpool. In England, not in Ghent. He's also a founder of Magellan Science Limited, which focuses on consulting on data analysis and longevity science. Very fitting to this time, his talk is titled "Winter is Coming: The Future of Cryopreservation." Joao, please take it from here.
Thank you. Well, thank you very much, Emil, and thank you for the invitation, and thank you for putting together this conference. I mean, obviously, again, I would prefer to be in Zurich as well, actually giving this talk and meeting you all personally, which is unfortunately not possible, but hopefully next year. And so, I mean, I'll start with a brief introduction. I know some of the people in the chat know who I am already, but I'll start with a brief introduction to those not familiar with my work and my ideas. And then I'll talk about a couple of different projects we've done, a little bit of work we've done in cryobiology, and also in more in the context also of cryonics. So I guess since when I was a very young child, this is me as a child, I realized that everybody dies, and we've got to do something about it. The idea of death was just like this, eternal oblivion. I mean, I'm not religious. I don't think there is an afterlife. I think that when you die is just like a eternal prison. So really the concept, I would say, scared me. It still scares me today, the idea of dying. So I wanted to do something about it. I wanted to, let's try to fix it in some way. And so I started to focus on aging. So my day job, so to speak, and most of my work focuses on aging. So as Emil mentioned, I'm a professor at the University of Liverpool. I lead the Integrative Genomics of Aging group. And we do various levels of research on aging, combining both computational approaches and experimental work. So this involves systems biology, bioinformatics, machine learning, AI, but we also do dietary manipulations of aging in animal models, cellular models. We study cell senescence, and we do genetics of longevity in human cohorts and populations. So we do a large variety of work. I mean, if you're interested in our work, obviously, please visit our lab website. So my idea, I guess, when I started on it, I became a scientist not because I wanted to spend my days applying for grants and writing papers, but because I wanted to do something. I wanted to, make a change, in particular at the level of longevity. I wanted to extend our lifespan. I wanted to cure aging. My dream from when I was a child is I want to cure aging. That's what I want to do in my career. And that's why I developed my academic research in this area. And well, as Emil mentioned, I'm also doing some work in biotech, which I won't go into detail today. I do think that once you start reading into it, although there's been a lot of advances in the field of aging, for example, in pharmacological manipulations of aging and genetics of aging, I don't think we're going to, or I think it's very unlikely, I wouldn't say it's impossible, but I'd say it's very unlikely we're going to cure aging in my lifetime. I think it's just a too complicated process, and our understanding of biology is too limited for us to cure aging in my lifetime. I mean, I wrote this paper some years back in rejuvenation research on the prospects for curing aging that goes into, the more detailed discussion of this, and of course happy to discuss in more detail later on. The point was, okay, we're not going to cure aging. If you're not going to cure aging, then that means we're going to die. As between eternal oblivion and which is death, I mean, is there another choice? And that's why I became interested in cryopreservation and cryonics, because of course, cryonics, again, without going into a lot of detail, there will be other talks on cryonics. It gives a possibility of future life. I think there's limitations of cryonics and cryopreservation that I'll mention in a minute, but between eternal oblivion and death and a possible future life, cryonics gives that possibility, even though, I think there's a lot of arguments, whether that's a high possibility or a very low possibility. I would argue it's actually a very low possibility at the moment. The reason why I say it's a low possibility that we're going to have a future life if an individual is cryopreserved is that there's a lot of scientific problems to it. There's very limited research in cryonics per se. I would say in cryopreservation and cryobiology itself, there's quite limited research at the moment. And in cryonics, there's virtually no academic research. There's some anecdotal evidence, there's some small studies, but very limited. So, for example, when we look at how much brain damage occurs during cryonics, we have very little data. I mean, Ramon actually presented some data, for example, and there is some data on structural integrity. We know fractures occur and so on, but we know little about molecular damage. I mean, I'm actually a cell biologist by training. We know very little about cellular survival, about molecular damage that occurs. There's something that I would suspect it's very high because I've frozen small amount of cells. I've cryopreserved small tissues. So based on that, extrapolating to cryonics, I would assume there's extensive damage, but I've never looked into it. And to my knowledge, there is no strong data on this. But so I think there's still a lot of question marks and a lot of problems regarding cryonics as a scientific approach. So what can we do at the moment? So I guess what I've been trying to do, on one hand, I've been trying to do some research on cryobiology. I mentioned a couple of examples of what we've been trying to do. And this is in spite of the fact that there's very little funding in the UK. I would say worldwide, there's little funding for cryobiology research. And there also seems to be little interest from students. So for example, my lab, we have quite a few graduate students, but they're mostly interested in age-related diseases like cancer, or they're interested in aging and longevity. It's relatively rare to have students that are interested in cryobiology and cryopreservation, although there are exceptions, and I'll mention one in a little bit. So in spite of these limitations of not having people, pairs of hands to do experiments and funds, we've been trying to push for some research into cryobiology. So I'll show you about two papers we've been involved in recently. So this one I won't go into a lot of detail because we published this a few years back. So basically this was a collaboration with Greg Fahy and others. But the goal of this work was to study cryoprotectant toxicity in vascular endothelial cells, which are important. So they're important cells in the vascular system. So they'll be crucial for cryopreservation of organs and for cryonics. And I mean, very succinctly, what we showed was that endothelial cells are resistant to ethylene glycol. They recover well from exposure to ethylene glycol. And then we also did some gene expression profiling using iThroughput approaches that I won't go into detail, trying to gather some insights into what could be causing the toxicity from cryoprotectant as well, ethylene glycol in particular. I mean, the paper we published already a few years back, so again, I won't go into details, but all the results are available online. And I mean, and this work was funded by Longevity. Now, more recently, we focused also on Drosophila. I'm not a biologist, Drosophila biologist, but we have several collaborators here. So for those who are not familiar, so Drosophila are these tiny fruit flies. They're widely used in genetics and biology studies. They have a limitation, which is you cannot cryopreserve them. So you can cryopreserve mouse embryos, for instance. You can cryopreserve C. elegans, roundworms, which we also use as a model. But it is not possible to cryopreserve fruit flies. Well, there's a paper written several years ago that says it's possible, but it's not been reproduced. I would stand by, again, there's some details I'm skipping. Happy to discuss in more detail, but I would stand by my statement that it is not possible to cryopreserve fruit flies, which is a big limitation actually for biologists to use this model. So what we tried to do was we tried to make advances in cryopreserving Drosophila larvae, which you see here on the right. And for this, we employed a permeabilization solution of, well, 10% aptame and 9% limonene. The timing of this is also quite important, and it's available in the paper that is now in press. But the point is that we created this permeabilization solution. And when you expose Drosophila to it, so this on the left here, you'll have larvae, Drosophila larvae. These are live and these are dead ones. And these are basically permeabilized with a dye, with a fluorescence dye. And as you can see, there's a very clear difference that when you permeabilize them before you put a dye in, the animals take up a lot more of the dye. So it is working in making these animals uptake the dye. And I mean, you can do it from a more quantitative perspective as well. So you can look at controls, which I think is about 7 or 8% of the animals, 7 or 8% uptake of the dye, compared to I think something like 70% when you permeabilize them. So we show that this is working in permeabilizing this Drosophila, live Drosophila. And then what we wanted to do, of course, is we wanted to advance in the topic of cryopreservation. I mean, we did only one experiment for reasons that I'll come back in a minute, which was basically to expose the Drosophila larvae to a high concentration of cryoprotectants, namely ethylene glycol and DMSO. So what you see here is survival of the animals. So if you don't permeabilize them, all of them survive, because the ethylene glycol, the cryoprotectants don't go inside the larvae. If you permeabilize them without the cryoprotectants, most of them survive. But if you permeabilize them with a toxic dosage of cryoprotectants, then the vast majority of them die, which means that the cryoprotectants are going inside the animals. Now, these results are, so as I said, this is a paper now that is in press in the journal Fly, so it's available online and all results are available. This was work done by Andy Murray, who was a graduate student, a master's student in my lab, who was interested, so he was one of those exceptions, and one of those exceptions, a person who was interested in cryopreservation and was able to drive this work forward. As it was only a master project, so relatively short time, we were unable to continue it beyond this. So our goal next, if we have a student, so Alex is now a PhD student in Warwick, because I didn't have funds to for a project on cryopreservation. So he's not in my lab anymore. But if we have funds to continue this, the next step would be to try to cryopreserve the Drosophila larvae, as we can already show that they can uptake the cryoprotectants. So that's the work we've done so far. We're trying to push this. I mean, we just applied for a small project with Roman, who the previous speaker, on applying machine learning to develop cryoprotection methods for cryopreservation methods for the retina. So we're trying to push this. As I said, there are limitations in terms of funding and pairs of hands. So the other thing that we've been working on, and this is also with Roman and quite a few other collaborators, some of them are here in the conference, we wrote this quite big review of cryobiology and cryopreservation. The title is the same title of my talk, Winter is Coming, the Future of Cryopreservation. But the point of this review is, on one hand, to highlight the benefits of developing improve or enhancing the protocols for cryopreservation at the level of organ banking for organ transplants. That would be fantastic if we could cryopreserve whole organs, for the storage of engineered tissues and stem cells, and for, I'd say broadly speaking, for regenerative medicine. So we want to highlight that. We also talk about emergency medicine. I mean, there's some interest even from the Department of Defense of the United States in cryopreservation, and maybe even for space travel. So it's the paper aims to highlight this importance of developing research in cryobiology and cryopreservation, hoping to foster more research in this area. And we also discussed some of the limitations and challenges of current methods. It's quite a big paper. If anyone is interested, it's currently submitted for publication. If anyone's interested, feel free to drop me an email and I will send it to you. I mean, we're looking for feedback and opinions from others. I mean, ultimately make the argument that, you know, we need more research into cryopreservation. We need more research into cryobiology for lots of medical reasons. If this advances occur, then we will reach the point of having a method for human medical biostasis or cryonics. So that's the point of our argument. So we want to go from, so this would be traditional freezing, with what develops the ice crystals, and this is vitrification. This was another figure that Alex Murray, who's also a co-author in the article, made. So that's something else we are working on. As I said, we're Roman and quite a few others. Jose Cordero is also in this conference, and a few others that are here today. The last thing I will mention is, this network I created a few years ago called the UK Cryopreservation and UK Cryonics and Cryopreservation Research Network. I mean, the goal is to advance research in cryobiology, including cryonics. I mean, focuses in UK, basically because I am located in UK. So that's pretty much the only reason, but it's actually composed of academics from all around the world. And really the goal is international. It's about fostering research in cryobiology and cryopreservation, including cryonics. So see what we can do to advance research in this area. I think one of the problems we have in cryonics is, I mentioned this if you see me give talks on cryonics in the past, I think one of the problems we have is the level of public perception, or PR. I think we have a problem that there is a lot of skepticism from the public and for medical professions regarding cryonics. So really one of the keys to improve reputation of cryonics. So if there are news stories, then we can engage with the media on that. We can make statements. We can engage with other medical professionals. It's something I've done when they're critical of cryonics. I'll send them an email, or I'll tweet them. I'll contact them and say, listen, why are you being so critical of cryonics? I mean, what are your scientific reasons? And a lot of times, I would say most of the times, there are no, because we don't have hard data on cryonics, there's no really strong scientific reasons for it. It's more, preconceived notions. So really trying to show that what we want ultimately is for medical biostasis, is that to show that this is a high-tech, professional, and reliable approach. Now, I know we're not there yet, but that's where we want to go. And so that is the role of this UK Cryonics and Cryopreservation Research Network. And if anyone's interested in joining, again, feel free to contact me. So I guess what we want to go with, my goal in all of this is to create this bridge to the future that is medical biostasis. So develop the methods and the approaches that will allow us to have safe, efficient, reliable medical biostasis, which I think would be a revolutionary technology. And again, convince also the medical professionals and biomedical researchers that, this is a lofty goal. This is something we should aspire to, because it would have tremendous medical benefits. So, so in summary, I've told you that, well, I think many of you are convinced that death is really, really, really bad. It's. I know there's people who will try to convince you otherwise, but really, death is the worst thing that can happen. Really, it is, and it is this eternal oblivion that I cannot even rationalize it. It would be wonderful if we could cure aging, but I don't think it will happen in our lifetimes. So if we cannot cure aging in our lifetimes, then we will die within our lifetimes. And so cryonics is this backup plan. Now, it still has a lot of technical limitations. There's a lot of problems. There's still a lot that we don't know, because there's very limited research carried out in cryonics. But we can try to make progress. We can try to make progress in cryopreservation. I showed you our first protocol for permeabilization of live Drosophila larvae, which I think is a first step into cryopreserving Drosophila. I told you about our review of the cryobiology field. As I said, if anyone is interested, I'm happy to send it to you. And again, it's something else we're doing to gather attention and raise awareness and really show that this is a worthwhile field that we need to invest more into. Oops. And lastly, I told you about our UK Cryonics Research Network to foster research in this area. So with this, I thank you very much for your time and attention. I mean, this is my lab. As I said, we mostly focus on aging, but I should highlight Andy here. This was a photo before the lockdown. This is, I think, from last year. So this is Andy here, who did Andy Murray, who did the experiments in Drosophila I told you about. And so thank you very much for your time and attention.
Joe, great. There are a few questions that I quickly would go through. So first of all, you kind of already discussed it a bit. Is there any update on the funding from US agencies on DARPA and others, both for cryopreservation in general, for soldiers and astronauts? How are they moving ahead with these funding?
So in UK, that's very limited. There is some funding in the US. So I know, I think people from the US, I think Greg Fahy is speaking later today. I think they'll be better able to update you on that. In the UK, there's very limited funding for cryobiology. I would say there's actually very few groups working on cryobiology. I mean Roman is in the UK. So he's one of the very few people working on cryobiology in the UK. So I'd say it's very limited at the moment in the UK. I think what we're trying to do, so for example, in this project with Roman is, okay, so I do a lot of machine learning and do a lot of bioinformatics. So, machine learning is quite a hype topic at the moment. So, can we sell a project on applying machine learning to cryopreservation? Maybe that will, maybe that will be sufficiently interested to be funded. So we'll see. Fingers crossed on that. But that's what I've been trying to go. Instead of a project just on cryobiology, bring in some other more sexy element, like machine learning is very sexy at the moment.
Right. Awesome. Yeah. Then the question that kind of goes, I guess, towards your work that you do with the outreach program in the UK. Mm-hmm. Do you think that cryopreservation can be a mass market thing, like what I hearing in the UK and from the people that you talk to?
That's a good question. A mass market. Well, I don't think it will be, incredibly popular like, machine learning. But certainly I do think there's a lot of room for it to grow. It's about making it mainstream. I guess that's the question. If you can have, people who have a disease, or we go in for, emergency medicine would be a good example, or battlefield medicine. If it becomes mainstream that when you have your life is in danger because of a gun wound or a disease, and you're just cryopreserved, then yes, that will become accepted and it will become adopted by the masses. But we're still quite far from that point, unfortunately. I do think that, there's a lot of very strange medical procedures that are done already. So I don't think cryopreservation in that sense is stranger or than some things that are already done. Open heart surgery it's kind of a pretty incredible procedure, if you ask me, for example, and it's done regularly. So it's a question of, one, having the biomedical advances that show that this is reliable. And then at the level of public outreach and public communications, that showing that this is a worthwhile endeavor and that, it's backed by science and we can do it. And yes, then and at that point, it will be much more widely accepted.
Right. Thank you for that. Two more questions. First one being, do you think that research of Arctic ecosystems can provide valuable insights to advanced cryopreservation? I guess talking about animals that cryopreserve.
Yes, I think so. So yes, there has been some work on that. My lab hasn't worked on it specifically, but we've done, I've done some work on Arctic species actually, but not in a context of cryopreservation, more in context of longevity, because species on lower temperatures, they tend to be longer. So we've done some work on that. I would say yes, but the caveat or the limitation is that biology is complicated. So sometimes there are complicated pathways involved. Other times I think there's been some studies showing that particular animals that can secrete some sugars into their bloodstream to protect against cryopreservation. So I would say yes, it's absolutely it's a topic that would be very worthwhile to explore further, because we can learn a lot from biology. There's lots of examples of innovations in medicine that we learn from animals. We learn from evolution. We learn from the natural world. And I mean, just if you're interested in more details, I actually published a review on this in EMBL Report in 2015, which briefly mentions this cryopreservation as well. So yes, absolutely.
Right, and then last questions for now. Could you elaborate on the arguments why fruit flies can or cannot be cryopreserved?
So that's a good question. I've never tried to cryopreserve fruit flies myself. So I can tell you the larvae, for instance, they have a particularly waxy cuticle around them. So that's the reason why they're not easy to cryopreserve. So what our method does is permeabilize that waxy cuticle to different agents, to cryoprotectants, but also, I mean, this method can be used for toxicology, for drug testing, can be used for a lot of other applications other than cryobiology. So that's the problem of the larvae. I'm not entirely sure about adult fruit flies, why they cannot be cryopreserved. I guess it will be, again, difficult to permeabilize them to the cryoprotectants. I would think that's the limitation. So there are a couple of papers. I think there's a Nature paper from like the 70s talking about cryopreservation fruit flies. But what everybody I asked told me is it doesn't actually work. It's not reproducible. So we just don't have a method. There's not been that many research on cryopreservation of fruit flies, but what everybody works on fruit flies they've asked told me is it would be brilliant if we have a method, because for example, when we create a genetically modified strain, let's say a long-lived strain for studies of aging, we just have to keep the animals alive all the time. So we have to keep them breeding. We can't cryopreserve them, put them in the freezer, say there's a pandemic. We do work in C. elegans and cells. We just freeze our stuff and that's it.
Right.
But I assume my colleagues who work on fruit flies, they have to keep going to the lab. They have to keep feeding those worms because they have to keep reproducing. You can't just freeze them. So it is an important bottleneck of research. And I think our approach, I think it's a first step. I mean, that's the thing. A lot of these are first steps. It's incremental into developing cryopreservation for Drosophila.
Perfect. Thank you so much. Unfortunately, we don't have more
pleasure.
time for questions right now, but we would then later on tonight or in the evening have actually a few more. And sorry for every attendee who didn't have their question answered, but there's another chance at the end of the talk. Joao, thanks a lot.
Thank you.
So now we would go for lunch break, not only because it's only also 1:30, but also we start the conference relatively late so that everybody from the East Coast and the West Coast, both on an attendee and from a speaker side, has it a bit easier to attend and doesn't need to get up literally in the middle of the night. So we would come back and restart with the next speaker at 2:20 p.m. So slightly less than an hour, 50 minutes from now. See you then, and until then, have a good lunch. Welcome back after the lunch break. I hope you all had a good lunch to Biostasis 2020. Just as a quick reminder for those who have recently joined us, because now probably some people from the East Coast in the US are able to also join. If you have any technical difficulties with the stream, then there's a backup stream under the domain biostasis2020.com/stream2. So please use that if there's any technical difficulties. And one more note, please, if you have any questions for the audience, from the audience to the speakers, please put it into the QA tab on the main stream, which is biostasis.com/stream. And now, without further ado, I would like to introduce our next speaker, Dr. Francesca Minerva, and the talk is called Ethics of Cryopreservation. Francesca is a research fellow or postdoctoral research fellow at the University of Ghent. Her research focuses on applied philosophy, including cryopreservation, and she's also an author of The Ethics of Cryopreservation. And again, her talk is called The Ethics of Cryopreservation. Francesca, please take it from here.
Thank you. Hello, everybody. I had to say I slightly changed the title of my talk. It's now it's cryonic selfish, because I thought that at this conference in particular didn't need to introduce the ethics of cryonics, because people expect to be quite familiar with cryonics and to be quite convinced that it is morally permissible. But maybe I want to play the devil's advocate on this occasion. And I think the question I'm about to discuss is one of the most relevant when we discuss the ethics of cryonics, the one that I am still myself not really convinced of. So this is my book, The Ethics of Cryonics. Is it immoral to be mortal? It's published by Palgrave 2018. And I discuss the ethics of cryonics at large, and also the issue I'm discussing today. Okay, I'm not sure how to do that. Let me check, because start my video. Okay, so let's go back to the slides. You can see my slides still. Can you see my slides? Okay, so so you're probably already familiar also with the cost of cryonics, depending on the cryonics facility one wants to use. But in general, the cost of cryonics is between $28,000 and $200,000, depending on whether it's only the head preservation and which kind of cryonics institute want to use, and whether there is a trust fund attached to the preservation. Some insurances have cryonics packages that cover the cost with a monthly fee between $30 and $100. I think they're available everywhere across the world. They're quite accessible. Obviously, the younger one is when they start, the cheaper it is. So overall, let's say that cryonics is expensive, but it's accessible to people in the Western world have low, medium income. It's not crazy expensive if one has the insurance. So how much does it cost to save the life of a person in a poor country? GiveWell, which is a meta charity, has estimated that in 2016 it was between $900 and $7,000. So if we consider whole body preservation at Alcor, with that amount of money, one could save between 28 and 220 lives in a poor country. So if we consider all the possible fees, a cryonics intervention, if one give up on their plan to have cryonics, they could save between 4 and 220 lives. And the same amount of money could be donated to save the life of people in poor country. So there is a question about whether it is really too selfish to choose cryonics when using the same amount of money you can save between 4 and 220 people. And I think this is a very important question. So if one is a utilitarian like me and thinks that there is no reason to be partial towards their own life, then it's quite hard to justify why one should choose cryonics instead of giving money to charity. But even if one is not a utilitarian, it is still quite difficult to justify how much value we want to attribute our own life to extending our life with cryonics for, I don't know, we discussed how long, instead of saving the lives of 4 to 22 people. And this is a serious conflict. So if cryonics has only a small chance of succeeding, then the reason for not choosing cryonics over donation to charity would be very strong, because you're using all this money to possibly extend your life, but it's very unlikely that you might succeed in doing that. And instead you could save the life of all these people, for sure, people who are alive now. But even if the chances of cryonics were really close to 100%, even if one is really optimistic about the chances of cryonics succeeding, cryonics would still be quite selfish, because you still choose extending your own life against saving between 4 and 220 lives. But then if one looks at how we spend money in healthcare, things become quite interesting. So now with COVID-19, we've discussed a lot the cost of intensive care units, which fortunately is free in Europe, in a sense, what we pay through taxes. But in the US, they're quite expensive. So the daily cost of intensive care unit is $5,000. The admission cost is $32,000. An elderly person spends about 1 week in intensive care unit. So the estimated cost of intensive care unit for survivors 1 year, so just like a percentage of those who can actually manage to survive the intensive care unit, is about $62,000. And only 33% of the patients who survived the first year actually survived for more than 1 year. So it's very expensive, and only 33% of the people who spend this amount of money in a certain age group actually survive for more than 1 year. So it's quite expensive if you think about much you're paying for extra year of life. Then I also started thinking about, I don't know if you remember this case of Charlie Gard. He had a very severe genetic disease. And the doctors in the UK said that there was no therapy for him. He should just let die. But doctors in the US claimed that they could test a therapy on him, and the parents raised 1.3 million pounds in donation, so that they could afford to take Charlie to the United States and try this experimental therapy. Eventually, Charlie died. But it was never debated whether it would be moral to try to save the life of one child, when with that amount of money, between around 1.3 million pounds, between 240 and 700 people could be saved if the money was donated to effective charity. So the life of Charlie Gard was estimated to be valuable, to be comparable to the life of 240, 700 people. That's a lot. That's a very expensive life. But then now you may ask, okay, well, you're discussing about people who are alive, and we should always try to do whatever we can to save a life. No, it's not completely true, because there is a budget. We can't spend any amount of money on saving a life. But now think about people who don't exist, and we might never exist, and we have no interest in existing. And that's babies. Like, a lot of people have babies, and they're quite expensive. And in the UK, for instance, raising a child for 21 years costs about 230,000 pounds. With that amount of money, between 42 and 230 lives of people in developing countries could be saved. I think this is a particularly interesting experiment, because we're thinking about people who don't have a right to life because they have no interest in living, because they're not even conceived, they don't exist. So there is, I mean, at least it's an interesting question whether people who are not born have a right to actually be brought into existence. But we're spending a lot of money without still knowing the answer to this question. One, there are other people dying. But we don't just spend monies on healthcare and in children. We also spend a lot of money on things that are not really essential, like beautiful houses or expensive cars, expensive clothes, expensive everything. What about cryonics when we start thinking about how we spend money and how we spend money in such a selfish way? Is it true that cryonics is more selfish than these other options? My hypothesis is that people think that cryonics is a waste of money because they assume that cryonics has no chance of succeeding. So any amount of money you spend on cryonics, be it £1 or $220,000, they say it's a waste of money because it's not going to work, so you should do something else with it. And they even say that it's best to use this money to buy an expensive car, because at least you enjoy the expensive car and you get something out of your money. But just like they would consider cryonics just like burning a pile of money. However, I think that cryonics has a chance of succeeding. We don't know yet what is this chance, but we hope and we think there is this chance. And the point is that if cryonics will succeed, its expected utility is going to be extremely high. So this means that cryonics could be the most cost-effective treatment in the history of human medicine. So unlike the case of intensive care units, where you can buy, we say like $62,000 a few years of life, cryonics could actually add hundreds or thousands, or we don't know how many years of life. And this makes this estimate of cryonics very complicated, because on one hand, we have low chances of succeeding, or at least a lot of uncertainty. And on the other hand, we have the possibility that this treatment will be actually the only treatment really investing in, because if it works, a lot of quality adjust life years qualities are going to be added to everyone's life. So it's difficult to compute, I think, for humans. Now let's assume that $200,000 could save 220 lives. So that's the most effective way of the most optimistic estimate about saving the life of people in poor countries. Suppose you can save the 220 lives, and that each of these lives would be extended by 50 years on average. So overall, with $200,000, we could save, we could add in total, collectively, like 1,100 qualities, because each person we save adds 50 more years to live thanks to our donation. But what if cryonics succeeded in adding more than 1,100 years of life to one individual then? I think that would be a worth investment then, because we can add the same amount of money, or maybe or time, or even more, to the life of one person. Now, some people say, well, that's still selfish, because that you shouldn't choose to extend your own life it's best to extend your own, so the life of many people for a smaller number of years instead of extending your own life for 2,000 years. And also people claim that once the average life expectancy is reached, cryonics or any other treatment is futile and selfish. There are two objections to this. Not all the people who opt for cryonics have reached the average life expectancy. Some people die very young. There was recently a case of this girl in the UK who was cryonics and she was just 15. And also, like, life expectancy is not fixed. Indeed, we are seeing that life expectancy is increasing. So, but some are saying, okay, well, then cryonics is permissible only for those people who die young. But this seems to suggest also that the ideal amount of time needed for a person to fulfill their potential corresponds, by a weird coincidence, to the average lifespan. I think it doesn't work that way. I think that what happens is that we humans know that we have a limited amount of time. So we plan our life and set our goals according to this very limited lifespan, and not to what we actually would like to do. And indeed, if we could live for much longer, we would have different goals and we would do different things. So it's not like by accident our life is like 8 years average, and then, you know, we happen to have done all the things we wanted to do over 80 years. That's not the case. Also, this argument fails to take into account individual circumstances. So if you're a woman living in a country where women can't study, can't work, can't really do what they want, even if you live for 100 years, you probably haven't accomplished all the things you wanted to accomplish because there were rules around you that didn't allow you to. So I think that if we were able to extend life, we would actually be able to enjoy this added years to our life, even though we had reached the average lifespan. So to sum up, I think, my time is up. Cryonics can be considered a selfish investment with possibly a very low potential to succeed, but people are willing to spend a lot of money on things that are not really necessary, including houses and possibly children. Selfish investments are very common, but people don't get criticized for buying a beautiful house or a beautiful car. People don't say, oh, why did you buy the house? You could have saved so many people. To say that people often fail to make the best decision, that doesn't mean that they absolve responsibility for the choices they make. And I think we do have a moral obligation to understand if we are spending our money in a way that is coherent with our values. And for me, this means understanding whether cryonics can be an ethical choice. And I think that's possible. I think that cryonics can be a non-selfish choice, especially if cryonics works very well, in the sense that cryonics succeeds at extending the lifespan by thousands of years, so that it will make it worth it to spend that amount of money to extend one life for 1,000, 2,000 years, when you consider that using the same amount of money on saving a life in people in poor countries would only increase their lives by 30, 40, 50 years. And overall, the absolute value would be higher for the life extension obtained through cryonics. In general, if we want to accuse people of being selfish and to invest their money on their own well-being, that we should focus on other things. There are a lot of things we do for ourselves that are overall as expensive as cryonics or more, and that we could use it in better ways. So given that there's a chance that cryonics will succeed in adding very many qualities, I don't think it's a waste of resources. So, but obviously we need more data, and this is sort of optimistic assessment because I think cryonics has a chance of succeeding. Thank you.
Thank you so much, Francesca, for the insight. Just one or two quick questions. I would ask you to answer relatively quickly because we're running a bit behind time already. So the question would be, would you choose to spend $100,000 for a cancer treatment for your child, or donate that money to people in extreme poverty? And maybe as a combination to that question, another one, wouldn't cryopreservation or cryonics just be an extension of emergency medicine, which is just very expensive in normal cases as well?
Yes, I agree that a lot of, I think that, so like, I don't know if I mean, most likely if I had to spend $100,000 on myself, I don't have children, so, but I guess that would extend also to children. I would do that and I think most people would do that because we think that it's worth saving the life of our own life. And I think that's exactly the same problem, that, you know, people are very quick in accusing cryonicists on wasting a lot of money on their own life, but they make the same decision on a daily basis for themselves. So I think the problem is that how people think that the chances of cryonics are very low, but indeed, as you say, it could be that cryonics is just extension of a life-saving treatment. And as I mentioned in my presentation, it could be the most effective life-saving treatment that we ever developed in the history of medicine.
Right. Thank you so much. Due to the time, unfortunately, other questions we would need to postpone till the end of the conference, because now I would like to welcome our next speaker, David Wood. Just as a quick introduction, David is an independent futurist, consultant and writer at Delta Wisdom, and the chair of London Futurists, where he has organized regular meetings in London mostly on futurist topics and techno-progressive topics. His talk will be about anticipating changes in public attitude towards death and cryonics.
Many thanks for the invitation to be part of such a fascinating conference. I will start with a number of confessions. First confession is I'm not a scientist, but on the other hand, I have studied quite seriously the philosophy of science, and I'm more than happy to argue with people whether the scientific language of cryonics is good science or bad science. And also in my background, I had 25 years of experience in high tech, in the mobile computing and smartphone industries, where I saw innovations move from being fringe and strange, then with the earlier smartphones, into mass adoption, although I also saw many examples of possible innovations not taking off at all. I'm a friend of the cryonics community. I have been tracking with interest what's going on in this community for a couple of decades, but I have to confess I am still a cryocrastinator. I have not got my arrangements with any cryonics provider sorted out. In fact, I would call myself a level 2 cryocrastinator. And as was mentioned in the introduction, I am the chair of London Futurists, but in this talk, I will not be saying what I think the future definitely holds. I'll be outlining some issues about the future, but also the past of cryonics. And in this case, I'll be quite happy to be proved wrong with some of my assertions. Having said that, let's move on to my talk, anticipating changes, possible changes in public attitude towards death and cryonics. So as a futurist, let me draw a timeline with 5 points highlighted, not to scale. Here we are today, 2020. I'll be looking back a bit to 2005. Why I start there, I'll explain shortly. And I'm looking forwards, possibly 100 years to when cryonics is a successful enterprise, a clearly successful enterprise. It might be sooner, of course, if there is something akin to a technological singularity in the middle of the century, or it might take a lot longer to achieve the goal. In fact, it might never come, of course. The goal that there is frequent, successful reanimation of people who have been cryopreserved in the present day and in adjacent decades. But I'm not looking so much at that possible future. As a futurist, I want to focus on how we can get there. So I'm going to look at what might be possible in around 15 years from now. I did say this chart was not to scale. In 15 years, perhaps, it might be possible that cryonics has moved from its present status today as being a very fringe activity with only slight interest, often comic interest from the mainstream. It will have changed into being a regular part of the mainstream culture with close interoperation, close integration between standard medical procedures and standard medical personnel and those taking part in cryopreservation. At the same time, there could be a major change in public attitude, which I'll be talking about. But that's 15 years in the future. Can we concentrate on what we should be doing in the shorter term? If foresight is just talking about the distant future, it has failed. The point of futurism and foresight is to get people to change what they do in the present term, in the present timescale, to increase the chances of attractive futures longer term. So what might happen in the next 5 years? And of course, we can talk about, that has already been mentioned in this conference, the possibility of more funds being invested in research and development, the possibility of more well-known public figures, celebrities of various sorts, influencing people to take cryonics seriously. But how could we get these outcomes to happen? So I'm going to be looking at a couple of things we might do in the next few years, which basically will come down to reducing some of the negatives of current cryonics and accentuating the positives. But before we get there, I want to look backwards. Good foresight is grounded in good hindsight. I'm going to claim, and this is where I might be wrong, that not much has changed in the world of cryonics in the last 15 years. I picked this date because I remember reading the book, The First Immortal by James Halperin 15 years ago. In fact, this is a screenshot from Amazon, which reminded me when I bought this book. It's a book that sets out lots of issues about cryonics in a fictionalized form. I remember discussing this with my son at the time, and I certainly was looking at cryonics before then and afterwards. But what has happened in these 15 years? In some ways, there has been tremendous changes. Smartphones, computers, networks have come along in leaps and bounds. Artificial intelligence moved from being hyped and disappointing to being transformational in many fields. Renewable energy has plunged in prices more than most people forecast. There's been rapid changes in genetic sequencing, reprogramming of genetics, synthetic biology, and yes, a surge of interest in many aspects of rejuvenation biotechnology. We haven't conquered aging yet, but there have been many very fascinating steps forwards. That's in the wider field, but in the world of cryonics, I think, and some people may prove me wrong, I think there's been a limited additional interest compared to what was 15 years ago. Limited additional funding, little breakthroughs in advocacy for cryonics. And yes, there are good arguments, but most of these good arguments existed 15 years ago, 30 years ago, maybe even 45 years ago. And there have been some changes. There's been some breakthroughs in the evidence for what we might call digital cryonics, in which brains are put into this state from which they'll never recover biologically, but they might have their data read out and transformed into running on the cloud. So that has changed in the last 15 years. And there have been some interesting experiments. For example, the memory retention in C. elegans worms. But this has not yet scaled to larger organisms or larger organs. So if it is true that there has been little progress in the last 15 years, what are the grounds for thinking there might be better progress of the sort I have referred to in the next 15 years? And of course, futurists often talk about exponential progress. It's often said that disruptive changes go through a long period of disappointment before they become devastating. And so the trajectory in that case is slow, 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 faster, faster, faster. But on the other hand, I have to say from my experience in business, most so-called disruptions fail. Most interesting ideas never really take off, and the trajectory instead is slow, 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 slower, stop. And many companies, as I've said, died in the so-called chasm or in the valley of death. Many industries that people expected to progress have not progressed as expected. For example, there has not been much progress in supersonic jet airliners since my childhood. What's happened with nuclear fusion? Well, it's still perhaps 25 years in the future. What's happened with blockchain? Slow, slow, slow. So how can we tell the difference between the trajectory that cryonics may be on? Can we identify seeds of tipping points? Can we go beyond fatalistic observation? Can we get involved as an activist in encouraging seeds of tipping points? So I'm going to refer, as I said, to two possible sorts. Those that take the friction and the slowdowns and negativity out of the current cryonics world, and those that accentuate positive evidence. In terms of reducing friction, let's look first at reducing the awkward need to have to travel to another country to die if you were signed up. It is good news that there is possibly a growth in the provision of local providers, such as EBF in Central Europe, and I think we're going to be hearing about others tomorrow and throughout this conference too. So that is an encouraging step forwards. So of course, throughout most of cryonics history, there's been lots of plans for local providers. Are things going to be better this time? It's still hard to say, but it certainly would be better if there were simpler standby procedures, and one other headache could be solved, which is the worry that if you die, you might become brain impaired in the process. It would be far better if in some ways there was support, legal support for cryoeuthanasia. Controversial and difficult topic, but one that we need to keep on the discussion. What about having more confidence in cryonics organizations? Very often when I mention cryonics, even to people who are positive about futurism generally, they will say, oh, wishful thinking, some of these guys are cowboys, some of these guys can't be trusted. And it's for a long time been discussed that maybe there could be some independent verification of cryonics organizations meeting some agreed standards. Again, though, this has not moved forwards as fast as some of us might have hoped. Then there's one cause of cryocrastination, which is the worry that you might sign up for the wrong organization. You might sign up for the best organization today, but in the future there might be some better option. And if that's not solved, there is a logic to cryocrastination, and the logic is a good design principle that you should defer as long as possible the design choice when it's unclear which is the best option. If, on the other hand, it was easier to switch cryonics providers, and I know some people will tell me it probably is already, but that would get rid of that negativity. What about the costs? This was one of the subjects, of course, in the last talk. Sometimes people say, well, of course, we're going to reduce the costs via scale effects. And I saw in the chat a few minutes ago a reference to a paper by Ralph Merkel, who said that once there are millions of people being cryopreserved, the cost could come down to as little as $3,000 per full body process. So yes, many industries have benefited from scale effects, from learning effects, from practice makes perfect. If there are lots of people going through the process, then there is scope for innovation and things can indeed come down in price, just as what happened in the world of smartphones, which moved from being incredibly expensive only affordable by rich yuppies and drug dealers and business people who had far more money than sense, to a situation when even the poorest in the world can have a pretty good smartphone. In fact, a much better smartphone than these early adopters. But, you know, that needs these scale effects to kick in, and there are currently very few people being cryonically preserved every year. It's probably just a score or two at most. So there's not much prospects for that cost coming down anytime soon by that mechanism. Another means in industry in general where technological costs come down is by better integration, avoiding expensive handoffs, duplication between different groups of people, complicated connections. Ideally, we would integrate the work of cryonics volunteers with the work of existing medical professionals. But as we heard, the mainstream medicine currently has a low regard for cryonics. Often it's a grudging relationship. And so that needs to be solved before we can expect the cost to come down by that route. Then the other mechanism in industry is you can bring down the cost of your own projects by piggybacking, by taking advantage of other initiatives. And there is some encouragement here, as we have heard in the previous talks in this conference, the possibility of reduced costs of cryopreservations of organs that could lead in turn to better mechanisms for cryopreserving the whole body, or at least the skull. So there's some prospects there, but it is still moving painfully slowly. Then there's another argument that says, well, the costs aren't that much. Let's just go on the offensive here. Let's defend the costs of cryonics. And I have some sympathy with that. I had the argument the other day that it's cheaper to cryonically preserve yourself than do what is currently quite a fashionable thing, which is to scatter your ashes into space. Turns out that was an exaggerated claim. I looked up online. There are companies that will put your ashes into space for as little as 2,000 UK pounds. And in fact, they even offer a cheaper version where a ceremonial portion of your ashes are put into at least a trajectory to take you into space and then down again. So I don't think that's a good comparison. And in any case, that mechanism has clear evidence that it works in its own terms, whereas with cryonics, the costs are much higher, and it is completely unclear to most people whether there is any evidence of it ever working. So when we compare cryonics with open heart surgery, which costs what, about $100,000, or with heart transplants, which can cost upwards of $1 million, again, the comparison doesn't really work, because there is good evidence that lives can be saved, that quality can be increased, quality adjusted life years, whereas with cryonics, there is still the worry that portions of your family wealth will be tied up potentially indefinitely that could be put to other clearly beneficial purposes. So we need more evidence to transform that equation. And here's my brief assessment of the evidence that's been pursued recently. There is a good structure of preserved brains, at least in some cases by CT scans, but not at the level that can give us confidence that the memories and consciousness could be brought back. And indeed, there is better evidence of preserved brains for what I've called digital cryonics rather than biological cryonics. But hopefully we'll hear better about that shortly. We've heard about reanimation of animals from reduced temperatures, from extreme hibernation, but not as low as the minus 130 degrees of liquid nitrogen. And we have to beware extrapolation. This is suggestive, but that's at most. And as for reanimation of animals larger than the C. elegans worm, there doesn't seem to be realistic prospects of that happening anytime soon. A lot of the research in this seems to date back to the 1970s or 80s. So what's a reasonable next step there? The best I think we can hope for is reanimation of organs from radically reduced temperatures, and the work that has been referred to already, which is understanding how to limit damage from the various antifreeze mechanisms. So that takes me on to my last two slides as a kind of summary. How can we change public opinion on this? And it's my observation that most people, when they think about cryonics, including many of us in the cryonics community, we are in any case in two minds about this whole enterprise. On the one hand, we say, yes, it would be great to be able to meet our loved ones again, and great to see the future. And it's just wrong that people are penalized for dying at the wrong time, whereas if they were able to live longer, they would have a much longer lifespan. But at the same time as we have these optimistic thoughts, there is the counteracting thoughts that my family will think I'm crazy, my family will think I am selfish, and indeed the main pressure from society is the expectation that we should pass on our savings to other people when we die rather than keep them locked up. And we might even think that we ourselves are being deluded. But this dichotomy, the balance can be changed if we have a better narrative about how a future society can be realized in which it is able and willing to reanimate us, where there is more positive evidence that can reduce the fear of us being deluded or our family members being deluded. Indeed, positive evidence that can inspire people to be more courageous, to become strong, positive advocates for cryopreservation, so that we will then collectively say, no, this viewpoint that society expects us to pass on our savings to others when we die, on the contrary, society should increasingly expect us to give our loved ones, our family members, a good chance to live again. So I refer to ancient philosophers who have said, and here's a picture of a Greek philosopher, though I might have had a picture of the Buddha here or many others instead. It is sometimes said by them that the main task in life is to prepare for death. I've never liked that sentiment. I've often retorted that the main task in life is to live with a flourishing abundance. But perhaps we can look ahead to something a bit in between, that by 5 to 15 years, more and more people will say the main task in life, or maybe not the main task, but a vital task in life, is to prepare for a successful cryopreservation. That we will say to each other, have you got your pension plan? Have you written a will? And where is your cryopreservation facilities? And if we don't do that, we will feel irresponsible. So these are my remarks, and I'm more than interested in the questions that may turn up. Thank you so much.
David, thanks a lot for this great talk. Due to the time limitations, we will only do one question now and then postpone the other questions to the end of the conference. And the question is, in your opinion, which of the following methods is more likely to allow you to see the 23rd century? Cryonics, mind uploading, or anti-aging? We can't hear you right now, at least I can't. So, in your opinion, which of the
So I'm about 25% optimistic that anti-aging will arrive in good time for my own life. I say it's about a 50% chance it will arrive by the middle of the century, and there's about a 50% chance I'll still be alive by then. I multiply these probabilities together and get a 25% chance. In terms of the possibilities of cryonics working, I haven't got a good figure for that, but it's probably at most 25% too. So there's another chance. As for mind uploading, I remain skeptical about that, and I will remain skeptical until AIs probably teach me the right philosophy of mind that will give me the confidence that if my mind is uploaded, it will be me rather than an alien copy of me.
Fair enough. Thanks a lot for the insight. Our next speaker will be David Gifford. David, a quick note before. Could you turn your camera on if that works?
Okay.
Otherwise we can also
in
the meantime, I'll briefly introduce you. David is has a background in perfusion, is a perfusionist, and is currently a transplant perfusionist specialist at the Cambridge University Hospital. and his talk will be about perfusion, or has the title, Perfusion and Torpor, Techniques as Precursors for Human Cryopreservation. David, take it from here.
Thank you very much. I'm delighted to have this opportunity to share with you a couple of ideas that come from transplantation that may be useful in the in improving the prospects for successful human cryopreservation. But I wish to point out that this is a, a personal view. Okay, so as yes, I'm a perfusionist, and my background is for many years has been a perfusionist in heart surgery and transplantation of all kinds, as well as in the intensive care unit looking after patients on long-term bypass systems. This is a recent photograph of a colleague of mine looking after a patient, COVID patient on veno bypass. Okay, so I'm interested in looking at trying to change the odds and therefore improving the prospect. And by doing that, I was thinking of something called the Warren equation, which was from an essay published in 1989 by Harris, looking at basically the odds, the probabilities of chronics being successful. And they came out with a lot of different elements and gave a weighting for them all, and therefore an overall prospects for success, which as you go along becomes very, very limited. I looked at this and thought, well, probability C, the favourable conditions of suspension can possibly be elevated, and so therefore improve our odds. So that's really where I'm coming from. And so in order to do that, I want to talk first about circulatory arrest. In other words, how we die, and talking about energy loss and catecholamiine storm. Also to look at how perhaps mild hypothermia as the first step could help to make a bridge between limited resuscitation to more extensive resuscitation. How that might apply in a perfusion settings for not only for organ preservation, which I'm using as my template, but also for resuscitation of the chronics patient, and that would be step 2. And how that may impact on the more rapid recovery of patients from cryopreservation. Okay, so just looking at this rather colourful graph, which shows between age 1 to age 95 in 5-year strips, the likelihood of you dying from any major disease. But at the end of the day, normally what really happens is that you're not dead until you're cardiac dead, until your heart has stopped. So that's what I'm really going to concentrate on. And if we think of it from a metabolic point of view, from a metabolic debt point of view, we would see as a patient reaches the end of their life and they move from unconsciousness into Cheyne-Stokes, into the agonal phase, into a cardiac arrest situation. And the patient is declared dead at that point. However, I would suggest that actually the decline is actually much more severe during the time when the heart is still beating or trying to beat, and providing extremely bad conditions for the cells, primarily because of increased levels of cytokines, which are occurring at this time, leading to something called a cytokine storm, which is devastating. If we look at just the heart itself, then the simple consequences of having increasing demands on its viability and a reducing oxygen supply means that bad things happen and continue to happen more rapidly, leading eventually to cell death and anaerobic metabolism and cardiac arrest. So ischemia, the problem is that once we move from aerobic to anaerobic metabolism, what do we see? We see a huge increase in energy consumption. And so therefore energy is rapidly depleted, and metabolites then accrue. And so we're leading to this concept of, in simple terms, of a battery that's discharging and you're unable to recharge it. I'd like now to sort of step across to look at a little bit of hibernation, not the way that we see things in science fiction movies, but in actual medical practice. And when we look at medical practice, particularly in cardiac surgery, what we see is that if you're at normal thermia, 37 degrees approximately, and you've had a very good circulation up until the moment that your heart is stopped, that you've got about 4 or 8 minutes before you start to experience any level of brain damage or dysfunction. Now, as you cool, you get additional time, perhaps as much as 60, 65 minutes, but you'll notice something about these curves, that as the temperature decays, the increase in time is not quite what we would hope for. So it's if something else is going on. We're not actually hibernating animals. We can't change our metabolism to affect great benefits that hibernating animals do. So we're going to have to make do, for the moment at least, with mild medical hypothermia. And this will slow metabolism. This will be safe down to 32, 30 degrees, provides good cell protection, and is simple to effect with techniques such as cardiopulmonary resuscitation, using anesthetics and topical cooling. And there's the potential of using chemical induction, which has been used in animals such as adenosine and hydrogen sulfide. And the idea with this is that, yes, the batteries are going to be discharging, but at least they're discharging more slowly and will buy you time to get to a more extensive facility. So, and yes, this is a thing. Yes, we do this in clinical practice. It's done medically. So this is good. The bad side is that you hit a point where bad things happen, and we find that this doesn't work much below 30 degrees, and 28 being kind of the barrier under which this won't work in a rescue setting. And the reason for this is very simple. Most of the processes within the body, major processes in the body, are temperature dependent, and some will just go into dysfunction faster than others when you lower the temperature. And so those have a detrimental effect on the utility of more profound hypothermia. The one thing that we're trying to do, which is reduce the metabolism, yes, that's fine, that works, tick the box. But we do have other elements such as an increased risk of infection, reduction in platelet count, resulting in additional bleeding. Cardiovascular is an issue because once you hit 28 degrees, your heart will arrest. And, actually having seen that in a live setting, I know this to be true. So how do we can apply this in a chronic setting? Hopefully not by the roadside, but at least we can apply this simple techniques such as cardiopulmonary resuscitation, ventilating the lungs with oxygen, applying a drug protocol such as an anesthetic, such as propofol, which will lower metabolism all by itself, and heparinize the patient so that you prevent clotting. Using cooling blankets and wraps to systemically cool the patient, topically cool the patient, and possible endo-cooling systems which will make that cooling much more rapid. Cooling down to 30 is possible, and this is to enable by time for you to indulge in something a bit more radical. And that I'm proposing is the use of extracorporeal perfusion for the chronic patient, for donor organs, donor patients, and it is successful in restoring metabolism and getting control of all the parameters that you need to be successful there, as well as providing deep cooling should it be needed. But it is relatively expensive and requires training, but it does put the patient back on the mains and so enables the batteries to be recharged. So the model that I want to examine a bit more closely is the difference between donation following brain death and donation following circulatory death. Now, the two are distinct in that the brain dead group can be considered to be pre-agonal. In other words, their circulation is very good until their organs are removed. So the organs tend to be well perfused, in a good state, and they're not toxic in any way, unlike the circulatory death patients, the post-agonal patients, which their organs are in a poor state and are often show poor utility. And this is demonstrated in many studies, so I won't go into that, but just to say that the evidence is there. So what have we chosen to do over the past decade or so is to use an extracorporeal circuit as an intervention into trying to prevent that deleterious effects occurring. So what we're trying to do is to treat that hypoxic agonal stage, that warm stage, and restore the organs back to a good quality for removal and retransplantation by increasing their energy stores and removing their metabolites, and so therefore making them more robust to survive by something called a reperfusion injury. So and it's just really a question of recharging those batteries. So a couple of slides here suggesting that yes, DCD organs can be improved in this way, and we're doing it as a routine now. So we can use the big heart-lung machine, which we use for cardiac surgery, and it's got a really complicated circuit that's attending to it, but we don't need that. A simple circuit is adequate, and it's simply just an in and an out, a pump, an oxygenator, a heat exchanger. Here is one that's being used for a donor patient, someone who's died from circulatory arrest, who's being resuscitated to a point whereby their organs can be removed. So that's the premise. The other thing that's required, and we also use a device with the donor organs, donor patients, is something called a cytokine absorber. And this removes these activated cytokines and enables, again, an improvement in the quality of the organs. So in the chronic setting, the step 2 is to establish bypass to rewarm patients surprisingly up to normal temperatures. And the reason we want to do this is because we want to normalize biochemistry, and that takes about 2 to 4 hours. So we want to recharge the batteries. We also want to remove the activated cytokines. And the reason for this is because they are immensely damaging to the circulation and impede the quality of the perfusion. So looking at the cytokine storm, the otherwise known as the immune-mediated hyperinflammatory state, a whole shed load of interleukins are activated, and these are very destructive. And if we show you this little animation, I hope that you can see that in the red boxes at least, that the circulation has been impeded. Now, using some sort of high-capacity cytokine absorber, those metabolites can be removed, the cytokines removed, and the circulation is restored. This is a device that's being used extensively in this year that we're in, as far as the COVID-19 patients are concerned. So this is real stuff. So it's very simple. The idea is that we clip the top off the toxicity of this cytokine, enabling recovery to take place. Clearly, this is really quite a complex area and something that I can't go into any more detail here, so we'll move on. Just to say that, yes, it does actually have a positive impact on patient recovery, as shown by these lactic acid results. Okay, so what have we done so far? We've rewarmed the patient on ECMO, we've recharged the batteries, we've normalized biochemistry, we've fixed the cytokine problem, and now we commence deep cooling, lowering the hemoglobin, because we want to make the blood thinner by removing excess cellular components to help in the perfusion as we cool. And then at an appropriate point, move to a conventional chronic vitrification process, chronic cool down protocol. And then at the deep cooling phase, we would, suggestion is that perhaps as a possibility that we could use microparticles perfused by the machine to infiltrate all of the capillary beds for later magnetic or ultrasonic preservation and rewarming. So at the end of the chronic process, when the patient is being reanimated, then this may be a way of getting appropriate core rewarming rather than having to rely on very exotic nanotechnology. So that's the looking at this a little bit more closely, we know that, and it's been itemized by previous speakers, that chronically cooled cells don't all survive. 70% survive rewarming. I'm not talking about the cooling, I'm talking about the rewarming process. And here's an example of different cell types. Vitrification can, of course, help and is being used. But the magnetic nanoparticles have been used to provide inductive heating clinically in the use of cancer, as well as for chronically stored tissues, but only very small amounts. That being said, if we've got a handle and access to the circulation through good perfusion, then the possibility of distributing these particles within the organs of cells and possibly an entire body may be a practical proposition for practical rewarming, rather than just trying to dunk the cells in a pot somewhere. So that really is the, apart from just to say that with inductive heating, there are issues with these particles. One is particle size. What particle size do we choose? The suggestion is that we actually don't use nanoparticles, but we use microparticles of around about 2 to 4 microns. The reason for that is because as they are platelet size, they're going to stay in the circulation. Nanoparticles will tend to get into the extracellular spaces and perhaps into the cells and cells and be an issue to remove in subsequent rewarming, whereas microparticles staying in the capillary beds in the circulation can presumably be flushed out once the recovering patient is reaching post-freezing temperatures. And so just to summarize, I'd just like to say that I hope I've implied the problem of energy loss and the problem of the cytokine storm, how mild hypothermia, can buy us a little bit of time for an extra therapies, how the DCD model in transplantation, organ transplantation, points the way for improving techniques for a perfusion for the chronic patients to try and restore them to a pre-agonal state as though they'd never actually lost the circulation, which would, and then finally, if the prospect of inductive warming through either ultrasonics or magnetic systems is practicable for chronics, those then can be infused into the patient. But clearly all of these elements are going to be complex and are going to increase the complexity of the procedure. So, but the idea is simply very simple, to try and improve the odds. So by improving the odds of suspension, also improving the odds of it will work overall. And with that, I'll leave the last word to...
What's the only particle of hope I have, and it's just a particle, is to be frozen.
So let's hope that we can increase that particle by 10%, or not 10%, by 10-fold. Thank you very much.
David, great talk. There are 2 questions for which we have time currently. One would be, in your opinion, oh, sorry, that's the wrong one. The right one is, how do you judge the utility of current perfusion technologies for ex vivo, so isolated organs, for studying systemic perfusion?
It's a great question. There's lots of systems now where we use isolated organs, where we put isolated organs on perfusion systems. So I've been intimately involved in, for a long time, a system called Organox, which is for taking isolated liver and using that and so recovering isolated organs for subsequent transplantation. There are similar systems for hearts. There are similar systems for lungs, and there are more simple systems for kidneys. So there's been lots of interest and lots of development over the past few years to try and move from putting donor organs just on ice or in a cold bag to put them on an active perfusion system, which buys additional time. So the idea is that then you can get something instead of having to let's say retransplant a heart within 4 hours or so, that you might have 24 hours in which to get the job done. I don't know if that answered the question, but that's my answer.
Fair enough. I can't unfortunately here ask back to the audience. Maybe really quickly, last question. What is used to capture the cytokines? Okay, a cytokine, there's in the past we were using something called a leukocyte depletion filter, which is just a simple screen filter, high capacity screen filter, which has got, but it has limited capacity. So so that's how it's performed. We're now moving to, for this kind of problem, now moving to these high capacity absorbers, which are in themselves microparticles, and they've been coated so that the cytokines will stick to it and so be lost to the main circulation. These work to about 55,000 Daltons, so they're medium-sized molecules that they can absorb. So that's the kind of technology that's being used. That being said, it's a technology that really hasn't received FDA approval yet, but because of the COVID problem, these things have been rushed into clinical use. That means that we've suddenly got a lot of information, or developing a lot of information, on the utility of such devices. So it really is landing. Perfect. Thank you so much. Talk to you later.
Thank you. Bye.
Our next speaker will be Jacob Hoekstra. Just as a quick introduction, Jacob is a chair at the Biostasis Science Foundation, specializing in advancing scientific research and medical-grade human tissue stabilization. And of course, he's one of the pillars of the activities in Europe. Jacob, take it from here.
Emil, thank you very much. And I'm very glad to be part of this conference, which is very professionally organized, in my opinion. And it's great to be part of this. You invited me over to come to Zurich with our new vehicle, but unfortunately this is not possible this year due to COVID-19. But I'm happy to come over next year and use this opportunity online to present our capabilities developed in the Netherlands and for the use in Europe. This talk will be about the capabilities we have developed. But I would like to start with the concepts that are the initial theory, the fundamental basics, which have made us develop these technologies. Let's see. Okay, I will first present the concepts. There are 8 concepts, in my opinion, and let's start with the definition of the word biostasis. What is biostasis? I think we should make a definition for biostasis to be stabilization and preservation of biologically viable human material. This is to make a distinction between the preservation of already deceased material and human material which is still biologically viable. In this way, the field can develop as a scientifically developed field, which also includes, for example, military use of biostasis technologies, space travel use of biostasis technologies, transplantable organs, the preservation of these, and also common scientific research donors, for example, brain donors. Nowadays, if you donate your brain to a university, the brain usually arrives after a few hours after the person has deceased. Yet, if we could apply biostasis technologies, then the brain could be preserved in a way much better, so that the brain can be delivered, at a much higher quality to a university for research purposes. The next concept, is what do we do in the Netherlands? We are Biostasis Sciences Foundation. We were founded and registered in Amsterdam in 2017, which is 3 years ago. There are 2 founders and 2 directors, myself included. We are a non-profit organization, so our aim is really to be independent developers and provide technology to any organization who needs it. And our ultimate goal is science-driven advance of at exponential rate. So, well, to really make biostasis grow and grow and make it go at an exponential, high exponential rate. The next concept is scientific advance, and the very important option to be able to drive with biostasis material every day. If you want to develop something into something which is scientifically validated, then you need to be able to do this on a daily basis and not every month or every 2 months. You need to have equipment and vehicles and personnel to be able to drive on a daily basis or even hundreds of times per day worldwide. Then all this scientific data can be gathered together at specific research centers and analyzed so that there's some actual improvement. So what we are aiming to do in the Netherlands is to create daily logistic services between, for example, houses where a person is dying, or a hospice where such is happening, and also hospitals, and then provide the logistics from these places to, for example, universities and research centers all across the world. If you are a brain bank donor, you donate your brain to scientific research, to a brain bank, then, for example, you would die at your house or in a hospice, and we could provide the logistics to move you from your house or hospice to the university where this research with this with your brain is performed. Of course, every organization has different protocols and different ways of treating the material. So essentially, what we would be doing is we would be performing the protocols as provided to us. So we would not come up with protocols. We would receive protocols from a certain entity like a brain bank, for example, in Amsterdam, or an organization in the US. They would provide the protocol for us to be able to execute it on this specific person. Well, our goal is really to have 24/7 operational readiness and infrastructure with vehicles and trained staff in every region. So we currently start with 1 vehicle in the region of Amsterdam, but our goal is really to expand this into Europe and into the world. Technological advance and availability is very important. If you want technology to work and to become better, then it has to be widely available. So not just in 1 location in the world or in 2 parts of the US. It has to be everywhere. This is especially important for organ donors. If an organ donor would die at home, then it's usually very difficult to prepare this organ donor in a way where the organs can still be of use in the hospital. But if there would be stabilization infrastructure to pick up such a person from their home, where the person is dying or already has died, and then stabilize this person en route to a hospital where the receiving person who is to obtain the organ is located, then you can really create a more seamless way of getting more donor organs as well, because there will be more quality organs. And by doing this, you gather a lot of research data, which is applicable in many other locations in the world. And so if you have a lot of locations where you're active as an organization or as multiple organizations, all this research information can be used to make more and more improvements. Well, we develop logistics technologies, and that is our focus. We focus mainly on technology, and we are open to any partnerships with clients, suppliers, or others. exponential growth in human resources is also very important, because if you only have the technology but no people to use it, it's not very useful. So our goal is also to train medical staff, train academics, funeral providers, and these people are generally open to being trained in more advanced procedures to stabilize a human being. Well, we've already been in contact with a few of these, and these are very interested to learn these principles to be able to expand their field and their knowledge. So it would be useful to have worldwide training facilities in the US, in Europe, in Asia, or Australia, everywhere. What is also very important, in my opinion, is the ability to have 24/7 live people monitoring. So we have partnered with a supplier of technology, which can be installed on a smartphone or which you can wear as a device. There's different types of devices available. And then what will happen is you would show up on a map with your location, but nothing would happen until something goes wrong. For example, if you haven't moved for 30 minutes, or if you press an SOS button on your wrist, then something would start happening. And this system is backed up with a 24/7 staffed alarm company, which has call centers in all parts of the world. So, for example, if you have an emergency in Germany, then you will have a German person talking to you, or in the Netherlands, it's a Dutch person, and they will usually pick up the call within a minute. But while this is ongoing, we will already be prepared, for example, by receiving SMS messages, which happens in the background. And plus, your family can also be alerted if you desire so, which also happens after a certain number of user-defined minutes, a number of minutes. So you can configure it yourself. If you're interested in this service, then please go to the URL in the bottom of the screen and let us know that you're interested in this. At first, we are able to provide it for free to a certain number of people, even though we need to pay for this service. The next thing is very important, I think, is pre-mortem care. Patient telemetry and death with dignity. Patient telemetry can help you in assessing when a patient is about to deanimate or die. So you connect them even in a hospice to very complex telemetry systems, so you can monitor this person and really make a good prediction of when the person will die. And we have acquired such systems as well, so that even people from across the world can monitor a dying person. Death with dignity is very useful in trying to optimize the process. You can have a standby team waiting at a designated moment chosen by the person who is dying, to make the intervention, the standby intervention at exactly the right time, so that you don't have to invest in a random situation, but you can prepare the whole team at a specific moment. Of course, in the Netherlands and in Switzerland, these are very important developments where these options exist. And the last point on this list, post-mortem support for next of kin. Well, if you take an organ donor from their house after they have just died, or a scientific body donor, just a common scientific body donor, then well, I've heard stories that next of kin just stays behind and they're just alone, and the person who died has been taken away. Well, just as an example, in the Netherlands, we have partnered with a funeral services company, which is very willing to go on the standby team with a person skilled in empathic, making talks with the next of kin, while the technical team can prepare the person, and this funeral service provider will stay with the next of kin, even when the deceased person has already been taken away. This is, I think, a very important development for the whole world. Well, what we do in the Netherlands, we design all kinds of technologies. In 2017, we started with this international shipper of biological material. You can ship a whole person in this box. We have used it now twice, for a shipment to Russia and one to the US. and it returned pretty much undamaged. It's a very, very high quality box. Well, we produce these for any entities or organizations or people who are interested in getting these boxes. And I have designed a new model, which I will build into 2021. Which will have even better cooling, so it will be able to go colder than dry ice temperature and still be able to retain cold for a certain number of transport days. The box in the photos is from 2017, and it can stay on dry ice temperature for about 5 days, even though international transport usually takes about 12 hours or 24 hours. But it's better to keep cold as long as possible. Well, our offices in the Netherlands, we have purchased a total of 400 square meters of building space, which is 55 minutes from Amsterdam Airport, 20 minutes from Lelystad Airport, which will be the #2 airport in the Netherlands in the next years for all European flights. And it's only 1 minute from the main highway, from one of the national highways in the Netherlands. So, and we've chosen a spot where there's only a traffic jam maybe once a month. So you can easily access the road and get your vehicles on the road. The main purpose for our buildings is research and development and training personnel, staff, and volunteers. Well, here are some photos from our buildings. Here in the left, you can see, well, this is where pretty much where the construction was ongoing. The construction was somewhat further. We really insulated the walls completely for sound insulation and things like that. And this is how the presentation room for training looks right now, but it's not completely finished. It will be finished around January 1st, and it can host around 10 to 15 people on a training every time. Well, we have constructed bathrooms with showers and things like that as well in our buildings. For example, if you have a standby team waiting for a certain situation, then of course it's better to have all facilities in the building. It's not completely finished yet. I was actually working on the glass this week. But we also have a bedroom and things like that. We have really developed our facilities here. This is, for example, the garage. Well, we started with the heating in the floor. We really we have air conditioning as well and climate control and ventilation. But we really started from almost scratch in these buildings. And now it pretty much looks like this and this downstairs. We also have a kitchen downstairs in the garage, and well, the door can be opened even remotely through the internet. So in case someone is not available to open it, and everybody can get electronic access, whoever desires it. Well, the most important thing we have developed is our first vehicle, which is called the Biostasis SR01001, and SR stands for Stabilization Responder. It's factory produced, which is well, that doesn't happen very often in this field, but there's only one ambulance factory in the Netherlands. I contacted them if they were interested in producing a new type of vehicle for post-mortem care. So, and this was the first vehicle of its kind in the Netherlands, and they were very interested in producing it, and also very interested in biostasis applications. So I visited the factory 15 times and met with a lot of staff there, and a total of 8 factory ambulance engineers have worked on this project, plus myself. So we have tried to develop a very sophisticated vehicle, and I will, well, it utilizes a modular rail system, for example, so that it can be changed for every type of situation within approximately 3 minutes. Okay, I will now move to the vehicle because I see we don't have a lot of time left. Well, on the right-hand side here is, for example, remote telemetry. There is a dummy in the vehicle. I will open the vehicle in a few seconds, and you will see there's a dummy, and it's now running in demo mode. But just as an example, we can monitor the dummy even from the other end of the world in almost in real time. There's a few seconds delay, but and it's the system is certified and engineers come here every year to check the system and test it. So it can really be used for checking if a person has really died or things like that. Well, this is the inside of the vehicle. I will get in on the side. and I will make it fast. Well, as you can see, there's a dummy here. We utilize different types of or new types of medical hardware, which has not been used in the field of biostasis before. It's all the latest technology. The patient monitor is here. It's wirelessly able to measure the patient status, so you can easily walk around with 5 to 6 people around the person. The chairs can be put into any direction and folded up and things like that. Well, we have a hot and cold water sink, 40 power outlets for power use, which are all behind waterproof caps. Well, we have a freezer going to -22, a fridge going to the temperature of 0 degrees and between 0 and 10 degrees. And well, the side of the vehicle is all equipped with rails where you can mount any types of equipment, perfusion type of equipment, or even embalming equipment if you would like. And so the vehicle can really be tailored to specific needs of and requirements of an organization who demands for standby capabilities. And you can also get surgical trays and iPad holders for these type of mounts. On the roof are 2 4K cameras with 25 times optical zoom, so that it's possible for a university or a hospital or an organization on the other end of the world to zoom into the operation. And you can see about 98% of the vehicle from these camera positions, and really zoom in onto a millimeter level to be able to steer a situation. And you're also able to speak to the team inside the vehicle. So because the vehicle is equipped with 14 antennas, 4 Wi-Fi, 4 4G, which will be converted to 5G in the future, and a number of other vehicle antennas, 2 GPS tracking systems, and things like that. So this is really a very sophisticated vehicle. And the engineers were very proud to be able to build it. What we can also do is take the suitcases out from the wall within a few seconds and be able to bring them into a hospital. Obviously, as you can see, we still need to develop and finish the cooling system, which will be mounted on top of this stretcher system. So this is, by the way, an hydraulic stretcher system, which is capable of electronically lifting 350 kilograms in total, and we have tested that. So it can, it has, the wheels can be retracted and raised. So you don't need a lift gate to move the person into the vehicle. So this was my presentation for Biostasis 2020. I hope to see you next year, and I hope to bring the vehicle then to Zurich. Thank you very much.
Jacob, thanks. Great presentation. There's one question, and also to get a bit back to time. In your opinion, what's the role of 24/7 health monitoring system in biostasis?
People can feel comfort in wearing it. Especially is true, of course, for people who are 70 years and older, because they can fall or they can have any type of accident, or they can, the alert system is not just to alert the biostasis provider, because you will alert the call center and they will determine what needs to be done. But in the background, the biostasis provider is alerted. So that, for example, we can already make the vehicle ready, which takes about 6 minutes from the time of getting a call or a signal to getting the vehicle ready to drive out, and then it takes a minute to enter the highway, of course. So we can be en route within 10 minutes, but this all happens in the background while the call center is in contact with the person who is in an emergency, which can be any medical emergency or even a robbery, and then of course we can return with the vehicle without doing any work.
Perfect. Thanks for the insight. And thanks for your presentation. Now, our next speaker is Christopher Gillette. Hi, Chris. Chris has been working with EBF as a research fellow to better understand the sentiment towards biostasis in the US and also in Germany. And the talk will be Sentiment Towards Biostasis. Chris, take it from here.
Hi, everybody. I'm Chris Gillette. I'm going to be presenting some work that I did with Emil at EBF. We've conducted a large comprehensive survey of the general public's awareness of, interest in, and attitudes towards biostasis. This was an online survey completed by a little under 1,500 people recruited via a local community social networking site. And our sample represents US internet users. So it's a little bit younger, a little bit more tech savvy than the general American public. But always with surveys, that caveat applies that it's impossible to eliminate bias. Our goal always is to reduce it and understand the bias we're creating, and to be very thoughtful about all the decisions we make to reduce bias. And certainly I think we've done a very good job of that here. I'm confident that the results we have reflect reality. So let's get into it. Awareness of cryopreservation. This is an interesting topic because cryopreservation has been out there in the air for a long time. It's been a big part of popular culture. It challenges really basic ideas about the nature of death and the nature of medicine. But how many people have actually heard of this? What do they think about it? Well, now we have some good answers. 77% of people have heard of cryopreservation. Of this group, of our sample, a little over a quarter have actually actively researched the topic. This is a pretty strong indication of interest because people are taking it upon themselves to take that active step to look into it. And this is certainly stronger than just checking a box on a survey. But despite all this awareness, most people have not thought about this topic. A little under half of people say they've never thought about it, never seriously considered it. It seems to me to not fit in the same slot in the brain as do, say, burial, cremation, or CPR for that matter. It's not thought of as a part of critical care medicine. It's not thought of as an extension, or rather a viable alternative to the other end-of-life options that are out there. We asked people where they've heard of this topic before. The number one was internet articles, then fiction and nonfiction, TV and film, and word of mouth also stood out. Internet articles and nonfiction television. This is reporting by journalists. This tends to focus on the activity of providers, either as a general introduction to the topic, as a sort of curiosity with a light scientific background, or to report on something newsworthy going on with one of their providers, which tends to be lawsuits and that kind of thing. The fiction, television, and film is a science fiction that we're all familiar with. And then word of mouth, I assume, is a sort of distillation of both of these, the public reporting and the science fiction, with also the urban legends, Walt Disney, that kind of thing mixed in. And so this constitutes basically the information space of cryopreservation that's out there in the public eye at the moment. We asked people, we gave them sort of quiz. We asked them the number of providers, the number of cases, and we let them give an answer as their response. And people's performance here was quite poor, as you see. To be fair, it was a pretty challenging quiz, but there was something interesting here, which is that people tended to overestimate the number of providers and underestimate the number of cases. I think that's interesting because you would expect if someone overestimates the number of providers, they would also overestimate the number of cases. But it seems what's happening here is that there's a large chunk of people whose perception of the landscape of cryopreservation as it's practiced commercially today is one where you have a lot of small, presumably new, inexperienced providers who are offering cryopreservation to a small number of people each. And so the institutional longevity and the consolidation that we see in cryopreservation today is not something that's appreciated by the public, it seems. So there's a lot of awareness of this topic. How much interest is there? We tried to measure this a couple of ways. We just asked people, are you interested? A quarter said that they are. And half this group had not considered the topic before. The lightest way that you could express interest in our survey was by agreeing with the statement, I think cryopreservation is an exciting idea and I plan to research further. A little less than half of people agreed with that. Next, the next strongest one was I would actually like to begin a conversation to talk to your provider about getting coverage. Fewer people agreed with that, but still a pretty impressive fraction of people. And then the strongest indication of interest came when we asked people, well, what do you actually intend to do with yourself when you die? Burial, cremation, cryopreservation, or I haven't decided. And 6% of people said cryopreservation. This is the least common choice. I think burial was the next most common at around 8%. But this is quite impressive. It's certainly more than I would have expected. It's hard to know how seriously to take the 6%. Very few of them had done anything to actually formalize this wish, even something as simple as telling friends and family about this desire. So I think it's sort of now that you mention it, I would like to do that, but it's desire nonetheless, and that's quite interesting. We also asked people what the most important considerations were when choosing between different end-of-life options, and cost was number one, which is consistent with surveys done by the funeral industry, that kind of thing. With people's attitudes towards biostasis, there was an interesting effect. People's attitudes were much more positive than I would have expected, and than I think that many of you would have expected. One thing we did was we asked people, we gave them a bunch of different attitudes that one might hold towards cryopreservation. And we asked, do you think that most people hold this view? So we asked, do you think that most people think that cryopreservation should be illegal? 14% said yes, I think most people think that. We asked, do you think most people think that this is a scam or a ripoff? 32% of people said yes. But when we actually asked people, should this be illegal? Only 4% said yes. That's much less, I think, than many of us would have guessed. And there's another positive note here, which is that of this group, the 4% that's agreed with that statement, they tended to have not researched this topic before. They're not particularly motivated. Presumably it's not something they really think about. I think I remember that out of our whole data set, less than 1% had both researched the topic and agreed that it should be illegal. So, so that's a positive thing. We asked people to choose from a list of reasons why one might not sign up, might not have signed up yet. Number one was, I haven't thought about it. Number two, or maybe number three, was cost. And then way at the end was, I don't trust the provider. So it seems that people generally agree that providers are acting in good faith. But then when we ask people what problems they perceive to exist with cryopreservation today, the providers did come up. There was this concern that the provider may go bankrupt. That was the second most chosen option. Number one was that the practice damages the brain too much. People were able to choose more than one option here. I think this is pretty spot on. I think most of us would agree that the scientific challenge of the crudeness of current techniques, and then the institutional challenges like the risk of bankruptcy are kind of the main areas to focus on. And then included in that, we asked people if the stigma around this topic matters to them, and they said it didn't matter to them. That was one of the least commonly selected. So looking back at this, if we want to put people into 4 buckets, I think it basically goes like this. A quarter of people are just disinclined to the topic. They have personal, philosophical beliefs about death and attitudes towards death that are just incompatible with this practice, and they're not interested in signing up. And that's fine. It's a personal belief or feeling that they have. The good news here is that this group is not hostile. They have a live and let live attitude for the most part. Now, half of people are open to the topic, not closed off. They haven't really thought about it, but they would agree with the statement that it's interesting. Maybe they'll look into it. Now, I would be very interested to see which groups these people tend to go to once you prod them to think more seriously about this topic. That's an area for more research, perhaps. And then 20% of people seem interested. They've researched it. They have attitudes towards death that are compatible with this topic. One of our major findings here was that a person's attitude towards death, whether they want to live longer or not, whether they think death is natural or should not be avoided or not, was very strong in predicting whether someone was interested personally. And then 5% of people seem strongly interested. They say they intend to be cryopreserved. Not only have they researched the topic, but they've found a provider's website, prices, and that kind of thing. Now, again, I don't know how strongly to take this 5%, but if we really could get 5% of people to think very seriously about this topic, maybe take concrete action, that'd be quite something. So to close here, there's a lot more that could be said. Please look to this PDF that we've put together to communicate our findings in more detail. And if you have questions, there's a Q&A feature. You could also send me an email or schedule a time to talk on the phone. You're all very warmly invited to do that. I'd really like to meet all of you. And to close out, I want to thank Emil for his close collaboration on this project. His contributions are very important to having a good result here. And a lot of good things are happening at EBF, as Emil's about to tell you about. So thank you all very much for listening, and I look forward to meeting you. Thanks.
Chris, thank you so much. 2 quick questions. I don't know if you're still there. Can you still hear me?
Yeah, hey, I'm here.
Ah, perfect, Chris. Quick question. So could you first elaborate a bit on sampling? Like, who are these people that make up this data set? What's the demographics? Just a bit of an overview to understand what's the bias in that regard.
Yeah, this is a group of U.S. internet users. So it accurately represents the gender distribution of the United States, but it skews slightly younger than the actual U.S. population. And we also found that the participants are a bit more tech savvy than the average U.S. population. So we would consider it to be a sample of U.S. internet users, which is still valuable. But it's important to keep in mind that people who are tech savvy internet users younger would probably be slightly more interested in this topic than someone who doesn't have those traits.
Perfect. And then second question real quick. Did you ask the respondents if they're aware of funding cryopreservation via life insurance? Because then, of course, it becomes relatively affordable on a monthly basis.
No, I don't think we mentioned that.
Yeah, I don't think
we tended to communicate expense as a monthly cost, framing it as framing cryopreservation as coverage, similar to life insurance. So although we didn't specifically mention life insurance, I think that a similar effect was created in the mind of the respondent.
Perfect. Thanks a lot, Chris. Great talk.
Thanks, Emil.
As our last speaker in this session, Ashwin de Wolf. Ashwin is the CEO of Advanced Neural Biosciences, and their aim is to use low-temperature technologies to preserve human central nervous tissue. Ashwin, take it from here. The talk title is Outcome Metrics.
All right. Well, thank you very much. I would have really hoped to be in Switzerland. I'm sure it's not the first time you hear this today, but we'll do it like this. I don't have a whole lot of time. The title of my talk is Outcome Metrics, but I probably will just talk about one in particular. A very brief introduction in addition to what you said. So yes, my name is Ashwin de Wolf. I've been an ELPR member for almost 20 years. In 2008, I created this neurocryobiology company with Shana Fedra, and we're working hard to get some really good papers out right now. One was published this year. I also co-authored a comprehensive book about human cryopreservation procedures, and I'm editing Cryonics magazine for ELCOR. So, okay. So the topic of my talk is outcome metrics in human cryopreservation. I think like sometimes one of the assumptions, even within our community, is like until we attempt revival, there's not much we can say about how well a case went. Well, that is like not really correct. And we can actually get very specific about certain things. So let's start off with, let's see, slide. Okay, Sorry about that. So what are the elements of a good cryonics case? Now, this is of course very crude, but I think it captures the most important things. So, you know, ideally we'd like to keep it to 0 to 5 minutes of normothermic ischemia. Well, eventually we don't want any ice formation. We would like low cryoprotectant toxicity, and during long-term storage, we don't any fracturing. So how can we have a kind of a good idea whether we are achieving that goal? So Mike Perry and I developed a measure called the SMIX, which stands for Standardized Measure of Ischemic Exposure. And the fourth quarter issue of Cryonics magazine will have a more detailed mathematical exposition of how it works. Some of the work actually goes back many years, and if you are a reader of Cryonics magazine, there was actually an article by Mike Perry about cryonics modeling and math that discussed some of the preliminary work on that. But what the measure basically does is expresses the total ischemic exposure as an equivalent of normothermic ischemia. As you know, you're not always at normothermic ischemia during cryonic procedures. You have all different kinds of temperatures, but the SMIX transforms that into a measure of normothermic ischemia. Okay, so creating a measure like that can get very basic and it can get very sophisticated. And we try to kind of create a trade-off between these two things. If it's too crude, it doesn't tell you a whole lot more than the time a person was between the time the person was pronounced and the time cryogenic cooling starts. So two things we incorporated in our measure is Newtonian cooling and metabolic support. So Newtonian cooling is like kind of the cooling rate of a patient decreases as the difference between the patient and the cooling medium decreases. And that is for a given cooling method. So it's not the case that you will see the cooling go down, the cooling rate throughout the whole cryonics case. What is more likely, like during cardiopulmonary support and initial ice bed cooling, we see it increasingly like getting less and less effective. And then you go to washout and then you get much faster cooling rates. But that, you know, as sooner you get closer to 0 degrees Celsius, you get slower and slower. So our measure kind of incorporates that. The other thing that the measure incorporates is metabolic support, oxygenation, ventilation, because clearly if you provide sufficient metabolic support to the brain, there is no ischemia. So the SMIX for that portion would be 0. So the SMIX calculates the equivalent normothermic ischemic exposure from circulatory arrest to the start of cryogenic cooling. I mean, that is, I guess, somewhat arbitrary. If you're vitrification, one might argue that even like in the high sub-zero degrees range, there might be some ischemia, but I think in any kind of meaningful diffusion-based chemistry kind of stops when you start this really rapid cryogenic cooldown phase. Okay, so how do we achieve like the perfect SMIX, which kind of would be 0 actually? So you would not want any delays between circulatory arrest and the start of procedures. There would be adequate metabolic support during all stabilization procedures. Metabolic support during the initial stages of cryoprotection as well. I mean, there is some point still during cryoprotection perfusion, where you can still oxygenate the patient. And then you go to sub-zero perfusion for the highest cryoprotectant concentration. And if you would do that very meticulously, you would be able to calculate an SMIX score of 0. So that would be perfect. And I mean, I think it should not even be mentioned that it's kind of hard to do in practical cryonics. The only way that probably can be done is if you would have like hospital-based human biostasis. Okay. Okay, so here's a CT scan of an ELPR patient. You can see a scale, and the scale represents the different colors represent how much cryoprotectant there is in the brain. So let's look at the brain alone, and as you can see, it's very shrunken, which right now is indicative of a good case. There are a number of caveats about that, but that's not really the topic of this talk. And if you look at the brain, you see that almost all of the color corresponds to a concentration of M22 that is sufficient for vitrification. You see like some greener spots, and whether they make the cut or not to prevent ice formation, kind of hard to say on a CT scan, but this is a pretty darn good case. This is a case that was like rapid stabilization, local case, no delays, no out-of-state transport. Okay, here's some more math to put things into perspective. So Mike Perry and I did some math, and it turns out if you would only rely on cooling in cryonics, the cooling rate to stay ahead of 5 minutes of normothermic ischemia would be 2.89 degrees Celsius degrees per minute. Well, if you have any like experience with a cryonics case work, that is like totally impossible to achieve. Like even if you do ECMO-based cooling, that is a cooling rate that it's just not going to cut it. That's why providing metabolic support during all parts of the procedures is really, really important to get that S-mix down. Some other numbers for perspective. Keep these numbers in mind when you think about cryonics cases that do not happen locally, but where patients are shipped on water ice. So 24 hours of cold ischemia is 1.85 hours of normothermic ischemia. 48 is 3.69 hours of normothermic ischemia. 72 hours of cold ischemia is 5.54 hours of normothermic ischemia. Okay. So what do we know from experimental research? Well, one thing my lab has done very extensively over the years is to see what the effects are of ischemic delays on cryopreservation of the brain. And we've looked at conventional cryopreservation, but actually a lot of the results also apply to aldehyde stabilized cryopreservation. I mean, fixation does not reverse prior ischemia or seal like leaked vessels. So these kind of results apply to like kind of both procedures. So very briefly, after 6 hours of cold ischemia, you cannot really recover a functional EEG. This actually goes back to the work of Robert White. I think that was in the 60s where he did that. So that's where you lose viability by contemporary biological criteria. So what happens after 24 hours of cold ischemia? So there's no normothermic ischemia at all prior. Well, there's still breakdown of the blood-brain barrier. There will be vascular leaking. There will be cellular edema. There will be high intracranial pressure, cerebral edema, perfusion impairment, and that, of course, leads to ice formation. Okay. So this is an example of a CT scan where a brain was cryopreserved after significant ischemia. So there was unattended cardiac arrest at room temperature. So the person basically died without anyone present and anyone knowing. So it took a while. There was clearly no cardiopulmonary support. And then we calculated an S-mix for that of 12.72 hours. Now, so that would be the equivalent of 12.72 hours of normothermic ischemia. Now, if you look to the left and you look at the color scale, you will see in the brain mostly red and black, which is frozen blood or concentrations of cryoprotection that are so low that it basically is almost equivalent to doing a straight freeze. So this is pretty bad, but it shows you like what you get when you have an eye S-mix like this. Remember, I said a good S-mix, would be more in terms of minutes or less than an hour. And here we're talking about 12.72 hours. Okay, so I think what this pretty firmly establishes, at least within what I can say in this short presentation, is the limitations of what I would call the deploy and fly model, right? So like your cryonics organization is in, let's say in Switzerland, and you try to respond to a case in Arizona, in the United States. I mean, a lot of things come to that. There will be a super delayed response in case of sudden death. There will be delayed response in case of rapid decline. There might be compromised response travel time limitations, because right now with COVID-19, the brain is certainly no longer viable by biological criteria, but there will also be compromised ultrastructure, poor perfusion, and ice formation. So what are the solutions? Well, the best thing would be to build a cryonics facility in every state or country. Well, clearly that is not feasible right now. I wish it would. I wish cryonics would be more popular, but that's not the case. So another approach would be vehicle-based transport with metabolic support. So you'd basically stabilize a patient in the field, but then keep the patient on bypass with low flows and oxygenation. That can be done. It's logistically hard. And one of the drawbacks is if the patient is already fairly ischemic, then doing this like prolonged bypass will create a lot of edema. So I think that's not really an ideal solution. But in theory, it could work and really reduce this S-mix. I think the best approach will be what's called field cryoprotection. We don't use the word field vitrification because what we actually do is just do the cryoprotective perfusion in the field. Then we ship the patient on dry ice to any kind of cryonics facility for long-term storage and like the remainder of the cooling. So imagine field cryoprotection is basically a case where every aspect of the cryonics procedure will be done on site, with the exception, of course, of the long-term maintenance, which will be done at the cryonics facility. And this is my last slide already. Actually, so we're working right now with an entity called Blue Phoenix on the first whole-body field cryoprotection vehicle. So this vehicle in principle has all the equipment and supplies to do a whole cryopreservation from start to finish. And the idea, of course, of a cryonics rescue vehicle is not new. I mean, actually, the photo I'm using right now is the old Alcor cryonics rescue vehicle, which currently is the New York City cryonics rescue vehicle. And the one we're building will be bigger because it will have to be able to do whole body field cryoprotection with incorporate cryoprotective perfusion and all that good stuff. But I'm pretty excited about that. And this will be a practical solution to some of the issues that I mentioned earlier on, what you get if you cryoprotect a very ischemic patient. Okay, so thank you. I think I kept it pretty crisp and short, may even allow for a couple of questions. So a lot of the research that we do is supported by Biomedical Research and Longevity Society, the Alcor Life Extension Foundation, Blue Phoenix, as I mentioned. And I want to thank especially Dr. Michael Perry and Michael Benjamin for working out with me the whole S-mix concept and collecting all the data to apply that. If you want to read a lot more about my writings or see the first like hospital-based human biostasis protocol, you can go to biostasis.com. And I think that's it for me. And I want to thank Emil for having me. It's a pleasure.
Ashwin, great talk. Thanks a lot. Just real brief, 2 questions because we have 1, 2, 3 minutes. So you said 2.89 degrees per minute to stay ahead of ischemic damage. But what adjustment would you make if we would have CPS to remain in circulation and respiration?
Yeah, so that is like really excellent question. And when we're developing the S-mix, that was kind of a head scratcher. We have decided that when there is cardiopulmonary support, to do a reduction by 50%. The reason is that like, well, as you know from regular CPR, it's relative suboptimal compared to normal physiological circulation. When there is metabolic support during washout and bypass, we give the complete 100% discount. So during that period, there is no S-mix. So that is kind of, I mean, there are a lot more complicated issues here, but that's kind of to summarize how we've approached it so far.
Fair enough, great. And another question, if one wants to achieve reversible cryopreservation in adult mammals, which model organism would you use? Mice or bats was suggested. What would you suggest?
Yeah, I mean, small animal research is a lot easier, especially in the US. I mean, I think there's sometimes the misconception that a smaller mammal is easier than a large mammal, but in terms of the requirements of like cryoprotection and recovery, there's not a whole lot of difference. In fact, the microsurgery that needs to be done for small animal models is actually like can be a lot harder because it's a lot smaller, but you can probably do a lot more experiments in a small animal model. I mean, we have a project going to recover functional EEG after cryo protection and cryogenic cooling. So we're involved in that kind of research ourselves, but we're only confine ourselves to the brain, because if you want to revive the whole animal, you run into the issue that any element that is essential for recovery, for example, the lungs, which are notoriously hard to cryopreserve well, being so fragile, can be a showstopper. So we would be focusing in our research on recovery of brain activity.
Right. And lastly, is there a time frame when you want to be ready with field cryopreservation in some regions of the US?
Like the Alcor Life Extension Foundation is actually doing field cryoprotection. Right now it's confined to neuros, and they do it on a case-by-case basis. But I think you're gonna see more of it in the future, because there is emerging evidence that that outcome is better, even if the field cryoprotection procedures are slightly less sophisticated than in the Alcor operating room. That's an open question right now, but at least speaking for myself, I think based on the evidence I've seen in my lab, field cryoprotection is just the way to go. Not just for conventional cryoprotection, for any kind of preservation, if you want to like really cut down on that ischemic time. The vehicle we're building, it will be just a prototype, and what is going to happen exactly after we've shown like how to build that kind of vehicle, we don't know yet, but hopefully it will create interest and some kind of positive changes in the field.
Perfect. One question just actually came in, so since we have another minute, let's do one more. What do you think about Yuri Pichugin's work on opening the blood-brain barrier? Would you like to add, I guess, a blood-brain barrier opener to the standardized formula of M22 or VM1?
Yeah, I'm strongly in favor of it. We have some preliminary evidence in our lab that you can get better viability back from brain slices that have been put to a viability test after cryoprotectant perfusion, in which the blood-brain barrier was opened. I mean, you saw that on the CT scan, that the cryoprotectant-induced brain shrinking is very, very severe, and it is still an open question whether you can unload the cryoprotectant in such a way that it looks good again. But so far, I haven't seen anything that really impressed me, and it's not really hard to do to modify the blood-brain barrier to eliminate that cryoprotection, that dehydration. I should mention, of course, that if you have a lot of ischemia, the blood-brain barrier breaks down and you will not have any dehydration whatsoever. Now, of course, it is not the way you want to open a blood-brain barrier, because you will get a lot of intracranial pressure and edema.
Perfect. Thank you so much.
You're so welcome.
We will take a small break right now, and then come back with the last session of Biostasis 2020, and then, as I already mentioned a few times now, with a Q&A and panel at the very ending. So Ashwin, see you there. We will take a break until 4:40 CEST, which is around 18 minutes for everybody else's time zone. So see you in a few minutes. Welcome back to Biostasis 2020, and especially welcome to those from the West Coast who probably are now about to join or already have joined. We're now starting with our last block of talks, where of course, due to time zones, also most or all of the West Coast speakers will be partake. Our first talk is by Eric Vogt. Just as a quick introduction, Eric is a co-founder and cryomedic at International Cryomedics Experts, one of the standby providers that handles cases outside of the US and internationally. Eric, please take it from here.
Great, thank you. First of all, pleasure to be here. Thanks for inviting me to speak about standby stabilization and transport. We're really excited about where this is going, especially when it comes to field cryopreservation. And I really like to talk to everybody about some challenges that we've experienced when it comes to normal SST, and some lessons learned that we've learned when it comes to FCP. You know, currently the current method of SST, in which we know when it talks about deploying a team to the patient's bedside, having the team readily available with equipment. It's very effective when there are no time or distance challenges. And all the cases that we've been on, there's always been some sort of time or distance challenge. And that really means that those are the components that we take a look at and see how can we eliminate those time and distance challenges. If you have the foresight, though, you know, it's very effective. But getting that team to the location, the bedside of the patient or the member, whichever one you want to call it, is really the challenge. You know, as we talked about before, you've heard some people say the goal really is to have 24/7 operational readiness and infrastructure with vehicles and trained staff in every region. Jacob talked about having his vehicle ready in a certain location, and ideally we would like to have a facility and in every state in every country, but as you know, that's just not feasible. So when we talk about an optimal cryopreservation, we're really talking about getting the resources there at bedside, the standby, the stabilization, and the transport piece of it. Now, what I'd like to do kind of really is talk a little bit about each of these components and how that applies. When we talk about the standby portion of it, the standby is, although most people think that we're just kind of putting the team there at the patient's bedside and we're just waiting for legal death to occur. While that's true, there's other things that are going on in the background. So we really talk about preparing for the legal pronouncement, the logistics of getting the team and the equipment there first of all, setting up the equipment, preparing for the logistics after we do the standby, during the stabilization, how we're going to arrange to get the patient from whatever facility we're in into a facility that is ready for surgery. All of these things take a little bit of planning. And again, that takes time in order to do that. In an ideal world, you have enough time to prepare for all of this, and you're going to plan for each of these things. What we really want to do is we want to stand by as close to legal pronouncement as possible. That means we want to be at the patient's bedside, right at legal pronouncement, so we can begin our procedures to minimize that warm ischemia and even cold ischemia for those people that are being put on ice without the rest of the standby. It's very difficult to predict the date and times of legal death. I mean, sometimes we don't know that. We would like to have the luxury of knowing this in advance and be prepared for us to be there. But oftentimes we've seen that that's been not the case. And so, we're always in a rush to get to the patient's bedside. We're scrambling to get our equipment, there, whether it be by ground or by air. So really, we want people to be conservative. We want them to be a little conservative when it comes to deploying the team at the patient's bedside. Again, we want to be there as early as possible, ideally before legal pronouncement. That's the best case scenario. And obviously, we've had some challenges when it comes to COVID. COVID has really put a damper on how we operate, and especially when it comes to international travel, we've seen quite a few challenges when it comes to COVID. Really, what we're talking about here is time and temperature, the two main components which we're faced with. Obviously, the time piece of it, and then the temperature, eliminating that warm ischemia is extremely important. So the stabilization, and this is where I think a lot of people get confused. For me and my personal experience, really the stabilization encompasses quite a few different components. So what I really do is kind of break that down. Really, the stabilization involves rapid cooling, CPS, and then medication administration, and then an organ preservation washout. This is typically what we're used to, or cryonicists are. Understand when it comes to a stabilization. There's a lot of research that indicates that remote blood washout is superior to leaving the blood in the patient, which is very true. But it's also evident that prolonged ischemia is not beneficial to the patient. It could completely eliminated when there's smooth transition from stabilization to cryoprotected perfusion, and that's really what we're going to talk about when it comes to FCP. But this is kind of typical scenario when we think about stabilization. It's the cooling, it's the CPS medication administration, and then the organ preservation washout. So again, I break it down on the three components of the stabilization and to what we're familiar with, because really there's a lot that goes into the stabilization piece of it. When we talk about the rapid cooling of medications, typically what we do is we do rapid cooling via wet ice. A lot of people get confused when they say, ice in itself. Are we talking dry ice or wet ice? We're really talking about wet ice. We can't introduce cold temperatures of dry ice just yet because we don't have any cryoprotectant in the patient. CPS. Medication administration is going to mitigate some of that ischemia. And then a field washout, like we talked about before, which is organ preservation fluid. And this requires a surgeon and a perfusionist to be there at the bedside or at the facility in which we transport the patient to in order to perform the surgery to do this washout. We'll talk a little bit about some similarities when it comes to FCP and this field washout, and why it might be superior when it comes to FCP. The last piece of the SST is the transport. And really what we're talking about is how we're going to get the patient from the location in which they're either, facility in which they had legal pronouncement into the facility in which we provided the washout, and how we're going to get them to the long-term storage facility. And that's either going to be by air or by ground, depending on where we are. It could be by commercial flights or by charter flights. And then the last piece of it is going to be a domestic flight, or are we talking about international? All these things really provide or pose additional challenges to us, because each one of these components is a little bit different. Again, when we talk about what we're trying to accomplish here, time and temperature. Each one of these things here, you can see, is going to be a little different when it comes to time. Obviously, ground is going to be a little bit slower. The commercial flight is going to be a little bit slower than a charter flight. And then obviously, domestic is going to be a lot quicker than international flights. So when we're talking about a washout, organ preservation washout, we're still faced with the time constraints on this. Transportation to cryonics facility could be, again, I saw Jacob's nice vehicle there, and it's very impressive. And obviously, you would like to have one of these vehicles to take the patient to the facility to perform the cryoprotectant infusion. But that always isn't the case. And so when we do get there, obviously we have to do surgery. We have to get vascular access. The base perfusate is circulated and the cryoprotectant is slowly introduced over about 2 hours or so. And again, this is performed at the cryonics facility. So it's not performed at the bedside or at the facility that's near bedside. We actually have to transport the patient to a cryonics facility in order for us to do this. So what's the best case scenario? Well, again, it works very well if you have a fully trained and equipped SST team that's nearby and available. And again, time and temperature is really what we're faced with here. So we have to have somebody that is nearby. The transport time from legal pronouncement is short, depending on which cryonics facility you are signed up with. You'll have different requirements as far as the time from legal pronouncement to transportation to introduction of the cryoprotectant. Ideally, you would like that to be less than 24 hours, but we've seen some cases that have been extended beyond that. And you're also assuming that commercial air transportation is readily available. And as I mentioned at the beginning, COVID has really put a damper onto the readily available transportation. We've experienced that ourselves with international travel, very difficult to get to certain countries, a lot of paperwork that was needed, and we had quite a few challenges. Here in the U.S., we have two major cryonic storage facilities, Alcor in Scottsdale, Arizona, and the Cryonics Institute in Clinton Township, Michigan. So if you're taking a look at the continental United States, you can see that we can pretty much reach all of the U.S. Fairly quickly. We also are faced with a few challenges with COVID just interstate here in the U.S. So what's the reality of SST? Well, the reality of it is that the legal death, we want to make sure that we're there prior to the legal death, but often that is not the case. We're called at the last minute. It takes a lot of time in order for us to get to the patient's bedside, and oftentimes we're there post-mortem. You have a trained and fully equipped team. Most of the time, that's limited availability when it comes to fully equipped and trained teams. Most of the time, you're working with a lot of volunteers. I talked about the proximity to the cryonics facility. There's not many of them around the globe. And so we have that distance challenge in getting the patient from the bedside to the cryonics facility in order for us to perform the cryoprotectant infusion. And then the commercial air transportation, weekends, holidays, getting the paperwork available to transport is not always readily available, and so it makes it very difficult. So this is really the realities of SST. It's in a perfect world, things go very smoothly, but in my experience, in our experience with our team members, is there's always going to be hiccups, and we have to overcome those challenges. So the biggest one for us is really when it comes to the cryoprotectant perfusion, it must be performed within 24 to 48 hours of legal death. And going back to what I said before, time is of an essence. So the time portion of it is really what we're challenged with. Again, we're performing this cryoprotectant perfusion at the cryonics facility. So for those of us who've seen some of these things in the past where we don't have that time, the luxury of time, the typical outcome is usually a straight freeze. And that means we weren't able to get to the patient's bedside to perform the stabilization. We weren't able to introduce cryoprotectant in the field. And it ends up where the patient goes into a straight freeze. We put them onto dry ice and maybe ship them to the cryonics facility for long-term storage. So how do we overcome some of these things? And this is really where FCP comes into play. And you might have heard Ashwin talk a little bit about FCP. He's done a lot of research when it comes to FCP. And I really think that this is the future when it comes to standby stabilization and transport. So what is it? We're really talking about the replacement of blood and tissue water by solution of cryoprotectant agents. Really what we're talking about is the quality of care and how we're improving that quality of care when we eliminate the prolonged cold ischemia that's typically associated with remote cryonics cases. So, and again, if we don't have facilities that are nearby, we would consider those remote. If we have to take an airlines to the location of the patient, that's a remote location. What we're going to do is we're going to cool the patient. We're going to get the patient to dry ice temperatures fairly quickly. And that means that we would introduce the cryoprotectant before getting them to dry ice temperatures. But this is important. The dry ice temperature is important because what it does, it really buys us time. And then the packaging, prepare the patient for transportation. It will be done on dry ice. But we'll talk about some of the things as far as it eliminates some of those logistical challenges too. So the field cryoprotectant is really reducing the time between the legal death and the start of the cryoprotectant. We're not waiting to transport the patient from the bedside to the cryonics facility to perform the field cryoprotectant. We're actually doing it there near the patient. So sometimes we take the patient from their home or from the facility, and we will use a local funeral home, and then we'll introduce the cryoprotectant there in the field. If the time of pronouncement between legal death and the start of cryoprotectant is minimized, again, we can place the patient into long-term cryostasis without incurring unnecessary cold ischemia injury, and that's really what we're trying to prevent. So the standby of it really remains the same. Again, we encourage people to be very conservative when it comes to getting the standby team there at bedside. The cryoprotectant is introduced in a gradual manner. It's cooled as it's being infused into the patient. Refractive index is monitored, and then the flow rates are controlled by measuring line pressure. It is a little different than using a perfusion inside a controlled environment such as an operating room, but we've seen that it's been very effective. So I mentioned stopping time, and that's really what we're talking about. We're talking about stopping time and allowing us to have the luxury of making the arrangements to get that patient to a cryonics facility because they're not readily available, and they're not. We don't have those readily available around the globe. We cool them to dry ice temperatures for shipment to a cryonics facility, and we've eliminated that time urgency. We've seen that we were able to keep patients on dry ice for weeks at a time, and then ship them to the cryonics facility. Now, then, this is not a perfect world. Also, there are some shortcomings. Temperature and pressure cannot be precisely controlled during this infusion. It's a gravity-fed system. Again, it's not in a controlled environment like an operating room, but it is effective. The cryoprotectant concentration rises more rapidly, which is because of the gravity-fed system. We try to even it out with a teeter-totter, but it's not precise. The cephalon is perfused, while the rest of the body will not receive cryoprotectant, and this is some confusion. Also, some people think that we're talking about a whole body cryoprotectant. We're really talking about the cephalon only. And then dry ice is essential. We have to have dry ice wherever we are. So if there are some shortages of dry ice, or maybe we're in a remote location that doesn't have dry ice availability, this poses a problem to us too. We haven't seen that ourselves. Dry ice is fairly widely available. Excuse me. So we haven't run into that problem just yet. So really what we're talking about is a choice. The choice is between traditional SST versus FCP. When you have time and distance constraints, typically you're looking at a straight freeze, or if you're looking at FCP, we're talking about a cephalon cryoprotectant in which your brain is going to be protected with the cryoprotectant. And this is really kind of the difference between the two. What type of risk are you taking when it comes to traditional SST, and what type of risk are you taking or eliminating when it comes to FCP? It's interesting to think about when it comes to the FCP, and we talk about just cryoprotecting the cephalon only. But what we've found is that there is really no reason why we can't do whole body. And so one of the things that we're looking at now is whole body field cryoprotectant. If you take a look at the traditional SST, we're washing out with organ preservation fluid now. Why not just introduce the cryoprotectant at the same time? So the evolution, I think, and probably the experts who are looking into this, is that we may be able to do whole body field cryoprotectant. It's going to require a lot more cryoprotectant. Obviously, when it comes to transportation, we're going to have to take a lot more cryoprotectant with us. But there might be better outcomes when it comes to whole body field cryoprotectant. So just in conclusion, with FCP, we eliminate some of those challenges when it comes to the time, the distance, and really what we're talking about is preventing that warm ischemia, preventing this cold ischemia, and introducing the cryoprotectant right away in the field without the necessity of the operating room and the surgeon perfusionist inside the OR. And thank you very much.
Eric, thanks a lot for the talk. One question, one person asks, you mentioned that ideally there should be a preservation facility in every state or in every country. Why not a handful of the biggest cities like San Francisco, New York, London, and so on?
Yeah, great question. If we could have strategically placed facilities, that would be ideal. For us, I think we're talking about really getting the team there in a timely manner, and then from the time or from the location of the patient's bedside to the cryonics facility, you have to take that in consideration. So if they were strategically placed around the country, I would say that would probably be sufficient. But again, there's a huge investment, a lot of resources that go into that, and that may not be feasible based upon the numbers of patients that are being cryopreserved at this time.
Right, and then another question, which is more a general question, not necessarily but towards you, but I think you might have a good insight in that as well. Are you aware of anybody who's actively working on making legal changes? I mean, since you do international standby, you probably come across a lot of different legal systems. Like, what's your insight into that? Is there a huge difference, or is it mostly the same? What's your opinion?
There is a big difference depending on which country you're in. So even within the same country, there's differences. Canada, for example, there's differences within the country itself when it comes to the views of cryonics. So for the most part, we're seeing that we're able to do this in the majority of the countries, but there are some challenges when it comes to the legal aspects in different countries. So I would suggest that you start researching your country and the legal challenges within your own country, and reach out to some groups that may be in your country also, some cryonics groups. They may have some more information.
Mm-hmm. And one question just came in, so maybe last but not least. So it's actually two questions that I think can be combined into one. So one is, Eric, what is the availability or the variability and of availability of surgeons and perfusionists worldwide? And the other one is, how many professionals do you have available globally for FCP?
I can't answer the first question. I don't know that. There's a lot of surgeons and perfusionists out there. Are they well versed when it comes to what we're doing when it comes to cryoprotectant perfusion? That I really can't answer. I would assume that it's very low. Very few of them are familiar with this. And then as far as our availability, we have a team, a local team here in Phoenix, Arizona that consists of 6 team members. But we also have team members that are located throughout the United States, in Texas, in the Denver area, and the Seattle, Washington area, California, and in Florida. We also have some people that are in Hawaii. These teams are familiar with standby stabilization and transport. And we will typically send those teams with an expert that has been on many cases, either myself or another one of our team members, to be the lead. And typically it's a team of 4 people. But we have a pretty significant group of about 16 people who are readily available to be deployed.
Perfect, Eric. Thank you so much. Maybe see you later at the Q&A, because then we can answer. There's a few more questions coming in. But due to the timing, we would move on to the next talk.
Right. Thank you.
Okay, and actually as the next talk, I would give you a quick introduction of what we're doing in Switzerland and in Europe and in Germany with EBF and Tomorrow. Mostly though to answer your questions, not necessarily to present much, but mostly to take the time to answer questions about what's going on here in Europe and what's going on with EBF and Tomorrow. So first of all, maybe this very high level distinction between EBF and tomorrow, which I think is not necessarily always directly clear. So EBF is a pure research foundation. EBF will not do cryopreservations or anything towards that nature, but will fully focus on research, on biomedical research, on cryobiology research, and also on applied research. Tomorrow, on the other hand, is the, in quotation marks, customer-facing brand for everything that's in regards to sign up, to making it easy for people to sign up and all of that. And then secondarily, also for everything that is in relation to actually doing the cryopreservation procedures or the cryoprotection procedures, if we're more precise. And field cryoprotection is actually one of those things as well. Fundamentally, and this might unfortunately be a bit small, but I'm gonna read the points. Fundamentally, I think, or we think, there are two main things that can be improved in cryopreservation in this whole sector of biostasis to make this to generate more interest in the population. And one of the reasons why we're that interested as well is why we also did the study that Chris presented in a prior talk. He presented the US data. We also repeated this study with a smaller sample size, but with the same questions here in Germany. And the results are not exactly the same, but the tendencies, the trends are the same. And the idea basically being that there seems to be a significantly hidden interest, not necessarily hidden, but untapped at least, uncaptured interest that we're trying to capture. And then of course, what we're doing with that is increasing or having a significantly higher customer focus. Actually, it's not really correct that for customer focus, the EBF logo here it should be the Tomorrow logo, because Tomorrow will do everything customer facing. And then secondarily, scalability. Scalability, we heard it a few times during other talks today, will enable to make standby better. It will enable the whole sector to bring cryopreservation costs down, and so on and so on. So these two things, apart of course from also stability and medical quality and so on, which need to be there, otherwise this whole endeavor doesn't make sense anyway. But these are the two things: customer focus and stability and scalability. What we will focus on. And we do this with separating the organizations that are more or better at stability, and then organizations and activities that are better at being dynamic and being scalable. So when we start here with Tomorrow, as I mentioned, Tomorrow will be is the customer-facing brand of our whole group of organizations that we're building here in Europe. That will make it easy for people to sign up and then do everything standby related. Then for long-term stability, we're setting up a patient care trust that is very similar to what existing organizations like Alcor have, with of course a few smaller changes, but fundamentally the same idea. Then we have a research organization, which, as I mentioned, is called EBF. This organization will fully focus on research and on education. This is why we're here today, and this is one of the things where you see what we plan to do in regards to education. And then research projects and research endeavors, of course, are already ongoing and will extend over the next years. And then last but not least, which will be announced within the next month, is basically a long-term asset preservation organization that will be on one hand used for increasing security, but then also for funds in the future. And as I mentioned, this is now the data, and I think unfortunately it's also slightly too small to actually see on your screens. But just to walk you through it, this is the data that we saw in Europe here. And when we look at the higher levels of interest that Chris talked about, so where people don't not only say, well, cryopreservation or biostasis is generally an interesting topic that I want to look into further, but where they say, well, this is actually something that I really, really believe is something that I want to do myself. At that point in Europe, we have an interest, or actually this data is mostly from Germany, we have an interest of around 6% of population, which is in the ballpark of what we saw in the US as well. One big difference, the study in Germany is representative, whereas in the US, as Chris mentioned, it's the internet population. So, and now of course, if we take 6% of the German population, that's somewhere in the vicinity of 1.1 million, which of course is significantly and many orders of magnitude larger than how many people have cryopreservation contracts right now in Germany or in Europe or in basically everywhere on a percentage basis. So in the end, we believe that there is an untapped market that needs to be approached with new methods. And this is what we're doing with this organization that we've building. So we see I think 3 big problem contexts or sectors which hinders currently capturing this opportunity. One is membership. Being a member of a cryopreservation provider is very complex. It's very tedious. You need to actually put a good amount of work in to become a member, right? And last but not least, at least historically, in some circles, and mine included, it wasn't really necessarily a cool thing to do, right? So we're with Tomorrow targeting these changes and making a change here. Then secondarily, logistics are quite complex, and we heard that multiple times today with standby and stabilization and transport and so on. So for that, we're taking a new approach of how we're planning to acquire our members. And fundamentally, it kind of goes similar to a question that we already heard. It will be a city-by-city approach. So with a city-by-city approach, basically only marketing services and marketing the offer to people in a certain city, you reduce the complexity significantly. You can offer faster service, you can offer better service. You generate at some point at least some critical mass that then also generates word of mouth and a discussion around this topic, and ideally also makes it cool in certain in certain groups of people. And last but not least, I think we're trying to go a slightly different way with how we're building an organization. So I feel like, or the idea is to have a clear separation and not mixed focuses on organization, but have clear differences and and by that enabling scalability. And what I mean by clear focuses is basically that on one hand, we have organizations that need to be very dynamic. Membership acquisition is a dynamic process. You need to do marketing, you need to act like fundamentally you need to act like a tech startup that moves into a new sector. So you want to be highly dynamic. But then again, of course, in cryopreservation, you don't necessarily want to be highly dynamic, because you also want to make sure that quality and stability is an absolute given thing. And for that, we have kind of separated into 3 tiers of how we're acting or how we're balancing, on one hand, being very dynamic, and on the other hand, being very stable. Tomorrow is, of course, the dynamic one, doing customer acquisition and so on and so on. EBF is kind of in the middle with an oversight function, but mostly focusing on research. And then, of course, on the stability side is the patient care trust that has a full focus on stability with a large part of the statutes only dedicated to stability, and basically exact opposite of Tomorrow. And the patient care trust and EBF has an oversight function for Tomorrow to make sure that Tomorrow guarantees medical quality in everything it does. So just to give you a bit of an idea how this whole thing looks and how customers would approach it. So this is these are a few screenshots of our online sign-up flow and our app-based sign-up flow and app-based emergency app. Just to give you an idea, for example, sign-ups can be done fully digital. So you sign up digitally. We work with one insurance partner. So basically in the background, when you sign up, your life insurance contract is optionally done through us. So you don't need to find your own life insurance company which then insures you and understands what you want to do and all of that. But it's basically packaged in one product, where you sign up for a round, or you need to answer a questionnaire, because of course this is not a one-click thing due to the insurance, but it's around a 12-minute sign up. And then after around, if you don't have any medical problems, after 24 hours, the insurance approves, or well, slightly later, sometimes, let's say 48 hours, your insurance or our insurance partner approves your contract, and then you sign your final cryopreservation contract digitally, and then for added security, we would advise you to also put it in your last will. And this whole flow takes around 25-30 minutes of interaction. Of course, there's a bit of waiting time, but it's orders of magnitude faster than in the past. So 15 minutes interaction, and also moving this whole sector a bit to this coolness. Then standby, highly important. As I said, we're focusing on a city-by-city approach with that. So we saw one of these ambulance vehicles already in Jacob's talk. We have one here in Berlin that is fully equipped for field cryoprotection, basically a mobile operating room with a perfusion circuit, with a medical perfusion circuit actually, and so on, to do high quality standby stabilization and actually cryoprotection in the field for whole body. And then last but not least, we're building a cryobiology and cryopreservation research institute in Switzerland that will also double as a cryobiology in a long-term storage facility. Last but not least, this is us. Please reach out. First of all, also if you want to co-organize the conference, as I mentioned initially in my introduction, or if you're interested in what we do in general. And now, really briefly, I'm going to answer a few questions and then move on to the next speaker who's already waiting. So there was a question like, how far is the progress of building the facility? So due to COVID, unfortunately, we've been waiting for the building permits longer than we originally have intended. And we plan to, at the end of 2021, early 2022, the building will be ready. So at Biostasis 2021, next year in October, on the 22nd to the 24th of October actually, we're more than hopeful that we will be able to show you the building and show you the progress on site at that point. Last but not least, I don't know if there are any more questions, otherwise I would move on. There's a question about the combination of cryopreservation and assisted suicide in Switzerland, because Switzerland is a very liberal country. So while this is fundamentally, of course, of the interest for many people, we see a very, very clear separation between these two topics. So we would not get involved in it directly, at least not currently. In the future, this might be a topic that we're taking on. But again, like Switzerland is very liberal, so it can be combined, but it's not necessarily driven by us. For more questions, more than happy to answer them by email, because later on the Q&A and panel time, we will mostly focus on the other speakers. And now I would like to introduce our next speaker, who is Aaron Drake. Aaron, sorry. Hi, Aaron. As a quick introduction, Aaron has worked for many years with Alco and ICE as a medical response consultant, and currently he's the Director of Research and the Chief Specialist at Yingfeng Institute for Biological Sciences in China. Eric, please take it from here.
maybe it's afternoon or evening where you are. So again, my name is Aaron Drake. Just to give you a little bit of background about myself, I spent a little more than 7 years as the medical response director at maybe it's afternoon or evening where you are. So again, my name is Aaron Drake. Just to give you a little bit of the background about myself, I spent a little more than 7 years as the medical response director at afternoon or evening where you are. So again, my name is Aaron Drake. Just to give you a little bit of the background about myself, I spent all right. Hopefully I was getting the repeat of everything that I was saying before, so I'd like to start over. So again, my name is Aaron Drake. For about 7 years, I was working as the medical response director at the Alcor Foundation in Arizona. Was working a lot of international development at the time and really enjoyed that. Had the opportunity to branch out and do some consulting on an international basis. And I've been doing that for the past 5 years. The bulk of my consulting has been with the Yinfeng Life Science Institute in China. And that's kind of been my background. I've really had an opportunity to work on a lot of different aspects of cryonics, not just the standby stabilization and transport, but really in the development of a lot of different aspects of this program. And it's been a fascinating journey. I've enjoyed a lot of it. And I feel like I have a very broadened view of a lot of different aspects of this industry. What I kind of want to talk to you today about is what I see as the evolution of stabilization and perfusion, and where it may very well be heading. It's something that's already being done now, and I'd like to see if it can't be extended to the rest of the world. Let's go back and look at the history of cryonics, the early pioneers, if you will, those people that paved the way for us to be able to do our jobs now. Of course, cryonics is about 50 years old. And in the very early days, when the SST team would go to the hospital and introduce themselves and say, hey, we're here to have a patient be cryopreserved. They're going to be frozen. You know, the hospitals didn't want anything to do with these people. They were kicking them out, saying, you know, please go away. We'll call security if you stay around. And it was probably very difficult for them. Some success stories and some failure stories. It was really a science fiction fantasy, I think, by most medical professionals back then. And it may not have been articulated well enough for them to understand. And people may not have had an open mind about it. Over the decades, and specifically in the last decade that I've been in here, I've seen a lot of openness and intrigue about what we're doing. So even though there was some early pushback in the early days of when I started, now when we go to a hospital, there's a lot of intrigue. People are very excited. And I'm talking about the medical professionals there, the doctors, the nurses, the hospital administrators, rather than, you know, go away, you're not allowed to do this. There's excitement, and there's people, when they see what we're doing, the medical professionals, they say, oh yeah, I understand that. I do that same procedure. Oh, I see that piece of equipment. Yeah, we use that all the time. And so there's this development, this evolution of what medical professionals are seeing. And they're becoming a lot more accepting of that. Along with that, we've been able to have a lot more professionals on the standbys. Back in the early days, they were volunteers, many who had no medical training whatsoever, and they were just trying to put ice on and do some very simple things. Now you're seeing EMTs, you're seeing paramedics, you're seeing nurses, you're seeing doctors even go in and doing these procedures, participating in the stabilization. And I think there's becoming a much greater level of acceptance by the medical community, by the scientific community, as they see that we've upped our game, that our professionalism has gone quite a bit higher. I think we can continue to broaden that. And one additional step that I think will not only allow for us to have greater acceptance in the medical community, but will also be a huge benefit for our clients who become our patients, and that is ECMO. You may or may not be familiar with what ECMO is. ECMO is an acronym for extracorporeal membrane oxygenation. And I see this as the intersection of life-saving medicine and life-saving cryonics. Just to give you a little bit of background on ECMO, essentially, this is an external cardio and pulmonary bypass where an individual can be kept alive for an extended period of time by mechanical means. Currently, there are about 485 ECMO centers around the world. Last year, there were, I believe, 16,000 procedures, ECMO procedures that were done worldwide. So it's really becoming an intervention that is had great success around the world. The idea behind ECMO is if someone is in respiratory arrest, if someone is in cardiac arrest, we used to just be able to basically do CPR, intubate the patient, and try to ventilate them and try to keep them alive. But ECMO really has changed that in the sense that you can now keep an individual on ECMO without their heart beating, without their lungs working, for up to about 60 days, realistically. They have gone a lot longer than that, but that's kind of the time frame that most people have. So if we can keep bodies perfusing without their hearts beating, if we can keep that oxygenated blood flowing without them taking breaths, and do it by mechanical means and keep that patient essentially alive, why couldn't we use this application in cryonics? Well, we can. We're currently doing it in China, and let me give you a little bit of background on where I'm working in China, what we're doing there, and I'll circle back around to this ECMO and basically how we're doing it. So, as we mentioned before, Emil said I'm working for the Infeng Life Extension program, and if you don't know where it is, it's in Jinan, Shandong, China. Basically, it's a little bit south of Beijing, about an hour and a half by bullet train. The Infeng Group is a fairly large company. They're not just into cryonics. They have 4 major divisions here, as you can see. A little more than 7,000 employees over all the different groups. But the one group that I'm working with is their bioengineering, the group. You can see here's their latest facility that was just completed, which is about 1 kilometer away from where I'm currently working, and we'll be moving into this new facility here probably within the next month. They have their history basically is in cryopreservation of cord blood stem cells. And so they're very accustomed to working with dewars, to working with liquid nitrogen. And they really built an empire within a section of China with respect to storing the cord blood for newborn babies and using it for treatments for congenital diseases, et cetera. So they have a lot of experience in this particular area, and they've continued to really broaden what they're doing. Basically, they're looking at their biological program as a way to try to keep provide care really for their clients from birth until death. This involves doing congenital testing before the baby is born to look for any diseases, to harvesting the cord blood at birth and storing that, providing specialized treatments with that cord blood should they need it, providing care for people who may develop cancer, individualized cancer care, anti-aging techniques as you get older. And, should all those fail, then really the last step is to provide the cryopreservation. So they're really trying to provide the entire life cycle of the human and provide procedures for that. The program that basically they wanted to start was the cryopreservation program, and that's when I became involved with them in 2016. And in order to do that, they wanted to put together a large group of experts. I was taken over there under the National 1,000 Talents program and became a certified foreign expert in China. And they currently have about 26 employees on this cryomedicine team. Many of them are doctors. Many of them are medical professionals. Some are scientists and researchers, as well as just staff and some regular trained medical professionals. To really put this program together, they needed to develop quite a number of things. Really, they were starting from scratch. They wanted to build this from the ground up. So we had to really look at the infrastructure needed for a full service facility because they wanted to basically provide a service similar to Alcor. They were kind of modeling after that, where they had basically a long-term storage. But not only that, but they would have the medical teams that would provide the standby stabilization and transport at the hospital. They'd have the surgical teams that would be providing the perfusion. They would have all the administration aspect of this so they could begin to develop the clients and provide really a full service program. So they had to develop a lot of their own equipment and supplies. As you can see here are some of the doers that they've developed. They've got a lot of monitoring capability that they can access remotely. They have them in different sizes to accommodate different families. In China, there's a lot of interest in having the dewars individualized, so a family can purchase a dewar. So they want to store future family members in that dewar as they go. So they have them in different sizes for people to choose from. One of the other developments that they had worked on was developing a cool down box. This can take a patient from ambient temperature down to liquid nitrogen temperature in one controlled environment. It's computer monitored, computer controlled, and they can control the descent to exactly whatever profile they want to have for that individual. One of the other things that they've developed is their own cryonic surgical table. Basically, this is a very cold table that can run off either electricity or liquid nitrogen, and they can really control the environment of the patient while they're getting the perfusion done. So they've really kind of taken a lot of these pieces of equipment and either improved upon what was already out there in the community or developed it on their own. And so that's been, I guess I'm really proud of a lot of the things that we've been able to accomplish in those areas. The one development, though, that they've really, I think it's had a huge impact on the performance over there is that of the ECMO. This is basically our ECMO team over in China. You can see me in the center, and I'm flanked by all the doctors and the perfusionists who are involved in this program. So typically on an ECMO team, you've got a cannulating surgeon. You have an anesthesiologist who can also second as a surgeon. You've got an ECMO specialist, someone who can oversee the entire process. You have the perfusionist who's going to be there for the long haul, and then numerous medical people. So this is essentially the team that you're going to have, and we've been able to really develop this and do this for the patients there. To give you just a kind of a very brief view of what ECMO is in terms of the equipment. This is a diagram, basically shows essentially you're removing the deoxygenated blood from the body. You're running it through a filter to filter out any carbon dioxide, any impurities. You're running it through an oxygenator, so you're adding oxygen back into it. You're cooling it, and then you're taking it back into the body. And you can replace the heart and the lungs. So the idea is that we're actually going to set this up at the patient's bedside at the hospital. Essentially, whether the patient's in the ICU, whether they're in an operating room, the perfusion team is essentially going to go to the hospital and they're going to do a mobile ECMO procedure, where basically as soon as the patient has been pronounced, they can go ahead and start this surgical procedure right there and get them on bypass and start circulating oxygenated blood. Far superior than any external compressions that we can do. Obviously, the first level of compressions are just hand compressions, doing CPR. The next is we're using chest compression devices. All those, while can provide some level of perfusion of the tissues in the body with oxygenated blood, really pale in comparison to what the heart can do. This machine is even far superior. This will essentially do better than the heart was doing while the patient was kind of in their waning moments of life. And so we are now providing oxygenated blood to essentially keep that body alive physiologically. They're clinically dead because the heart is not working. They're legally dead because the doctor has made a legal pronouncement. But physiologically, we want nothing to change. And so in this photo, you can see the difference in the color of the blood. And that is essentially the difference between oxygenated and deoxygenated blood. At that point, we're basically going to load this mobile ECMO system up into the ambulance, and we're going to take it to the surgery center at Yanfeng. And from there, that system becomes their perfusion system. So while this is now a closed system, we're circulating oxygenated blood that's being cooled. Once we get down to that magic temperature where we need to start introducing the cryoprotectant, which is between about 15 and 20 degrees Celsius, now we just basically open the circuit and we can start introducing that cryoprotectant from where we were circulating blood and oxygen. So it's a very seamless way to keep the body very cold and allow the perfusion to occur without any delay and hopefully without any warm or cold ischemia that we've talked about quite a bit with previous lectures. So this is our way of saying thanks to the individual who has donated their body to this research project called Cryonics. And it's essentially what we do in China. So ECMO, I think, is something that could really play a big part. I think it's duplicatable in other parts of the world. And I'm going to be a big advocate and proponent of seeing if anyone wants to pursue this. I think that's something that's very, very viable.
Aaron, great presentation. Just two quick questions. Due to the timing,
Any
I
questions?
hope you will be able to be there later as well. But for now,
I'm not hearing
how many
anything.
patients are already at Yingfeng, and do you see how do you foresee the growth in cryopreservation there? I'm not sure if Aaron can hear me. Aaron, can you hear me? Aaron, can you hear me? I don't think he can. Maybe we can fix that in a second. Aaron? Aaron? Okay, Aaron can't hear me, and I also can't hear him right now. So we're going to move on to our next speaker and then bring back Aaron later and ask questions then. So as our next speaker, we have Professor Adam Higgins. right. Adam received his PhD from the University of Georgia Institute of Technology in Bioengineering and is currently an associate professor at the Oregon State University, where he works on cryobiology. His talk will be about strategies for reducing the toxicity of cryoprotectants. Adam, take it from you.
Thank you for the introduction. And so as you can see, the title of my presentation is Strategies for Reducing the Toxicity of Cryoprotectants. And I'll start, hopefully, by explaining the context. We're interested in vitrification methods, as I imagine many of you are. And the goal of vitrification is to bring the sample from an initial physiological state down to a final state down here below the glass transition temperature. And there are various ways of getting between this initial state and final state. But one kind of theme that is present in all vitrification methods is the requirement for increasing the solute concentration. And this allows you to reduce the melting temperature, reduce the isonucleation temperature, and make it possible to cool quickly into this metastable region below the melting curve and into the glass transition region without ice formation. So there are a variety of strategies that folks have used to try to overcome toxicity. One strategy is to try to increase cooling and warming rates. If you increase cooling and warming rates, you can get away with using lower solute concentrations. And so this is the strategy that for example, is being used with nanowarming methods that are currently being investigated. There are also molecules that directly interact with ice that can reduce the total concentration of solutes required to prevent ice formation, such as ice blockers or ice recrystallization inhibitors. Our focus has been, the focus of this presentation is going to be on selection of a mixture of cryoprotectants that will allow us to hopefully achieve a higher solute concentration, making it easier to prevent ice formation at practical cooling and warming rates. And we'll also be talking a little bit about how to select a trajectory between the initial state and the final state. Let's give a couple of examples. We could start at the initial state and cool down just above the melting temperature and then increase solute concentration gradually. This approach is known as liquidus tracking. We could maintain the physiological temperature and add cryoprotectants to increase the concentration and then cool rapidly to achieve this final state. And we're trying to come up with mathematical strategies, mathematical optimization strategies to decide which trajectory is going to be best. So I'm just going to start by talking about the mathematical optimization approach that we've been using. It has three inputs. There's a toxicity model. This toxicity model allows you to predict the toxicity of cryoprotectants during equilibration with a cryoprotectant prior to vitrification, and also during washout of cryoprotectants after vitrification. The osmotic tolerance limits here are limits on osmotic swelling and shrinkage. And these can be used as constraints in the optimization algorithm to prevent excessive shrinkage and swelling. And then we need some kind of mass transfer model to predict what happens when a sample is exposed to cryoprotectant. We take these three things and we insert them into an optimization algorithm, which explores a variety of different possible ways of moving between that initial physiological state and the vitrified state. And the outcome is a mathematically optimized method that's expected to be minimally toxic. We tested this with cultured endothelial cells. The mathematically optimized method was significantly better than the conventional approach for adding and removing these cryoprotectants. And the procedure that came out of this mathematical optimization algorithm was counterintuitive, at least to me. It's not something that I would have come up with based on conceptual reasoning alone. And so this, I guess we were pretty excited about this outcome. It shows that the mathematical optimization approach has promise, but there are some limitations that we're hoping that we can overcome to make this more broadly applicable. The first is that these methods were designed based on the toxicity of data for glycerol, and the only cryoprotectant that was used was glycerol. But most cryopreservation methods that are used for complex systems like tissues and organs involve vitrification solutions with multiple cryoprotectants. So we're hoping to extend this approach to enable prediction of toxicity for multi-CPA solutions. And then the other limitation is that it was applied to cultured cells, which made the mass transfer model pretty simple in comparison to what would need to be used for tissues and organs. And my time today, I'm going to focus mainly on this issue of the toxicity model and expanding that to account for multiple cryoprotectants. So I'm going to start by zooming in on the previously published data. This is for glycerol, and we recognize that in the trends in literature indicate that concentration matters, that toxicity of cryoprotectants tends to go up as concentration goes up, and that temperature should matter as well. And then it kind of maybe goes without saying that exposure time should also matter. So what we did is we did experiments for various glycerol concentrations and at 2 different temperatures. And for each condition, we used various exposure times to determine the kinetics of toxicity. And we measured a toxicity rate at each of these concentrations and temperatures and fit that to a model. So that was our starting point for optimizing methods for cultured endothelial cells involving only glycerol. So we want to extend this approach to multi-CPA solutions. There are a lot of CPAs out there. I'm going to focus on the kind of conventional permeating cryoprotectants. Glycerol is what we started with, but DMSO, propylene glycol, ethylene glycol, and formamide are also pretty commonly used in the cryopreservation literature. So we focused on those 5 single cryoprotectants. And we also wanted to look at mixtures. So we looked at binary mixtures of those 5 cryoprotectants and ternary mixtures of those 5 cryoprotectants. Again, we looked at multiple concentrations. So we had a total of 5 concentrations here that we looked at for most of these conditions. And we looked at multiple exposure times to enable us to estimate the kinetics of toxicity during exposure to each of these CPA mixtures. To begin with, we just focused on room temperature, but even limiting things to just 1 temperature, still we have a lot of experimental conditions that we're interested in testing. We're just looking at 5 conventional cryoprotectants and their binary and ternary mixtures, and that results in 340 different experimental conditions. There are over 150 unique CPA solutions to be tested. If we take into account that we have single CPA solutions, binary mixtures, ternary mixtures, and multiple concentrations at under each of these conditions. And since we can't directly expose to many of these CPA solutions without risk of osmotic damage, we did multi-step procedures to try to avoid osmotic damage for each of these procedures, and to carry out these multi-step CPA addition and removal procedures requires thousands of pipetting operations. So this is a pretty daunting task experimentally, especially if we are imagining doing this using manual pipetting with a multi-channel pipette, for example. So to deal with this, we turn to automated liquid handling. So we use the high throughput screening services laboratory at OSU. We got access to a Hamilton liquid handling robot there. And the basic idea behind our experiments is to use this 96 channel head in the liquid handling robot to transfer CPA solutions from a source plate to an assay plate where the cells are cultured. In each of these, we have multiple source plates for each condition. These source plates would contain the solutions for each step in the CPA addition and removal process. And the source plates were randomized so that we could have randomized treatment conditions on the assay plate as well to improve the accuracy of the results and reduce bias. So if this is what we're trying to do to add solutions to the assay plate where the cells are cultured, we end up with this randomized source plate. And the randomized source plate is going to contain solutions with a variety of different compositions. It's nearly impossible to do that by hand. Fortunately, the automated liquid handling robot has another feature that's useful for us. It has 8 independently moving pipette channel heads, and those can be programmed to move between tubes containing stock solutions and source plates. The stock solutions contain the 5 different CPAs that we're interested in at the highest concentration we're interested in, as well as buffer solutions like isotonic buffer and hypertonic buffer. And by combining stock solutions in different proportions, we we're able to generate all, more than 150 different unique compositions, starting with about 7 different stock solutions. And we could do this, we could program it to place each of these solutions in appropriate proportions in the appropriate well of the source plate to generate these randomized treatment conditions. So this is our automated liquid handling system. It turned out to give us several benefits, and I'll kind of go through those. The first is that it basically made it possible to do randomized treatment locations on the 96 well plate. This would be pretty much impossible to do by hand. Each of the colors shown in the picture here is a different solution composition, so you can't use a multi-channel pipette in this case to fill this. You'd have to fill using a single channel pipette, and that'd be pretty challenging to do by hand. This can all be programmed into the robotic liquid handler, and it does it for you. So randomized treatment locations are known to decrease systematic bias in well plate assays. Edge effects is a common problem due to the different conditions at the edge of the well plate. So people have seen systematic bias as a result of that. You can also end up with systematic bias due to manual pipetting issues. If you're using a multi-channel pipette and something happens with that multi-channel pipette and during one pipetting operation, it could have an impact on an entire row of samples, which could skew your results dramatically if it's not randomized. So this is a benefit of automated liquid handling. We can decrease systematic bias. Another benefit is that it increases pipetting consistency. So these are pretty complicated procedures. They involve multiple steps during CPA addition and removal. Those steps have to be precisely timed so that we can avoid osmotic damage. That can all be programmed into the robotic liquid handler. The other thing that could be adjusted is the offset between the surface of the culture plate and the pipette tip and the aspirate and dispense rates. So we can optimize those settings to ensure that we don't disturb the cell monolayer. This shows shearing of the cells off of the monolayer before optimizing settings, and this shows the cell monolayer that's intact after optimizing the settings. So with manual pipetting, it's possible for there to be variability in how that pipetting operation is carried out that could lead to loss of cells due to monolayer disturbances. So these things together ended up giving us improved results compared to our previous experiments. This shows results for 5-mol glycerol. The red shows our previous results. The blue shows our new results with automated liquid handling. And there's 2 things to point out here. We have lower variability with automated liquid handling. Our error bars, our standard deviation goes down by about a factor of 2. And in general, we're getting higher cell viability with automated liquid handling, especially at low time points. And this is potentially attributed to better, less monolayer disturbances in the automated liquid handling case. And another sort of maybe more obvious benefit of the automated liquid handling method is that it's faster. We're using a 96 channel head to do the solution exchanges. So that's about 96 times faster than manual pipetting with a single channel pipette, or about 10-ish times faster than pipetting with a multi-channel pipette. So we use this approach to generate a lot of data quickly and accurately. This shows results for single CPA solutions and the binary mixtures for 5 different CPA concentrations. This is the original data set that was fit to determine toxicity rates. And the toxicity rate constant is essentially just an exponential decay fit to the data. So we'll talk a little bit about some specific cases of for these toxicity rates. If we just look at the CPA solutions, the single CPA solutions, we see some interesting trends that are consistent with what others have seen in the literature. Propylene glycol and formamide show the most toxicity among the 5 that we tested, which was not particularly surprising. And ethylene glycol and glycerol were the least toxic among the CPAs that we tested. We can also take a look at the multi-CPA mixtures. So this graph here shows single CPA solutions at the far left. In the middle are binary solutions, and at the right are ternary solutions, mixtures of 3 CPAs. One trend that's pretty apparent by looking at the graph is that the more CPAs you add to the solution, on average, you end up lowering the toxicity. And this is mainly because when you are using a multi-CPA mixture, you end up diluting some of the most toxic CPAs down to lower concentrations. The other thing that we could look at with in this data set is the potential for synergy in multi-CPA mixtures. Fahey and colleagues has described neutralization of formamide toxicity by dimethyl sulfoxide, by DMSO. We looked at this. We also actually noticed even stronger toxicity neutralization for glycerol. So I'll walk you through that data set real quickly. We'll start by looking at the formamide toxicity rate. We see the toxicity rate for formamide that goes up with concentration. If we essentially take these same formamide solutions and add DMSO, we end up with this green curve. And so if we're adding DMSO at lower concentrations, we end up with a lower toxicity rate compared to formamide on its own. So there's some toxicity neutralization effect for DMSO. If we add glycerol to these formamide solutions, we see an even more noticeable effect, particularly if we look at the 5 mol concentration. So this shows the toxicity you get for 5 mol formamide plus 5 mol glycerol. And this shows what you get when you just have 5 mol formamide on its own. By adding glycerol, we reduce toxicity by about 3.5 times. So our goal is to figure out how to come up with a mathematical model of toxicity so that we can predict the best mixtures of cryoprotectants for cryopreservation, and to also use that model to design the best trajectory between the initial physiological state and the final vitrified state. Previously, we used a pretty simple model for glycerol toxicity that looks a lot like chemical reaction kinetics. We want to extend our modeling approach to multi-CPA solutions. If we just take the sum of these types of models for glycerol, but for all of the CPAs in the mixture, this would be accounting for the specific toxicity effects of each of these CPAs. It doesn't actually work very well to make predictions for mixtures, especially at high concentrations. At high concentrations, the data shows higher toxicity than what you'd predict using this specific toxicity model. And this has been observed previously too and attributed to a total CPA concentration effect, or perhaps an effect on the concentration of water. And this is thought of as a non-specific effect of the cryoprotectants. So we added something to try to account for that into the model, and we added an interaction term in the model to try to account for synergy. And this initial model was used to make predictions. We fit the model to the single CPA and binary mixture data set and use it to make predictions for ternary mixture toxicity. And there's some agreement between the predictions and the data, but it's not really perfect. There's generally an overestimation of toxicity in the model compared to the data. Although we did predict accurately the lowest toxicity mixture and the highest toxicity mixture. There's clearly some room for refinement there. So to sum up, we've developed this high throughput method using robotic liquid handling for assessing toxicity of CPAs and CPA mixtures. We've got an initial model, but we're interested in refining that toxicity model, and we're working with a colleague who's a physicist that's an expert in analyzing large data sets using complicated multi-parameter models. So we're exploring different model forms and working with him to assess that. We're also interested in examining temperature effects, and we've outfitted the robotic liquid handler with a custom plate that allows cooling or warming of the assay plate and the source plate. It's connected to a temperature controlled bath, which is shown in the back here, so that we can control the temperature during these robotic liquid handling experiments. And we're also interested in expanding these experiments to cover a wider range of potential cryoprotectants. We focused on 5 commonly used cryoprotectants, but there are a lot of chemicals that are similar that have not actually been tested to my knowledge. So the main features of these conventional cryoprotectants is that they're fairly small molecules, they're uncharged, and they're completely miscible in water. If we use those criteria and just look at the CFC handbook, chemistry and physics. There's a ton of different possibilities. I only included a few of them here to give you an example up to a molecular weight of 62, but there's hundreds more. So there's lots of possibilities now for taking this approach and expanding it for testing a wider range of cryoprotectants and looking for new mixtures of cryoprotectants that might be less toxic. And another area that my lab is interested in and working on is figuring out how to extend the mathematical optimization approach to tissues and organs, which will involve development of mass transfer models that will be appropriate for tissues and organs and better understanding that cryoprotectant transport process in tissues and organs. I'm not going to have time to talk about any of that work today, though. And so with that, I just wanted to acknowledge funding for this. Hirsch Foundation and NIH grant, the Cryonics Institute provided funding. We worked with the assistant director of the College of Pharmacy High Throughput Screening Services Lab for a lot of these experiments, and there are various students who are also involved. Well, they're the ones who did all the hard work. And I'll stop there. I don't know if I have time for questions. I tried to limit it to, just went automatically to the next slide. Tried to limit it to 20 minutes.
Let's do a few questions, because I have 3 quick questions. Maybe you can answer relatively quickly to go on with the next speaker. So first one being, are all these CPA mixtures mixed in equimolar concentrations?
Yes, that's what we started with. We started with equimolar concentrations, and I guess if you expand to a wider range of ratios of concentrations, there's an infinite number of possibilities. All of that can be programmed into the liquid handler if we want to test that.
All right, fair enough. Then you mentioned that the mathematically optimized cryoprotection protocol was counterintuitive. Could you elaborate there a bit? What was the counterintuitive nature, I think, is the question.
Sure. So generally when we think about addition of cryoprotectants, the focus is on using multiple steps to prevent excessive shrinkage because cryoprotectant solutions are generally hypertonic. It turns out if you use a hypotonic carrier solution for the cryoprotectant, you'll get initial shrinkage, but it will induce swelling after that shrinkage period. And the swelling during CPA addition turned out to be really helpful. We actually tested that by comparing CPA and hypotonic buffer to CPA and a conventional isotonic buffer, and the benefit of the optimized method went away when we used the isotonic carrier solution for the cryoprotectant.
Okay. And last but not least, one question saying, it looks like you added the summation of rates linearly. Any reasons to make that assumptions? Are all mechanisms on the same order, of the same order?
That's a good question. So initially when we were trying to just take our original CPA toxicity model and extend it to multi-CPA solutions, we considered, we have a mixture of glycerol and DMSO, for example. If we had 5 mol glycerol and 5 mol DMSO, we plugged in, we predicted the concentration that you get for 5 mol glycerol on its own and added it to the toxicity you get for 5 mol DMSO on its own. That would be what you'd expect if those 2 CPAs are exerting toxicity through independent mechanisms that were specific to those CPAs. But that didn't really work very well. There seems to be some effect that has to do with either interactions between the CPAs or their non-specific effects on the solution. Like as you add more and more CPAs, those CPAs are going to reduce the amount of water present in the solution, and that could also be resulting in toxicity. So we attempted an initial model that tried to account for all of those potential mechanisms of toxicity, and it worked somewhat, but we're still trying to refine that and improve the correspondence, I guess, between predictions and the data.
Perfect. Thanks a lot. Looking at the time, we need to unfortunately move on, but there might be more questions, and you might be able to answer more questions later in the Q&A and panel session.
Thank you.
Our next speaker will be Robert McIntyre. Robert has a master's degree in artificial intelligence from MIT, and in 2015 founded Nectome, a company that develops biological preservation techniques to better preserve the physical traces of memory. And that company was, among other things, also part of the prestigious Y Combinator program in 2018. Robert, take it from here.
Thank you, Mel. It's a pleasure to be here. So what I'm going to provide for you today is I'm going to go over the idea of engram preservation or memory preservation. And so this is the idea that we can preserve memory engrams, the data that's stored within neural tissue, using chemical fixation, which is binding proteins together with embalming chemicals, such as glutaraldehyde and formaldehyde. And so by the end of this, I hope you'll understand a little bit more about the information theoretic aspects of what it is to preserve something and why we can apply those to probably be able to preserve information that's stored in neural tissue. There's 4 parts. We're going to talk about what it means to preserve something. We're going to talk about how that might apply to the brain and whether the brain stores information in more of a static form or more of a dynamic form. We're going to talk about the very compelling neuroscience evidence that we've gathered over the last 30 years that relates to how the brain actually encodes memories. And then we'll briefly touch on the mechanism of chemical preservation and just how good glutaraldehyde is able to actually preserve information. So the larger context of this endeavor to preserve information, I think, falls within human history. In general, the better we've gotten at preserving information, the more we've increased the power of our society. So before we had language, you had intuitive history. This is just what you can personally observe and what you can go with other people and observe. If you have language, you can then build an oral history. You can start having a more complex society. And language, in my mind, is a way to preserve information. You can preserve a story and have it even though you haven't directly experienced it. The next step up from having language is to have written language. And now you can have the precise words that someone's spoken in the past. And I would say that that's essentially the era of history that we're in currently, the era of written history. And that's very powerful. It's led to our civilization today. But I would say that the majority of information that's actually stored within people, we're not actually capable of making that permanent or writing it down or archiving it in any sort of way. If you imagine the collected works of a person, all the things they've written, compared to what they're able to produce, right? Or you imagine the skills that a doctor has compared to what a medical textbook can show you, there really is quite a lot that's left behind. And so if we can preserve information that's stored in neural tissue, then I think that leads us to a new era of history called the era of living history, where we have access to the totality of information that's stored within brains. So the question becomes, what does it actually mean to preserve something? Because I think we all have a somewhat common idea of what it means to preserve things, but especially when you get into considering preserving complex biological systems, it can become a bit muddy. And so information theory can help us out here. And the idea of preserving is that you keep separate things separate. What does it mean for 2 things to be separate? It means that we consider them to be meaningfully different symbols. And transmission involves preserving and then transmitting. And preserving means that you keep different things different. All right. So in the context of preserving a book, for example, we both intuitively understand that burning a book doesn't really preserve the information that's contained in it. Why is that? Because 2 piles of ash essentially can't be distinguished from each other. But we also intuitively understand that you can transform the book in many quite extreme ways and still meaningfully preserve the information. So for example, if you scan the book and make a microfilm of the book, or you put it on the internet, or if you were to pour epoxy on the book and solidify it into a solid object, we understand that that would preserve the information. Why is that? Because if you have 2 different books that meaningfully have different information content, and you apply these types of operations to them, the artifact that you create from them, whether it's a website or whether it's a microfilm or whether it's an epoxy coated book, those are meaningfully different. If on page 52 of the book, you have an A in the third place, and on page 52 of a different book, you had a B, the epoxy is going to preserve it, the microfilm is going to preserve it, etc. So that's the first critical idea here, is that preservation means that differences stay different. That's what it means to actually preserve information in a more formal sense. And an important note here is that you can preserve something while being completely ignorant of what it actually means. So no one knows how to read linear B, for example, but it's quite easy to preserve linear B, even a large whole library of texts that were written in that language, because we know how to preserve the documents and we know how to preserve the forms in which it's written. Similarly, I think that, today we can begin to understand DNA and begin to even print DNA on our own. But the first time when we were able to preserve DNA would have been in the 1960s, when we knew enough about the structure of DNA that we could, with confidence, say that we could preserve its physical structure, even though in the 60s we didn't understand enough to read the DNA or print it or really do anything other than preserve it. So as we consider whether we could preserve memory engrams, the most natural question in my mind is, how are these memories actually stored? If they're quite physical, then they may be easier to preserve. But it's possible that memories may work as some sort of very ephemeral, sort of electrically mediated activity, and that any loss of that activity may erase those memories. And so for this, I think the most compelling evidence is deep hypothermic circulatory arrest. So this is one of my favorite medical procedures, and it's not as well known, although probably better known amongst this group, where a person can be cooled down to very low temperatures such that the electrical activity in their brain ceases. And yet, when they're warmed up properly, they can retain their memories and personality. And in fact, this is a figure from this paper where they took about 30 people who underwent DHCA and 30 who didn't undergo DHCA. And then they had a questionnaire by their families and their employers and themselves to see, well, do you feel different? Does their family see them as different? But also importantly, are they still able to function in their job as they did before? And each of these jobs are what they considered high cognitive demand jobs. So this is the result, and essentially this medical procedure, even though it zeroes out the electrical activity in the brain, does not lead to a loss of memory. So I would say we have very strong evidence for over 50 years that memories are physical in some way. And the question becomes, well, then how does the brain create and store memories? And to this we turn to neuroscience and the concept of memory consolidation. So, again, we're going through 4 different components here. What does it mean to preserve something? Our memories, and we learned that that means keeping things different. Are memories actually physically stored, or are they more electrically mediated? They're probably physical. Now we're going to look at what does neuroscience have to say about how these are actually stored. And to this, we have a tremendous amount of research over the last 50 years that's led to this concept of memory consolidation, that when you create a new memory, within the first 30 seconds, it's very electrically mediated. In 2 hours, it's unstable changes to synapses, meaning that there's the beginning of remodeling the synapses, but that it can disappear over time. And then greater than 2 hours, it's a permanent structural change to synapses. And these are very immensely complex structures. So this is a picture of what a synapse looks like at a protein level. I mean, it's a mess of very complex organization. And essentially, in order to write a memory to the brain, these individual synapses, which are these glowing dots in this 2-photon experiment, can change their size and change their strength and change the communication that's happening between 2 neurons. So in this case, they are watching a living brain as it's creating a memory, a very complicated and amazing and beautiful experiment here, where you see the synapse highlighted in red becomes larger, and then that change is stable over more than 2 hours. And so what you're looking at is the way in which brains create memories. If you go in and prevent these synapses from changing by preventing protein synthesis, or if you go in and intentionally reset the synapses, as you can do with optogenetic techniques, you will either prevent the memory from being formed, or you'll retroactively erase the memory. So there's a massive amount of evidence that it's synaptic changes that store memories at the lowest level. And I'd say this is equivalent to understanding that DNA stores information in nucleotides. What does it mean at a systems level that a synapse change? In what ways memory is actually encoded? I'd say it's still a mystery. But at the lowest level, how does the brain actually write information to itself? It does that through memory consolidation and the process of protein synthesis. So neuroscience, I would say today, gives us the most basic idea of how information is stored in the brain. It's like we understand how ink works on a page, and we understand how paper works, and we understand that if you want to tell a different story, it involves changing some ink in some way. But we don't understand a huge amount beyond that. Yet what we do understand is enabling us to preserve this information. And so the question is, can you preserve the state of these synapses using some method? And to make this a little bit more precise, I think it's helpful to think back to the book analogy. So, can you preserve two? You want to keep different things different. Could you preserve two books that are very different? Well, if they are very, very different, any variety of methods might keep that difference. So, for example, if you had a very small book and you burned it into a small pile of ash, and you had a very large book and burned it into a large pile of ash, those would still be different. So that's not a very interesting comparison. That actually does preserve that difference. What's more instructive is to consider two books that are identical copies of each other, except for a single letter being out of place. If your preservation technique can still keep those separate, then your preservation technique likely preserves books in general. And so the question here is, if you imagine two organisms that are virtually identical to each other, except there's, say, one synapse difference, would you be able to preserve that with a preservation technique? I think that's the right question to be asking when it comes to thinking about memory preservation. And for this, we turn to chemistry and we think about glutaraldehyde. So glutaraldehyde is it's used in various industries as a sterilant or an embalming chemical, and it's also used in neuroscience to prepare brain tissue for imaging. And it's an amazingly good molecule at preserving structure and also preserving the protein content of the brain, because the way that it works is by binding proteins together into a solid gel. So it's literally like a pair of molecular handcuffs that grab proteins and bind them together. And what you're seeing here is a 2-photon imaging of a living process in the brain. So the long glowing tendril is the process, and the bright glowing dots around it are synapses. So each of these tiny glowing dots is one of these complex protein structures. And then the lower picture is this same exact process that was then preserved with glutaraldehyde and then imaged with an electron microscope. And so because of the much more powerful imaging mechanism used, you have a higher resolution. But the important thing to consider here is that every single one of these glowing synapses that was observed with the 2-photon microscopy is still preserved after being fixed with glutaraldehyde. And this is a very difficult experiment to do because you have to actually see the process that you're interested in and then dig it out of the preserved sample afterwards. So this is kind of a tour de force of imaging. But it looks like a more higher resolution version of the image. So here's 3 synapses, for example, and here's 2 that are up here. And now you have 3 and 2 here. And you can now work out exactly how they're connected. And so even an individual synapse doesn't get deleted or changed with glutaraldehyde preservation. And so that tends to imply that we could preserve memory engrams, even though we can't understand how memory engrams work today, and puts us in a similar position as someone in the 1960s saying, well, here's a species that's about to go extinct. And certainly it's not possible to preserve or to write DNA or to actually resurrect an extinct species. But because we know the structure of DNA, we can preserve it and we can store it for a long time. And eventually we might be able to access that DNA. So yeah, I guess I'll take questions now.
Robert, thanks a lot. Just one question came in. Who and what is currently the market, the target market for Nectome and its services?
We're still working on research. So I think that it's absolutely critical to preserve human brains in a research context first, before making any claims about being able to preserve human memory engrams. Currently, we've preserved animal brains.
We just lost you.
I got muted.
Now
Okay.
you're back. Perfect.
All right. So we preserved animal brains, and those won the large and small mammal prizes, and that work was done at 21st Century Medicine. And is it likely to scale up to human brains? Certainly, because it's already scaled up tenfold from a rabbit brain to a pig brain. But the devil's in the details. And so the next thing to do, I think, is to run a brain banking program where human brains are preserved in a research context, and then they're analyzed with electron microscopy. And the idea is to get a similar result, or to at least describe the conditions under which you can get similar results to what won the large and small mammal brain preservation prize.
Right. One more question just came in. How's the electrochemical content of synapses preserved when using glutaraldehyde?
So can I share the screen again?
Sure, go ahead. I hope you can.
All right, cool. Can you see the screen?
Yeah.
Okay, so here's a paper where they wanted to see about the protein content of cells and whether they're preserved. And so what they did was they took fresh frozen tissue, and then they also took formaldehyde fixed and paraffin embedded tissue. So that's FFPE. And so this is actually a more extreme process than what's used in ESC. Not only did they fix this tissue with a looser fixation agent, they also then like dissolved the lipids out of it and embedded it in wax, essentially. And so what they did here was they dissolved the proteins and extracted them down to the amino acids, and they used mass spectroscopy to compare the overall amino acid content. And what you can see here is that the yield of proteins and the yield of peptides for the FFPE and the fresh tissues indistinguishable. Essentially, all the proteins are preserved, even by this more aggressive fixation than what's used in ASC. And that makes sense if you think about this protein, the synapse as this vast collection of proteins that's almost like supersaturated with proteins, and you consider just gluing them all together into a gel. Well, it already held itself together before. Now it's much stronger, and these proteins are all retained. The things that you lose during fixation are very small molecules and ions and things like that are more transient.
Right, thanks for answering. All the other questions, actually a few more came in just now. We need to, actually we have a bit more time, I just heard, because we're still waiting for the next speaker.
Okay.
So let's do a few more questions.
And this is a 3D image, by the way, that we're seeing now. I'll just put that up in the background. But this is one of the cells that was actually preserved, and you're seeing it. Oh,
right. Yeah. One more question. How important would you consider the use of glutaraldehyde in current cryopreservation and cryonics?
If you want to preserve the structure in a form that is interpretable by neuroscientists, right, in a way that you can compare to the published literature, then I'd say currently it's critical for whole brain samples. So there's been some interesting work with slices of living tissue that have been cryopreserved and then warmed up and then fixed and then imaged. But if you want an entire brain preserved and you want to get good electron micrographs, currently you need to use fixatives to do that. And the reason for that is, I think it's been discussed in previous talks, but basically you have a lot of shrinkage caused by cryoprotectants under normal conditions.
Right. Perfect. Thank you for answering that. I'll check if we are ready with the next speaker. Perfect. Robert, thanks a lot. We'll get you back in the Q&A later and have a few more questions for you. But for now, okay, I just heard he left again, so we can do one more right now. Greg, I think he's just there now, I think. So I think we're switching over to Greg Fahey.
Well, I salute you all. It's been a pleasure talking with you.
Talk to you later. Yeah, click it.
See that?
All right. Greg is one of the leading cryobiologists and biogerontologists. He's the Vice President and Chief Scientific Officer at 21st Century Medicine and has recently also co-founded Intervene Immune, a company developing clinical methods to reverse immune system aging. His talk today will be Cryopreservation Works.
Okay. Slightly different title, but the same idea. So I'm going to bring you up to date with some recent research in complex system cryopreservation. The reason being that this is the view of the subject of this meeting by many scientists. I'm all in favor of never giving up, but there are lost causes. And what can we say about science that may go a little bit farther than just optimism? So the first thing is that there's a general a statement that I can easily make, which is that many different complex systems can be cryopreserved. Back in Madrid in 2017, I summarized work that was just done at our lab, 21st Century Medicine, showing a wide variety of successful cryopreservations of pretty advanced systems, including pancreatic islets, liver slices, engineered skin, cartilage, corneas, and of course, brain slices. And in the case of brain slices, we were able to preserve both the ultrastructure and the viability of rat brain slices on the lower left. And in the case of rabbit brain slices, we were able to preserve the LTP response, which you see in the crosshatch bars is no different than untreated controls. And the last little panel at the bottom shows a frame from movies that we made of electrical activity coursing over the surface of these brain slices after they've been vitrified and rewarmed, thereby demonstrating no difference between vitrified brain tissue and control brain tissue. But these are just tissue level preservation examples, and what we would really like to see is some progress with whole organs. So in 2009, our lab published the survival of a rabbit kidney that was cooled to -130. All parts of the kidney were at -130 before rewarming, as you see in the black lines on the left. And the kidney did support life for 48 days until we took it out and had a look at it. As you can see, the inner medulla was partly preserved, as you see in the image on the left bottom, but the other half of the medulla was destroyed. So we did have a successful demonstration that a vital organ can be preserved, but not completely. So we've been trying to improve on that ever since. We're now at the point where we want to be able to get rid of that ice damage. And the way you do that is by warming very quickly. In our lab, we use radio frequency warming techniques. Other places, they have other methods, but this is what we use. And so we're now looking at the physical steps required to apply RF warming to vitrified rabbit kidneys. And the first issue that arises is that in order to apply this energy to the kidney uniformly, the kidney needs to be bathed in a uniform environment, namely the same perfusate that was used to vitrify the kidney has to surround it top, bottom, and all the way around the sides. And that means that the volume of the system that we want to warm back up is much larger than the kidney itself, about 3 to 5 times bigger than the kidney itself. And the problem that that creates is that cooling is slower for bigger volumes. So on the left, you see the bare kidney, and the cartoon shows the kidney sitting on this perforated platform while the scroll fan blasts it with liquid nitrogen vapor to cool it very rapidly. Whereas on the right, you see a partially immersed kidney embedded in the liquid solution, and it still gets blasted with the liquid nitrogen vapor, but of course, heat exchange is going to be a lot more slow. And during that time, there's more opportunity for ice to form. So this shows the numbers and introduces another point, but just as far as the cooling aspect is concerned, you see that the bare kidneys, both the core and the center, cool a lot faster than the immersed kidneys. In fact, it cools about twice the speed of the immersed kidneys. But then what happens to the immersed kidneys is that they have to be prepared to receive the electromagnetic energy. So they have to be warmed up to a temperature that's warm enough to partially mobilize the water molecules in the system so that they can then be free to respond to the field when it's applied. And you can see that when the field is applied, which is indicated by the RF warming label over there, the transition through the zone of ice growth around -60 is much more rapid than it would be with just the naked kidney being warmed by conduction, as you see on, with the blue curves coming back up. But the kidney has now been exposed to more nucleation during cooling and during rewarming. And the question is, what will that do to the ice formation tendency of the kidney? And this slide is a little bit complicated, but I'll try to walk you through it. So the answer in general, before we started these experiments, is that we realized that based on typical bare kidney cooling rates, we can determine how stable the kidney is by measuring the urine concentration of the perfusate as a non-invasive invasive measure of tissue concentrations of cryoprotective agent. So our conventional understanding of the situation has been that if we can get a kidney equilibrated to the point where its urine has about 92.5% of the concentration of M22, which is our perfusate, then as long as we warm it at 80 degrees a minute or above, we're in the no ice damage zone. But if we are a little bit shy of that warming rate, then the kidney will be damaged by significant amounts of ice, about 8% ice versus less than 1% ice as you cross this safety line. Furthermore, if you load the kidney to 94+% of the perfusate concentration, then you only have to warm it at 40 degrees a minute. So by all rights, if we were to load it a little bit over 96%, we might be able to warm it up almost by conduction at 12 degrees a minute. But we now discover through trial and error, and I'll show you some of the data later, that the nucleation induced by the slower cooling and the pre-warming causes ice to grow so much more rapidly that even though we are warming at 90 degrees a minute with RF warming on average, indicated by the hexagon, that's actually what we have to get to 96.7% equilibration to prevent ice damage, even at that 90 degree a minute warming rate. So in other words, the safety curve with the points on it has now been lifted up to something like the higher curve. And you can still achieve ice damage if you can warm faster than what we're looking at in this specific example, but the bar has been raised. So in a way, we're chasing our tail. In order to warm rapidly, we have to make it harder to warm without ice. So we're getting to the point where we may have an option. We either use RF or we just load the kidney up much more thoroughly and warm it by conduction. So we may be comparing those two approaches pretty soon. Now that's an insane level of equilibration of the kidney. It's been hard enough to load it up to 92.5% or 94.11%. So if we want to go to 96.7%, is it even possible? We think so. So what you see on the left, the black line is our standard protocol. And as you can see with our standard protocol, we get over 94% cryoprotectant in the urine by the time we hit 51 minutes of perfusion time, which is the time which we would normally want to vitrify the kidney. But now we have this new standard, the gray bar, which means now we have to get all the way up to almost 97%. And our standard protocol doesn't do it. But with the new protocol and a new solution indicated by the red points, we can get there only 2.5 minutes after the standard perfusion time, which is probably okay. And if we use that kind of perfusion approach, the kidneys still survive about as well as they do under baseline conditions. So the MPE and MPS experiments in particular, although it's imperfect and we still have some more work to do. But one good bit of information is that this is the appearance that we now get when we warm up a vitrified kidney using RF power and transplant it. The kidney looks lovely. There's no ice damage on the surface like we used to see. It produces clear urine. The ureter is not ruptured. There's no hemorrhage in the ureter. There's a little surface blood, but the ureter itself is in good shape, and the kidney will produce blood-free urine for 30 minutes. So we think that we're getting very close. The other way that we judge the quality of the preservation is by using this ice damage test. As you see on the left, kidneys that have ice formation during warming have damage to the vascular system, and when you try to wash the blood out of the kidney, it doesn't come out of the medulla. It's trapped in the tissue because it's leaked out of the blood vessels. We see kidneys more like the kidney that you see on the right these days, in which there's essentially no blood trapping in the kidney. So we can maybe do better than this. We have a lot of maneuvers we're working on right now, but one of the interesting things that may be possible is to intrinsically reduce the toxicity of cryoprotectants. These are some interesting experiments that were done by a collaborator of ours. They exposed whole C. elegans worms to M22 at room temperature for 5 minutes, and they can actually survive 50% M22 without any special pretreatment, but give them 75% M22 and they're all dead. But there's a treatment you can apply that allows them to survive that 75% M22. And they've actually applied a couple of these treatments to rat or mouse kidney slices. And as you can see, the M22 treatment by itself results in a lot of red cells, which are dead by vital staining assays, and that's greatly reduced close to control levels by their interventions. Hopefully, we're going to be able to apply those approaches 21st century medicine next year and see if it works for us too. Okay, so let's talk about brains. I believe that Robert McIntyre was just talking about brain shrinkage, and he's right about that. There is a lot of brain shrinkage if you perfuse with M22, which makes structural detail hard to see. And the question is the detail still there? At least the essential detail is still there. So you see a cold hypothermic control brain on the left and an M22 perfused brain on the right, and the asterisks are nuclei, cell nuclei in both cases. And you can certainly make out the nuclear structure normally on the left, but what you see on the right bears no resemblance to anything that anybody's ever seen in real life before. And you can see some myelin unraveling and things like that. But the question is the essential part of the brain preserved? Is the connectome preserved, for example? So one way to find out about the degree of preservation of the brain is to reverse the shrinkage so you can see the brain better. And what the diagram shows is that as you increase concentration, the volume of the brain decreases pretty rapidly at first, and then it continues to decrease, but undetectably as you increase concentration. And then as you decrease concentration, at some point, the brain is going to begin to come back to its normal volume. And you should be able to therefore see less contracted and therefore more visible brain tissue at one of those points along the way. If you go too far, then the CPA leaking into the brain will cause it to expand to greater than its normal volume. which will also create all kinds of other problems, and that will give you bad results. So the point will be to pick a sweet spot in here to reveal the preservation of the brain without getting into an artifact caused by brain swelling. And if you do that, this is first result we got. Load up to M22 level, which is 9.44 molar, and then wash out to 5 molar and fix at that point. And as you can see, it appears that the brain is well preserved, and you can see synapses everywhere. There are some shrinkage spaces still, but and there's some things that look a little weird, but nothing that looks like it's affecting the connectome. So it looks very good at that level. But here's what happens if you go a little bit too far. Here you're washing down to 1 molar M22. You begin to see things that you don't necessarily like, some white spaces that are a little bit indicative that maybe you've got some intracellular edema or some such. You see a blown mitochondrion at one point, but you now see the synapses even more clearly since they're less contracted, and I've circled several of them for you. And you can see that all features of the synapse look normal, including presynaptic vesicles. You can see the latter a little bit better at higher mag. So this is just the same treatment, but higher magnification. And I've just put in some blue arrows showing some of the presynaptic vesicles that you can see in the synapse, and you see normal postsynaptic densities all over the place. So the synapses are circled in yellow as before. Now, if you wash all of the M22 out, you can also preserve the structure as long as you use an osmolyte. We like to use decaglycerol. Most of the time we've used too much and the brain looks pretty well contracted, but we're finding exactly what the right concentration is to use. I'm still putting the finishing touches on that experiment, but we did this experiment some time ago in which we used scanning electron microscopy to get an idea how intact the brain was. And based on scanning, you don't really see missing material. You see what look like intact neural processes. The other thing about brain preservation is that you're not necessarily stuck with the shrinkage. As Robert and I published in Cryobiology, it's possible to use SDS to open up the blood-brain barrier. When we did that and repeated the experiment of going up to M22 and then back to 5 molar M22, you see just beautifully preserved structure everywhere you look. And I don't think there's any doubt that the structure is well preserved in this case. So a bigger question is, what about human brains? We do have results from an actual human case. This is a histological section. We use double staining here to bring out all of the molecular background detail in the brain. The cell bodies are a bit shrunken. You see shrunken nuclei, but the white spaces are blood vessels. They're all normal. This is 20x. We looked at 40x. We looked at 10x. We looked at section after section after section. This is an actual Alcor case. And we saw no ice damage anywhere we could find it, at least on the light microscopic level. Of course, damage can hide on that level, so we used electron microscopy to look for ice damage, and there is no ice damage. Here you see the same dense, contracted kind of structure that we're used to seeing in animal brains, but with a well-preserved blood vessel and actual distorted larger fibers that are less distorted than what you see in animals. So it seems like the human brain actually does better than animals. This individual had some kind of strange brain disease, so there's some dark deposits in his brain. But other than that, things look, in my opinion, pretty good. But you can't see fine detail. And we didn't have a re-expansion technique to use in his case, although we're working on that. So in lieu of having such an expansion technique, we just looked at the samples under higher power electron microscopy. And you can see synapses very clearly defined, very distinctly present, and the neural processes seem to be pretty well preserved. We saw no evidence of ice formation on either cooling on the left or warming on the right. I won't spend a lot of time on the figure, but we don't see any ice melts in either one of these cases. And we hope to publish these and remaining results in much more detail sometime next year. So in summary, it seems that complex systems in general can be cryopreserved. That's result not only from my lab, but from the scientific literature. There are endless examples of pretty complicated systems being cryopreserved successfully. The question is, can you scale that up? And at least at the level of whole kidneys, we know they can survive after cryopreservation, and there's good prospects for doing better about reducing ice damage and improving the results there. It seems so far that the brain is not necessarily an exception. Ultrastructure seems like it can be preserved in animals, particularly if you do blood-brain barrier opening, but even if you don't. And we're beginning to get reasonable evidence in humans as well. So I want to thank all the people at 21CM who helped us get this data and our past collaborators whose work is represented in the talk, as well as the people that pay the bills at our company. So thanks very much for your attention. And if there are any questions, I'll try to answer them. But I cannot
Here we
hear
go,
anything.
Greg. Two quick questions,
all right.
3 by now already.
Okay.
What about rewarming with nanoparticles and magnetic heating? Wouldn't this give good uniform warming rates?
We think so. Now warming has some major advantages. It has some disadvantages. We think the ideal combination may be to use both RF warming and nanoparticle warming. We have prepared kidneys with nanoparticles and sent them to the University of Minnesota, and they've warmed them up successfully there. We're going to be repeating those experiments, and then it may be that University of Minnesota will actually send a technician and their apparatus to our laboratory, so we can not only warm them up, but transplant the kidneys, and that'll be very exciting. But we hope to be able to do that maybe starting before the end of the year, but more likely we'll do the beginning experiments before the end of the year and do the transplants sometime early next year.
Exciting, awesome. Another question would be, what is X1 and X2?
Yeah, those are interventions that were promulgated by my collaborator, and he hasn't authorized me to release it because he hasn't published his results yet. So I cannot share that with you, but I wanted you to know that something good may be coming down the pike. And I will work with him to get his results and our results published. So then we can tell you.
Awesome. Greg, due to time, we'll move on to the next speaker, but there's a few more questions for the Q&A and panel session. So just the same call would be the exact one. All right.
Okay.
Our last speaker in the main block before Q&A is Max Moore. He has a bachelor's from Oxford and received his PhD from the University of Southern California. And of course, everybody knows him from serving for more than 9 years as the CEO of Alcor, where he's still active as an ambassador. Max, take it from here.
Thank you. It's great to be here. And hello, good morning, good afternoon, and good evening, wherever you are. I want to talk a little bit about how cryonics organizations can survive over the very long term. Anybody who's discussed cryonics can predict that certain questions will come up or certain claims will come up. We all know what those are, but one of them is, well, cryonics organizations, even if it worked, in principle, are not going to last that long. They're going to fail and it's game over. I'd like to point out, of course, that there are already 2 organizations been around for close to half a century, and people who don't know that go, oh, okay, that's not too bad, I guess. But we will go into some of the other organizations that didn't survive that long. So it's going to be at least several decades, and some people would argue more than a century, before we can repair, revive, and rehabilitate people. So how can we maximize the chances that we're actually going to survive that long? There aren't very many organizations that have been around for a century plus. Some of you might have read my article in the previous issue of Cryonics magazine. I'm just going to touch on a couple of those points briefly, and I'll be doing a part 2 for the next one. But before I go into some of the details, a paper came out just a couple of days ago in Nature, I believe it was, which I think provides a rather nice metaphor. Some of you may have heard of this bug called Nosoderma diabolicum, or the diabolical ironclad beetle, which is a pretty great name to have, I think. It's a flightless, inch-long beetle that thrives actually in this area where I live, whereas most beetles live only for a few weeks. A diabolical ironclad, I just keep saying that because it just sounds good, can live for 7 or 8 years. That's basically like living for 1,000 years for a human being. And one way they do that is because they have this incredibly strong peck, crunch, and squash-proof shell, or exoskeleton. It has all kinds of internal layers, tight joints, plates that can shift in just the kind of right way. So it's both tough and extremely flexible under pressure. These things are just insanely strong. These are like Superman or Thor in terms of their invulnerability. They can actually, these lovely researchers drove over one back and forth with a truck. It's actually 39,000 times the body weight's beetle, and it took it okay. So if you scale that up to my size, that would be like 3,800 tons on my back, or 38.3 whales, if you prefer that way of measuring things. So it's kind of interesting how this thing is built. And they've been knowing this thing for a while, but they didn't know how it worked. Turns out that the top and bottom of the exoskeleton can shift in relation to each other. If one part gets squished, then vital organs can, internal goo can smush back towards the back of the beetle. And you're wondering, what does this got to do with cryonics organizations? Well, we'll see, because there's an important number of points to make about dividing organizations into various compartments, protecting them, and allowing resources to move between them. So it's actually kind of a nice analogy. It also has a really powerful back, and I won't go into how that works exactly, but it distributes the weight. It's very flat for it, first of all, not usually the usual beetle shape. So it can distribute that pressure around its shape. So it's going to be actually pretty interesting. We can probably build exoskeletons and armor based on that. But for cryonics organizations, I think it's kind of interesting to think about how can we separate different aspects of organizations and responsibilities so that if there's a high pressure attack on one part of the organization, another part can still survive and still give us a chance. So just very briefly, I want to summarize a couple of points from the first article I did on this leading up to this talk. Non-profits versus for-profits. And I'd be very interested, by the way, from you guys in Europe who may be having a different organizational model, because this is going to be pretty sort of non-profit oriented. And I'm very interested in how a private company might do well in the long term. I think it's a bigger challenge than non-profits, because if you look at the very long-lived organizations, you tend to find they are non-profits. They're often educational organizations, religious organizations, and so on. But there are ways that for-profit companies can allow the insiders to maintain control, even while going to the capital markets. Now, of course, Japan stands out as the land of the super long-lived organization. Last year, there were over 33,000 businesses in Japan over a century old, which is pretty amazing. We'll go through a bunch of those numbers that are in my magazine article if you're interested. There's a certain kind of organization that's over 1,000 years old, and there aren't very many of them. There are 14 of them in the world that we know of. 8 of those are Japanese. And those even have their own name, which I'm not quite sure how you pronounce it, because nobody seems to agree on the internet, but it's something like Shinise or Shinisi. These tend to be family-owned organizations that are very tightly run, and a family basically retains ownership within the same family through the centuries. And in fact, even in the European versions of this, or the youngsters are only 500 or 600 years old, it's a very similar situation. But what's kind of interesting is, and it's very difficult for a cryonics organization to be founded by a family. So you think, well, how could that be applied? Well, they kind of cheat. It's kind of interesting fact that in Japan, 90% of all adopted people are adopted as adults, which sounds kind of odd. You think, well, adults aren't usually adopted, but they're adopted for corporate reasons. Basically, they're adopted into families so that they can actually run corporations. So it's a little bit of a cheat, but it's kind of an interesting idea. So can we enjoy those kind of benefits? Well, that's one way of doing it. I guess there's some kind of equivalent of adult adoption. And I think the way you have to do that is by putting in place certain requirements for people who run cryonics organizations. And I'm going to get to a couple of those points. So I think we have to have a little bit of encouragement that there are universities that have survived for several hundred years. In my old land of England, they actually have a thing called the Tercentenarians Club, which is a UK trade association that only accepts members over 300 years old. So it's kind of a little bit snooty. And they have to be owned by the same family that started them. So if you think about what it's like to have an organization that actually can enter the Tercentenarian Club, they basically survived at least 47 recessions, a whole bunch of banking crises, stock market crashes, the start of the Industrial Revolution, the end of horsepower, 2 world wars, the defeat of Napoleon, and the rise of the internet. So that's pretty impressive. Okay, so what practically can we do? Well, I'm going to, again, be pretty alcohol-centric here. And I very much like to get input from people from Europe and other organizations that are starting up with maybe a different model or not entirely the same. So one part obviously is documentation. One real concern that we've had, because we're still a small organization, we're probably larger than other cryonics organizations, but we're still small. And when an important person leaves the organization, you lose a tremendous amount of information and experience. So we've been very keen on documentation. We have what we call the red books, which are collections of all the data on each of our patients. Those are stored both on paper in fireproof safes, they're stored in a server, they're also stored and encrypted in the cloud, so we shouldn't lose those. We've also been working over the last few years on SOPs, trying to make sure that everything we do is documented, everything from liquid nitrogen fill to how we do surgery and so on. We also have a bunch of provider manuals now, which we've been working on with, and you've seen ICE and SA also. So we work on provider manuals so that we have standard ways of doing things and they're documented. Also very important, so obviously I could go on about that quite a bit, but in this limited time, we talk about governance. This is particularly important, and this is why I'm very curious to know how a for-profit private company would work. Alcor has a self-electing board or a self-perpetuating board. Now, I noticed online some people like to criticize Alcor for that and say something bad, that we're self-perpetuating and self-electing. But the fact is, the vast majority of nonprofit organizations have that structure, and there's a good reason for that. If you have actually a member-elected board, it's not very hard, especially if you're fairly small, to have outsiders come in and take over, and that certainly does happen in certain kinds of organizations. I know that CI has other ways of trying to prevent that. You have to be actually not just a member, but a cryo preservation member of CI, and you have to be a lifetime member or at least a 3-year member before you can vote. So they have some kind of protection in there. So our board of directors comes from itself. It comes from the advisory board. We usually train people up on the advisory board first, so we know what kind of person we're getting, hopefully. And you won't get someone being pushed in by someone who actually wants to destroy the organization or damage it. So that's been going for 48 years, and it's worked pretty well at providing continuity of purpose. Again, all directors have to be members, they have to be cryopreservation members, and they really have a lot of experience. We also have kind of an increasingly robust committee structure. There's different levels of committee. We have the deployment committee, which makes decisions on when and how we're going to deploy on a case, which is quite difficult. For those of you who are kind of in the organizing stages, that's something you'll find is quite a challenge, because every time you deploy, you spend a lot of money and resources. And it's, there's also the possibility that if you deploy on one case, which doesn't happen, it could be at the expense of another case that does happen. So it's not uncommon that we've had 2 cases in the same day or the same couple of days or the same week. That's a real challenge. I think Eric Vogt touched on that, the difficulties of actually deploying. So we have the deployment committee, we have the research and development committee, which has just been elevated to the level of a full committee now. Legal and regulatory, that's a very important area. Let's say something else about that in a moment. The finance committee and various working groups. If you go to the Alcor blog, you'll see just yesterday I put up something about a new working blog for revival, rehabilitation. Something we haven't really spent a lot of time thinking about, but I think we should, because one of the big objections people have to cryonics is not that it won't work necessarily, it's that they're afraid it will, and they're going to come back and be all alone and poor, and who's going to take care of them? And so it's actually part of Alcor's mission that we will take care of you. And so now we have a working group that's actually going to be thinking about that and coming up with scenarios and look at, well, read the blog post, I won't say anything more about that. We have other things like conflict of interest policy, advance notice policy, which basically is a way of preventing a few directors railroading everybody else. Basically, it's kind of a veto power, which makes people have to wait 25 days before any new policy is passed. We're also very keen on openness. We do tours twice a week, or at least we did until coronavirus got around. We're going to be filming a virtual tour, making that available and answering questions. So we pretty much show everything. People can photograph pretty much everything. And of course, we publish case reports, something which was on hold for a little while, where we argued about exactly how to deal with privacy concerns. But those are now being published again. And I have to shout out to Linda Chamberlain, because she's done an amazing job in the last few years of pumping out dozens of them. So people who say, well, Alcor hasn't been publishing, well, just wait and see. You're going to get overloaded with case reports very soon. But while being open, we also have become increasingly keen on protecting privacy, and that's one of the things that delayed the case reports. And we have some members who want special levels of privacy. You probably already know that you can choose to be a public or a private member. Some people want even higher levels of protection than that, which can be quite challenging. Now, on the area of financial management, obviously this is pretty critical, because any organization that's going to last over long periods of time has to not lose money constantly. It has to stay ahead. So although Alcor is a non-profit organization, that doesn't mean you can make a loss. You can't do that over the long run. So we manage things very conservatively, and going back to the ironclad beetle and segmenting the organization, one thing Alcor has done, starting in 1977, created an irrevocable patient care trust fund. So this is like a separate segment of the organization in a sense. It's even legally somewhat separate. And just a couple of years ago, we got IRS approval to take that separation up to a higher legal level to make it what's called a type 2 supporting organization. So essentially, if the government was to crush Alcor Central, put us out of business, that should not affect, that's what the lawyers say anyway, it should not affect the patient care trust fund. That should continue to exist with all this, whatever it is, $16, $18 million in there, and maintain the patients. So that gives some protection to the people who most need protecting. And not only does that exist, it's managed in a very specific way, and there's a very strict rule about how that's managed. We can never draw more than 2% a year from it, for instance, and actually we haven't drawn that because we don't just want to maintain the money, we need to build that money up over time, because at some point, if it becomes possible, we're going to need to repair and revive our patients, and that's going to cost some money. Now, some individual patients may have a trust fund, and we've been helping people with that, asset preservation trusts, future income trusts, and so on. But we need to be able to guarantee that we're not just going to barely keep you preserved, but we'll actually be able to pay for the research to bring you back in the future. So we place, for each neuro case, we place $25,000 out of that $80,000 minimum into the patient care trust fund, and for each whole body, it's currently $115,000. So people say, well, why are Alcor's fees higher than some other organizations? Well, it's because we put a big chunk into that trust fund that's going to last indefinitely. And so far it has been doing very well. You can look at the finances on that. We also try to plan for inflation, and we haven't always done a great job on that in the past, because people don't like to think about inflation. And we realize that we're going to have to really push people a lot more to do that. And I can't get into the details of that. I know Rudy would tell you to buy 100 times as much life insurance, or at least 5 times, right, Rudy? That's one way of doing it. There may be other mechanisms by which we can help people keep up with inflation, because it is super low right now and has been for quite a while, historically unprecedented, but we don't know that that's going to continue. We also try to help out members who are struggling. We have a hardship fund. We also have a reserve account that we've had to dig into recently because of legal challenges. So that's a very important thing to have for an organization. Don't be running on the edge with your operating funds. Keep some money in a reserve account that people like me and Patrick can't just go out and spend. They actually have to get permission from the board to do that. Another thing to consider if you're starting an organization at some point is an endowment fund. Now, obviously, endowments are very common when it comes to large universities. We have a few $1 million in the endowment fund. Large part of it came from one donation, actually, one bequest. And what we do with that is, whereas most universities draw maybe 5 or 6% of assets annually, we think that's way too risky. Again, we have the 2% rule, because you can actually show statistically that if you keep to 2%, you have a pretty good chance of keeping it going over many decades, even with the stock market performing very poorly. Going to 3% actually massively increases the chance you run out of money. Now, hopefully you have an inflow of money as well. That's what the universities bet on, and so far they've been okay. But we want to be super conservative with that. So it's a little bit more complex than 2%, but that's essentially the way it works. So keep building reserves, keep building capital, never get too comfortable, don't try to live on a shoestring. It's very important also to have outside organizations review your accounts. we've actually been doing reviews, independent reviews for quite a few years. This year, we're actually going full-blown audit, which is a pain in the butt, frankly, because it costs a lot of money to do a full audit. But since we're a California corporation, that becomes required at a certain point. It just takes a lot longer and it's a lot more invasive, but it's a pretty good guarantee that, well, it's not a complete guarantee, but it helps to assure people that there's nothing going wrong. We also use proper accounting methods, GAAP accounting. I think in Europe it's called something a little bit different, but it's an equivalent. And other checks and balances. For instance, the finance director has no signing authority on any accounts. So the person who's kind of looking over the money cannot write checks or use credit cards. So we have various kinds of checks and balances like that. Then there are a whole bunch of security measures. I'm going to race through this stuff because I know I'm running short on time and everybody's been very disciplined so far. Congratulations. We have security measures, which I won't tell you about because I'm not as secure as if I do. We have a legislative consultant, and this is something that if you haven't thought of doing this yet, and I wasn't talking about finding out the laws in your particular country, I think that was John Aaron's or Eric's talk. We have a fellow, Barry Aarons, who's very, very good at keeping track of legislation and has in fact saved us in the past from a couple of problems, one of which was potential regulation by the cemetery board, which would be quite inappropriate. We managed to overcome that. And then because there was a scandal in the Arizona area with companies that collect body parts and sell them for research, there was legislation written that would have caused us problems. But Barry contacted the people who wrote it and explained what we do. And they said, oh, we're sorry, we'll just rewrite that. And they did. And so everything was fine. So that's kind of an important thing to keep track of because that can cause you devastating problems. And it's not a technical issue. I know a lot of cryonicists rather deal with the technical stuff. So another thing that's very important to do is periodic evaluations or reevaluations of your procedures. If you're starting a new organization up over there in Europe, you have a great chance to start fresh, to look at all the protocols and handbooks that other organizations have. And I saw on the chat, someone pointed out Ashwin and largely Ashley written protocol that we have, which is very detailed and in many chapters, and it's quite available. You can take a look at that and adapt it as you see fit. So we do that. We have after action reviews after every case. We will bring in our contractors, maybe ICE, maybe SA, our own people, and we debrief. We say what went right, what went wrong, and what can we do better next time. And that is feeding into some work that Ashwin de Wolf is doing currently. It's a multi-year project of meta-analysis. So that uses the after action reviews, it uses CT scan data, it uses all the measurements we take during a case to see how a case went as compared to the circumstances. You heard about different kinds of ischemic delay and cold and warm ischemia, various things that might have killed you in today's sense, whether an aneurysm or a heart attack. Which things make the biggest difference? And that is helping actually, I think, to drive the case for field cryoprotection, as Ashwin also, and I was on board with that. So that's pretty important work that's going on. And the CT scans and the other readings we take are part of what I like to call evidence-based cryonics. A lot of people think this is a scam, and they come and see the place and they get a bit reassured. But the more we can actually show what's going on inside the head, and the work that Greg is doing, obviously, and other researchers, is critically important. And we have some other interesting research coming up along those lines, go beyond CT scans that I can't talk about yet because Greg will crucify me if I spoil the show before he's published on that. But we already have a lot of interesting work coming out there to show that, yes, there is good evidence that this can actually work. Robert also touched on some of this. And finally, I was not going to mention this, but then I think Emil may mention this, scalability. Once you start growing at a certain pace, you can fall apart very easily. We've seen many organizations that, especially internet companies, that are growing very rapidly, and you think, well, they're a big success, and then they just crumble because they're not really built to scale very well. And frankly, we haven't been built to scale very well in the past. And I admire what you guys are doing over in Europe with the new sign-up process, which greatly reduces the labor. And we're actually doing something like that too. We're actually a little behind you guys, but we're going to have a full web-based system where you can sign up, where we help you with insurance, automatically populate the forms from the short application, and make it one heck of a lot easier than it has been in the past. So that means that we don't have to overburden our membership administrator. We don't put people off with a real or perceived burden of signing up. So those are some of the building blocks, and not all of them. And again, I very much welcome input from a different perspective, but those are some of the building blocks that cryonics organizations can use to combine the strengths of the diabolical ironclad beetle, the Shinizi, and the tercentenarians and the family firms. Thank you.
Great, Max. Thanks a lot. Before we start with questions for you, we would already ask everybody, all the other speakers already start joining the Zoom call again, because we directly start with the Q&A and panel after that. But until everybody's here, Max, there are a few questions for you. So first one being, does Alcor put a part of the funds in cryptocurrencies?
No. We have received cryptocurrencies in large amounts, actually, going back to something like 2012 was the first one. And then we received a $5 million donation that was in cryptocurrency. However, our policy currently is because of the instability of those, the volatility, that we're required to sell those right away. Now, I may or may not completely agree with that, but I would certainly not want to keep 5% in there. But no, right now we don't.
Right. And just a quick note, all the other speakers who already might be in the call, we would ask you to be muted for the time being, because otherwise we already hear you. Second question would be, do you plan to add any new revenue streams to make the organization more financially stable in the long term? For example, give an example here is long-term digital data preservation for people in cryopreservation, for example.
That's kind of a tricky question. Your particular example, I think, is a fairly easy one. Yes, I'm not sure how much of a revenue generator that will be, but it's certainly something we want to offer. And we probably would have to charge something for that, depending on how good a job we did. But in general, we're pretty careful about mission creep. Again, that's one of those things where if you look at the history of long-lived organizations, they're pretty tightly focused. They have to do a shift sometimes. That 1,400-year-old company during one of the major wars shifted from building temples to making caskets for all the people killed in the war. So they had to repurpose a little bit, but generally you're going to stay pretty tightly on focus. So probably not going to be doing stem cell preservation or egg storage or things that other companies already do very well. Absolutely.
And last but not least, a question for you. I'm paraphrasing slightly. Are you worried that cryopreservation becomes too successful, so that once millions of people are cryopreserved, that then, and regardless of the organization's strength, that some government might say, well, you know, some over-indebted government might say, let's plunder the trust fund. Any thoughts on that?
I don't think that's probably a big problem. I think it's actually a good thing. I think we'll have an awful lot of political influence once we're that size, frankly. There's a book I'm trying to remember the title of, female science fiction writer who has a future like that, where cryonics organizations are so huge and wealthy that they pretty much control everything. So I think if anything, it might be the opposite issue. But again, that's why we have a legislative consultant and keep an eye on that kind of thing.
Makes sense. All right, now I would say we switch over to the panel and Q&A. Maybe we can switch the screen as well. But I hope Aaron is already able to hear me, because, okay, perfect, that already looks good. So first question for you, because we didn't do a Q&A before. How many patients are currently, or how many patients are there already currently at Yingfeng? And do you see any significant growth in there? So I guess the question is about in China.
Need to unmute Aaron.
Yeah, Aaron, if you're still muted, you need to unmute.
There we go. I'm unmuted now, I
perfect,
think.
now it works.
I was pressing it on my microphone, but not on the keyboard. So thanks. So yes, in China, Yingfeng currently has 10 patients who have been cryopreserved, and I believe they have just slightly over 60 members. This being a coronavirus year, obviously it's been fairly dampened, but they've had a lot of press and media this year, especially given the recent movie that came out, Hope Frozen, about the little Thai girl who was cryopreserved when I was working at Alcor back in 2015. And that has stirred a lot of interest and generated a lot of people who are calling on that. Whether that'll actually convert to members and or patients, that's difficult to say. But so far, there seems to be a pretty high level of interest.
Cool. And do you also offer or consider to offer neuro patients?
Currently, China is not interested in doing neuro. They really want to do whole body. Part of it may just because of the negative press that neuros occasionally receive throughout the world. But they are they're really focusing on cryopreservation of all aspects of the body, whether it be the whole body or whether it be organs or tissue or the stem cell, cord blood. So that's really been their area that they're focusing on. So whether they would actually get to brain only, so far they've said no.
Okay, interesting. And last but not least, Max just mentioned data protection and so on. China traditionally might have some questions to answer, or got some questions. Do you see any problems with data protection for patients or ethical concerns in China? What's the opinion there regarding cryopreservation?
So they, because this is a government sponsored program in China, they were able to get government, not only government approval, but a certificate for this. There are certain expectations that they have to maintain anonymity and protection of information. I believe of the 10 patients that they have cryopreserved, they've only released the names of 2. And that was specifically because the family had requested that. So it seems like they've been very good about protecting the confidentiality and maintaining that, if that's what you're referring to in terms of data.
Yeah, I guess. Okay, thanks, Aaron. We'll go on. We'll go speaker by speaker initially, and then see how much more questions we get in the end. And then when there are no more questions or anybody needs to drop out, please do. Next, there's a question for Ashwin. In the sense of field cryopreservation, do you see it to be possible to become more affordable? Or do you think it's an extra added cost? Ashwin, you also would need to unmute yourself. I don't know if we can do that, otherwise...
All right, okay, here I am.
Now you go.
So in theory, it should actually be less expensive, because if you actually look, like, for example, at the cost schedule of Alcor, you see like a portion for the standby and stabilization aspects of a cryopreservation, and then you see cryoprotection, which is at the Alcor OR. In the field cryoprotection, that's rolled into one. Now, of course, you cannot just say like, oh, it will be 50%, but I think it's pretty realistic if you do it right to end up somewhere between like the 50, and 100% of that total sum. So yeah, cost savings could come out of that for sure.
Perfect. I think, Ashwin,
Now,
all the other questions we answered before.
I want to make one caveat to that. I mean,
go ahead.
If, let's say, you subcontract the field cryoprotection part to another entity, then of course you are at the mercy of what that entity will charge for it. So a lot also depends on how much control you have over the cost that you incur when you do field cryoprotection.
Right. Awesome. Next would be Chris. Chris, so one of the questions was, have you considered comparing cryopreservation with cremation half a century ago? Because this was, of course, also a topic that got significantly more mainstream appeal over the last, half century or so.
Yeah, that's interesting. I'm not familiar with the history of other burial techniques, but I do know that there's been a large shift towards cremation away from burial. One thing that we found was that interest in burial was closer to like 8%, and cremation and cryopreservation was not far below that.
Right. Yeah. Maybe as a follow-up, we can do that at some point.
Yeah, sure.
Then another question is, do we believe, or the question is, I imagine those people who had interest in answering the questionnaire are already much more likely to be positive towards cryopreservation. Do you think that adds a bias to the data?
Oh yeah, it definitely does. Participants were told that they were going to fill out a survey about brain freezing, and we chose that phrase because we felt it would interest people, and we felt it was something that people would recognize more than they would recognize more technical terms for it. We also provided an explanation of the practice, and you can find the text of that and some stuff that we've put out. Like I said, with any survey, we know that surveys are very finicky. It depends on how you phrase questions, the order in which you present them. Every little thing will make a difference. So the goal always is to be very thoughtful about all the decisions we make and make sure we're understanding and being honest with ourselves about the kind of bias that we're creating. But in the end, I think that we did a pretty good job of providing something that's valuable to this group.
Right. Absolutely great. Going further with David Gifford. David, we'll wait until you're full screen and unmuted.
Hi there, I'm there. Yeah, hi.
Right. Has the idea of perfusing magnetic microparticles for later rewarming already been picked up by any provider? Is this something that you have considered?
I think it's of theoretical interest, and it's something that I became fascinated by when it was evident that potentially it could circumvent the need for extensive nanotechnology for rewarming. Now, Greg has actually sort of mentioned this kind of technique and how you can use those kind of technologies in order to get the rapid rewarming that you want in order to minimize isonucleation. And so it does look at potentially as though it does have utility. Frankly, I don't really mind which method that the science finds is most efficient for us. I'm kind of a practical guy, and it's really what's going to work for the industry that is more important.
Right. Another question is that using ECMO, of course, is a highly interesting proposal. What's your opinion? How many minutes or hours or any time frame past circulatory arrest would ECMO need to be started for the procedure to make sense?
It makes as soon as possible. Essentially, in the clinical setting, i.e., with donor patients with DCD donors, we've got it down to a fine art, and we can get a patient that's formally declared dead to on ECMO in about 16 minutes. Now, I'm part of a team that's very good at what they do, and my surgeons are very good. And we spent time figuring out how we were going to make that happen and just get the moves right. So, and because we believe that every minute counts. Now, the idea that it's okay to leave a patient post cardiac arrest for a couple of hours and everything's going to be hunky-dory when you start resuscitation doesn't really feel valid when I've looked at patients who have has suffered the same fate. So it's something that there's minimum time. I don't have a time. Well, this time 5 minutes is okay and 55 minutes is okay as well, because I don't really believe that. It's that what you're looking at is trying to minimize that damage, and so therefore minimize the work you've got to do to get back to where you were, if indeed you can get back to it.
Right. Of course, unfortunately, in cryopreservation, the question is mostly about what's the maximum time where it still makes sense, right? Can you speculate on that, or do you feel like this just needs more research and it's very difficult to speculate on?
I think what we can say is that if at warm, and patients cool quite rapidly anyway, so it's naturally. So you probably can get more than 8 minutes. Topical cooling, and that's why I suggested it as a precursor, because if you can get in and you can start to do the basic resuscitation. So if you can actually affect cooling, which is going to slow metabolic rate, in 7 degrees will give you a 50% advantage. So you drop cooling, you drop the oxygen consumption by 50% if you can get 7 degrees. And so therefore your external cardiac massage, which you should be continuing, is good for about 55%. So between the two processes, you probably can maintain an equilibrium until you can either get onto the bypass, the ECMO that Aaron described and that I attempted to describe as well. So, I mean, I think I know that if somebody signed up for cryonics and cryonic suspension, and that the team is unable to get there for the first hour, then they've been unable to get there for the first hour. So do you say, right, it's too late? No, you don't. You go with it, and you attempt to recover as best you can, and we wait to see what fate awaits them ultimately.
Right. Thanks a lot. All right. Another question for Roman Bauer. What is holding back computational approaches for finding better cryoprotectants?
Yeah, I think that's an important question, because I think there's a lot of potential there. I mean, there are some molecular dynamics models that try to model in cryoprotective agents and their impact, and I think there's a lot that one can do there. There are some preliminary studies, but there's, I think, still a lot that we need to do. And I think the main problem is as generally in this field, I think, is funding and data. I mean, the more data we have, the more we can create computational models, the more detailed computational models we can create. So I think it's just a matter of research that can be invested.
Perfect. Thanks a lot. Going on with Greg Fay. Greg, what do you think is what would be needed to speed up research? What are the bottlenecks? Is it human capital? Is it researchers? Is it money? What's the biggest bottleneck?
I'd say the primary obstacle is lack of funding. We do have a stable source of income, and that's critical for all the progress we've been able to make, but we have a limited number of people. And if we had more people, we could do more. And human capital is an issue. There's a shortage of people with the right skills available to do this work, but to a large extent that can be overcome by training. So the main issue is having sufficient resources available.
Right. Another question would be, do you believe that the current cryopreservation protocols without fixation preserve the brain's anatomy just as well as with fixation?
Oh, definitely not. You know, the anatomical preservation that you get with Robert's technique is far superior to what we get. But the question is, does it matter? I mean, you don't have to achieve perfection in order to achieve success. And I think that's the point. And it's also a question of what do you give up for that lovely anatomy that, you know, you could publish in a textbook, but you can't do any biochemistry on that because it's quote unquote dead. You know, all of the molecular activities are arrested, the enzymes are cross-linked together, they're no longer functional, et cetera, et cetera. And although it may be possible to undo the cross-linkage by some advanced technology, we don't have that technology yet, and I don't think anyone's actually analyzed that problem yet, whereas things have been revived after cryopreservation all the time. So I think that by preserving the vast majority of the molecular inventory, you're sort of making a bet that that's more important than just preserving structure alone. And I think that's the right bet to make at this point. But that's my opinion.
Okay, thanks for that. And then last but not least, does RF warm liquids at the same rate as the rest, probably meaning the rest of the tissue?
Yeah, I mean, it's more or less uniform. RF warming does depend a little bit on the nature of the tissue. So for example, in the kidney, the inner medulla has a higher fat content than the cortex, and fat warms less effectively with RF energy than water does. So cortex will tend to warm a little bit faster than the medulla, and the medulla is actually the area that requires warming more than the cortex. The cortex is pretty well stable already. However, the great virtue of RF warming is that it's self-limiting. So once the cortex begins to get up to a temperature that might be dangerous, it actually warms less effectively. It gives time for the medulla to catch up. So it's not really much of a problem. And we think that we could scale this technique up even to human-sized organs. So even though it's not completely uniform, it's uniform enough, we think, to get the job done. And as I alluded to before, maybe marrying this technology with nanowarming would be a good idea, because nanowarming allows you to warm straight up from below TG without that pre-warming step that we had to go through, which causes nucleation. But it doesn't have the self-limiting feature, that safety valve at the end, when you're starting to get up to temperatures at which the CPAs may start to become toxic. So it might be that the best technique is to start off with nano and then kind of switch over to RF as you go along. But we will see. We will see.
Right. Thanks a lot. One more question for Adam. Do you think it could be useful to apply machine learning to predict toxicity?
Yeah, I do think that would be interesting. We're getting to the point where we can generate, I guess, large enough data sets to start thinking about machine learning. We haven't taken that step yet, but it's something we're considering. I think if we can come up with a model that is more related to the mechanisms of CPA toxicity, that will ultimately be more useful. So we're trying to attempt that first. But yeah, machine learning is a great idea.
Awesome, perfect. We're going on with Joao. And by the way, for all the other attendees who still have questions, please send them in, because we're just, since there's a large amount of questions, it got relatively difficult to see which question has been answered and which hasn't been. So if any question you ask hasn't been answered, feel free to send it again, and then we'll see it more easily, or come up with new questions, of course. But now a question for Joao. In your opinion, does the 2019 Yale study, in which researchers revived the disembodied brains of pigs after slaughter, open up any new avenues for brain preservation research?
So I think it gives, it was a very, it got a lot of attention, and it got a lot on the news. So from that perspective, I think it's interesting in generating interest and generating awareness. I'm not entirely sure how much it teaches us that will advance. I guess it could be a potential paradigm, a potential model, but in terms of basic knowledge, I'm not so sure it teaches us that much.
Right, and then another question. You discussed a bit that some of your students, or students in general, are less interested in cryopreservation than other adjacent topics like longevity and life extension. What, in your opinion, would awaken the interest of more students for this area?
Well, that's the great question. I mean, that's what I would like to know. If I knew an answer, I'd be doing it already. I guess the issue, and some of these topics have been touched upon during the conference, is that, something like cancer is perceived as unjust, unfair. If someone, particularly a young person, dies of cancer, that's unfair. We should do something. There's studies on where do people donate money to. So, for example, lung cancer gets relatively less donations, because it's kind of this perception that if you get lung cancer, it's because you smoke. So, it's fair. On the other hand, we have a very strong perception that childhood cancer is unfair. So I think that would be the main reason. But I don't know how to change it. I guess the point is showing that what I try to do when I give a talk on cryonics or cryopreservation is say, look, there's a lot of young people who have died or have diseases that if we had medical biostasis available, they would be saved, young people. So I try to focus on the young patients and the young individuals being cryopreserved. That's what I try to do, but I'm very open to suggestions if anyone has any.
Right. I'll let you know if I come up with something. Next would be questions for Eric. How many cases have you handled so far? How many for CI? How many for Alcor? How much work are you doing?
Well, collectively, our team members have performed over 100 standbys, stabilization and transport. So, this year we've done 5 of them. Most of them have been with Alcor, and we just did one in Portugal, Cryonics Institute,
patient. So, we're very proud that we have vast experience with both organizations, and we were able to perform the SST pretty much all over the world. Obviously, with the COVID-19, that's put a little damper on things, but we're always looking for new ways and new opportunities to be able to spread our knowledge. And we've done some training with SA also when it comes to FCP. So we're trying to enhance everybody's knowledge when it comes to standby stabilization and transport.
Right, as a follow-up question for that, the question is the recent case in Portugal. Portugal, of course, is not necessarily known as the most easy country for cryopreservation. Was this a case that is easily handleable, or was it successful in quotation marks under the given circumstances?
Under the given circumstances, it was very successful. We were presented with many challenges. Mainly due to COVID-19.
Right.
Even for us to get out of the United States into Portugal was very difficult. We had to, in fact, we went to the airport twice. We were turned away initially. And then our second challenge was getting the patient out of Portugal back to the United States. And we had to become very creative and had to actually drive to Brussels in order to get the patient out of Europe back to the United States. So a lot of challenges, but given those challenges, we were able to overcome, and it ended up being a successful case.
Perfect, good to hear. All right, going on for questions with Robert. Robert, do you have any sympathy for the view that there's more to someone's consciousness than memories? Is there a risk that the process might preserve memories, but not the original consciousness?
Sorry, do they have any other component to that question, or
no,
is that the whole question?
So the question, word by word, is: does he have any sympathy for the view that there's more to someone's consciousness than memories? Is there a risk that the process, I guess, cryopreservation or stabilization or glutaraldehyde stabilization might preserve memories, but not the original consciousness?
Well, so again, I'd point to DHCA, right? I think that certainly it could have been the case that we're constructed in such a way that if you cool someone down or you stop that continuity, that it erases someone's mind or otherwise impairs them. But it happens to be the case that that's not how people work. So, without that information, you certainly could, I'd have some sympathy for that hypothesis, right? But I don't think it's supported by the evidence. It's kind of surprising that that we work that way, and it didn't have to be that way, but it does happen to be that way. And then beyond that, there's this idea of this simulation hypothesis. Not that we're a simulation, but that in general the universe is computable, and essentially the universe being computable is equivalent to our current empirical physical models of the world. So if you're saying that there's something that wouldn't be retained by a proper process like this, then you're essentially saying there's something that's non-physical that you might be missing. And I don't have sympathy for that because there's no evidence for it.
Right, yeah. Then another question would be, if I create a checksums of 2 books and then burn them, will this will show a difference, but will not preserve the books themselves, right?
Okay, so like, let's say you take a 256 byte checksum of a book, right? Well, then that's 256 bytes. And so it's got a vast space of possibilities, but compared to the books themselves, it's much lower. So that you could say the bandwidth of that channel is insufficient to contain the books. And in fact, there's going to be millions of books that all sum to the exact same checksum, simply by the pigeonhole principle. You only have so many slots in the checksum, and you've got way more possible books. It's hard to find them. Now, one thing that does do is it does defeat this idea of saying, if I had 2 books that differed by just 1 character, then those checksums still are going to differ, right? That's sort of like saying if I had two brains that, you know, differed by one synapse, right? You cut out exactly the spot, that one synapse that differed, you know, and say, well, that's my preservation, right? This partial brain preservation. Well, yeah, that would maintain that one difference. But we're trying to make an argument that this preservation works in general. I'm not saying that if you can preserve one single difference, that means that it does work in general.
Right.
If you can preserve one single difference and there's no reason why that difference is special, it's more likely that it works in general.
Mm-hmm. Yeah, indeed. Another question for Ramon. What experiments do you have planned for the future?
Hello?
Yeah, we can hear you just fine.
Yes, now we are focusing on the heart mainly. So we are trying to do heart preservation, heart vitrification. In this, let's say that is the biological, from the biological side, and from the, let's say, engineering side, we are very concentrated on working with ultrasound, high intensity focus ultrasound.
Okay, cool. Then general questions for everybody, whoever might want to answer. What do you see as the biggest scientific challenge in the short to medium term? Maybe raise your hand, and then we're gonna, if you have an opinion, then we're gonna put you up. Anyone? Greg, go ahead.
Okay, I guess I would just say that the biggest challenge is to demonstrate brain viability. We're going to have to start adding and removing cryoprotectants and showing that the brains can take it and find out how far we can go. Presumably, low levels of cryoprotectant will not be a big problem, but there'll be a challenge in figuring out how far you can go and how you decide what endpoint is appropriate, because you can't wake these things up and necessarily get normal electroencephalogram readings off of them without worrying about ethical issues such as distress and so forth. So even if the brain is unable to tell you that it's not happy within circumstances, it may be. And so you have to come up with a variety of techniques that will be convincing, you know, that the brain viability is there, but that will not cause people on your animal committee to reject the experiment. I think we can do that. We're going to get into that next year. It's a more difficult problem than just transplanting a kidney, but it's very important for this discussion.
Right, awesome. Next question for Jacob. Jacob, with the ambulance that you presented, do you plan to outfit it for field cryopreservation or cryoprotection as well?
We can outfit it for any type of protocol. We're planning to start with just some basic field cryoprotection outfit, but we could outfit it for ECMO or for complete whole body vitrification approaches. The benefit is, of course, that the vehicle has rail tracks on all the sides of the vehicle, so we can reposition all the equipment whenever we want and install any types of new developed equipment which may be required.
Yeah, sounds good. One more question for David. David, since, of course, due to running London Futurists and so on and being in that community, you're in contact with a lot of people who are interested in this topic in general. How much interest do you see for cryopreservation specifically?
It's a topic that has often had small audiences at my meetings. So it's a reflection of the fact, I think, that people are maybe embarrassed to be showing an interest in this. And it's a shame that that's the case. So also it's a topic that maybe there's been less of a sense of visible progress. And perhaps if there's more visible progress, perhaps if there's something new to point to, then I think there will be more interest. Now, I've seen some interesting new angles in this event today. I think one thing I've learned is the possibilities that China may be leading possibly the next wave of cryopreservation. We have been saying collectively that one of the drawbacks is lack of funding for key research. But what I've heard is, and it's still a bit of a puzzle to me, a pleasant surprise, that the Chinese government is supporting the work that's being done by Yinfeng Bioengineering. So it's possible that just as China is investing very heavily in machine learning and deep learning and driving forwards artificial intelligence, possibly similar transformation might take place there. So that's one way in which there might be some sense of possible breakthroughs and faster progress. And in that case, again, I think an audience may be more interested.
Perfect. Thanks for the insight.
Aaron has something to say on that, I think.
Last but not least, one question for Francesca, who is not in the call. All right, then we have answered all the questions that are there for now. First of all, I would like to thank the speakers for their talks and for the insights that they've given. Thank you so much. I would also like to thank the audience for attending. Hopefully, all of us will be able to see each other and get to know each other in person next year in Zurich. I would also very much like to thank the team that helped me organize everything this day, Matilda, Renu, and then also Dimitris. Thank you so much. Of course, last but not least, I would also like to thank my colleagues at EBF for supporting everything and setting the foundation up together with me. So great work there. Just a few notes maybe to end. Tomorrow we have a few more community discussions which will be held on Zoom. Those are mostly about local topics in Germany, in Spain, in Portugal, and so on. If you're interested in any of those, if you check out the second day of the conference, which is called Community and Workshop Day, on top you see, or as the first event basically, you see a Zoom link which you can join during the indicated times if you're interested in one of the topics. And of course, next year at Biostasis 2021, we will organize a full day, 2-day conference. But this day we mostly held it to 1.2 days or so due to the fact that online conferences get quite tiring after a long time, or like after a long day of online talks for the audience. So hopefully we will be able to see each other again next year. Check out the page, which will be updated with information about Biostasis 2021 within the next days. In the end, thanks everybody for attending. See you next year, and until then, goodbye and have a great weekend.